Fondazione. Hello, everybody. Please take your seat. I see a number of friends who are taking their place. It's 8:30. I'm Giovanni Frisoni. I'm directing the memory clinic in Geneva, but I'm not Swiss, by birth at least. Despite I try to start the morning at 8:30 sharp. It's my duty to chair part of this morning's lecture, and it's my pleasure to introduce the first speaker, who is Michael Enneca. I think Michael does not need any introduction, but just for those who are inattentive, you may know that Michael is probably the top world expert on neuroinflammation in neurodegenerative disorders. And he's currently director of the Luxembourg Center for Systems Biomedicine at the University of Luxembourg. And the place of systems biomedicine nicely fits the complexity of neuroinflammation. But I'm sure that Michael will help us untangle some of the complexity of it. Michael.
Thank you, Giovanni. Very nice introduction. I think there are many experts on that topic, and I just want to raise some questions and discuss with you whether or not sterile inflammation actually contributes to neurodegenerative disease. For this morning, I will restrict most of the part to Alzheimer's disease, but we can expand this during the discussion. Now, living through a pandemic, we all know and realize that we're constantly exposed to environmental challenges by fungi, by bacteria, and of course viruses. And without our innate immune system, we wouldn't make a day, so to say. And I'll give you a little video here. This is an innate immune cell hunting for Staphylococcus aureus. And you can see that it needs to take a lot of clever decisions. It doesn't attack surrounding cells. It follows it constantly. And obviously is equipped with surface receptors until he can corner it, take it up by phagocytosis, and degrade it. So the innate immune system is our first line of defense. And these cells do not only exist in our bloodstream, they exist in the brain. We call them microglia as resident innate immune cells. In the lungs, alveolar macrophages, copper cells, mesangium cells in the kidney, Paneth cells in the gut, and of course bone marrow macrophages. They all protect these organs from infection, but they contribute strongly to their structure and function. And for microglia cells, it's about 100 years ago, Pio del Rio Ortega has identified them and described them in detail for the first time. And who is interested in this historical part, I would refer to a very elegant review written by Amanda Sierra and published in Glia some years ago. Now, in a nutshell, I think that most people in the room would agree that Alzheimer's disease is fueled by amyloid beta deposition coming down the APP processing pathway. And you see here that there are various ways of the brain to get rid of amyloid beta monomers at best through the glial lymphatic drainage into the CSF, but also to a great extent by microglia degradation through the release of IDE, for example, or phagocytic clearance. If that system gets out of balance, these monomers accumulate, as we know, into oligomeric structures and eventually deposit within the brain parenchyma. And this deposition starts an inflammatory reaction characterized here by CD68 positive microglia cells and GFAP positive astrocytes surrounding these deposits. And ultimately, the disease is characterized by the occurrence of neurofibrillary tangles. Now, this is showing you a cortical section stained with Congo red, obviously from an Alzheimer case. And long before Congo red has been actually used to stain human brains, it has been developed for something completely different. So Paul Bötticher in Leverkusen in 1883 was generating this substance as a stain for textiles, especially cotton. But because the Rhineland in Germany is way more rainy as Milan, it turns out that it would not stay in the textiles, so the company was not interested. And they put it back on a shelf, and actually he took it back to the little company in Lodz in Poland, where his father had a small pharmaceutical company, and he then worked on that. And by accident, he threw a vial to the ground containing Congorat, leading to the finding that it would stain the lab ground in a strange fashioned way. And that was the initiation of an observation that it would be a great stain for bacteria. So he went to Berlin and licensed it as a bacterial stain in '84. Now, what is the commonality between a bacterial surface and a human brain? In fact, it's the occurrence of beta sheet structured amyloids. And this has been found by Matthew Chapman in 2002 in this Science paper. And Chapman and colleagues showed that a variety of bacteria, which are very common to us, including Escherichia coli, Staph aureus, Salmonella typhimurium, and others, carry beta sheet structured amyloids on their surface. So this means that a myeloid cell, by nature, over evolution for millions of years, has been taught that a beta sheet structured amyloid is nothing else than a danger-associated molecular pattern. And these look exactly the same in the brain of human AD cases as on the bacterial surface. Now, nature has equipped myeloid cells, including microglia, with pattern recognition receptors, which ligation causes two major effects. One, which is producing an inflammatory reaction, including the output of cytokines, chemokines, nitric oxide species, other ROS, and complement factors. And at the same time, these cells stop doing a lot of senseful things for our brain, such as reduced clearance from debris. They have a reduced production of neurotrophic factors. Some of them are key to our neuronal function. And they have reduced synaptic scaling and pruning. Now, there are way more danger-associated molecular patterns arising during neurodegeneration in the brain of various neurodegenerative diseases. This is giving you an example. Chromogranin A, normally kept in vesicles in neurons. When neurons undergo degeneration, they release those vesicles. And chromogranin A has been already shown by Toupinot and colleagues from Strasbourg in the mid-90s to be as effective in inducing an immune reaction as bacterial cell wall components. Now, another example are S100 proteins. And here you can see MRP14, which is a key protein from this family. And halving this protein by knocking it down in an animal model, an APP, PS1 transgenic mice, kind of suppresses cytokine production, at the same time increases the capability of microglia cells to take up amyloid beta and degrade it. Now, this all comes on our genetic background, and over the past decade, we have experienced the identification of a high number of risk variants by genome-wide association studies. And a good part of these GWAS hits are actually important for innate immune signaling. TREM2, CDK, CD33, BIN1, C1q, and others. Like approximately 1/3 or more are really residing in innate immune signaling pathways. And this comes not only on a genetic background, it also comes on a life exposome. There's a couple of risk factors or risk behaviors of which we know that they lead to a rise in systemic cytokines. A good example is obesity. We know that midlife obesity is a risk factor, and obese tissue basically is 10% of activated macrophages. So your fat tissue, 10% of it is activated macrophages, constantly releasing low amounts of cytokines. We know that periodontitis is a risk factor. We know that aging itself by senescence is a risk factor, but senescence is leading to the release of a senescence-associated secretory profile of cells. And this senescence-associated secretory profile contains a lot of cytokines. Now, brain trauma, no doubt, and systemic infection and repetitive exposure to inflammatory conditions in our circulation is a strong risk factor too. Now, this is a very recent paper, I think it's 3 weeks ago, where the microglia regulome has been described in a very, very elegant Nature Genetic paper by Kosoy and colleagues. And what they did is they used an attack-seek approach to do transcription factor fingerprinting in over 140 isolates from human brains. So only microglia were isolated, and neurons and astrocytes. And then they used a way to look which of the transcription factor footprints would actually activate enhancer regions of the Chivas risk variants we know. And ultimately, they compared that in a computational way to sequencing data, single nuclear sequencing data. And the outcome of it was that if you look to neurodegenerative diseases down here, Parkinson's disease would be mainly driven by changes in neurons, and ALS to some extent by neuronal changes, but Alzheimer's disease in particular, as you can see in the lowest line, was highly driven by Chivas risk variants in microglia cells, or their function and transcription factor footprinting suggests that the microglia regulome is key, but through action of microglia and not neurons. Now, that may over-describing it a little bit, but it points to a fact that microglia cells take a key position in the pathogenesis of Alzheimer's disease. Now, if we study these mechanisms now, we often have to rely on rodent models and murine models, and for that we have to be sure that we investigate pathways that are conserved between species. And it's very important because only approximately 30% of our adaptive immune system and 40% of our innate immune system are conserved between mouse and man. So this is work by Erik Bodeke and colleagues from Groningen, and Erik has undertaken the effort to compare a lot of transcriptome data sets and identify a core human signature for microglia. So all these pathways are really important in human microglia, isolated from the cortex. And in the core of the core, you find the inflammasomes. And the most abundant inflammasome in the human brain is the NALP3 inflammasome, a highly regulated inflammatory key innate immune pathway. The inflammasome requires ligation of toll-like receptors or of a cytokine receptor, starting the production of proforms of cytokines and NALP3 itself. And then there's a high regulation and various licensing steps which build the inflammasome together in a wheel-like structure and forming the so-called ASC spec. The ASC spec is the active part, which has the outward-directed caspase-1 cleavage activity. And there's the full cleavage of proforms of IL-18, IL-1 beta, and gasdermin D. The cleavage product of gasdermin D forms the outer pore, allowing for the coordinated release of these cytokines by an immune cell. And we started to look at that in the context of Alzheimer's disease. And here you see a microglia cell containing an ASC spec in a frontal cortex section of a human AD case. And of course, we would not be able to judge whether that is a very active system. So we started to blot for cleaved caspase-1, and doing so in age-matched controls and AD cases, we found that there is a strong upregulation of this system. So massive activity of NALP3 inflammasome in AD patient brain. And I don't show this here, but it's also in early AD and MCI cases. Luckily, this is completely reflected in the transgenic mouse models, in this case by APP-PS1 delta E9 mice generated by David Borchert and Joan Jankowski. And you can see in an age-matched fashion there's upregulation of NALP cleavage. And there is production of interleukin-1 beta in these brains, which is suppressed when we knock out NALP3. So knocking out NALP3 in this mouse model, which is highly loaded with amyloid beta plaques, and in red you see the IBA-positive microglia attacking the amyloid beta deposit, like knocking down NALP3 would double the capacity of microglia cells to take up amyloid beta. And this is a new assay where we quantified the absolute phagocytosis of microglia by injecting animals with methoxyXO4, a dye which is able to label amyloid beta in vivo, and I'm coming back to that. The overall effect in transgenic mice was an 80% reduction in very old mice. So these mice, when we analyzed them, were 18 months of age, and you can see that there is a strong reduction of deposition. For the biochemical quantification, I refer to the paper. There is a 70 to 80% reduction of SDS and repa fracture of all the amyloid beta species. Luckily, this is not confined to modifying A beta, and there's a strong protection from spine density. And not only from spine density in a morphological way, there's also preservation from APP-induced suppression of synaptic plasticity, as evidenced by these measures of hippocampal LTP at the Scheffer collateral. Now, it would be bad if that would be just at the electrophysiological level, but it was transferred to the mouse behavior. We performed the very famous Morris Watermaze test. You can see that the background strains, wild type and knockout, they learn over 4 days the task very well to find the platform. They consolidate that information, while at the same time the APP-PS1 transgenic mice don't learn much. Then APP-PS1 knockout mice learned almost as good and consolidated that information as compared to the background strains. And in the day 9 memory test, they performed perfectly well and found the platform and confined their search strategy to the quadrant 1 where the platform had been. Now, sustained inflammation and sustained inflammasome activation in microglia cells kills these cells and leads to microglial pyroptosis. And pyroptosis of microglia cells, as well as of any macrophage in the body, would lead to the release of the ASC spec. So we looked at this phenomenon in the context of Alzheimer's disease, and you see here that we find evidence for ASC spec release in human tissue as well as in mouse tissue. And then we started to modify this system experimentally, finding a way to induce microglial pyroptosis, in addition quantifying it by microscopy or by FACS sorting. And I set out to prove that amyloid beta would be a fantastic inducer of microglial pyroptosis. And actually, I tortured a number of PhD students and postdocs, and they all failed. And so I had to do it myself, and I failed even better. But I was way more luckier because since my lab has used up all the unlabeled amyloid beta, I had to use the TAMRA-labeled amyloid beta. And that allowed us to look in the dish. And it turns out the moment the amyloid beta would face an ASC spec released by a dying microglia cell, it would very rapidly bind to it. And there's a movie now, and I want to actually point to this cell here, which will undergo pyroptosis, releasing an ASC spec. And you see that the red TAMRA-labeled amyloid beta forms a halo surrounding this very quickly. On an electron microscopy level, you can see here that this is A beta alone, this is ASC alone, and ASC acts like a magnet for the amyloid beta peptides. Makes no difference if we study amyloid beta 1 to 40 or 1 to 42. So we hypothesized that when microglia cells would undergo sustained inflammation and experience microglial pyroptosis, the binding release of the ASC spec would bind to amyloid oligomers and allow for deposition. So doing the first assay to that was looking at cross-seeding, and this is an assay that shows you the conversion of a monomer to an aggregate. You see in green the aggregation curve over time in the absence and then in the presence of these ASC specs, and I hope you can see this enormous shift to the left, which is a sign for cross-seeding. In the blood system, it looks exactly the same. Here we see in the absence of any ASC specs, we see hardly any fibril or oligomer formation after 24 hours. And this is increasingly the case when we raise the concentrations of ASC specs. And interestingly, this is A beta 1 to 40. And you can now hypothesize whether ASC spec binding converts A beta 1 to 40 biochemically in a molecule that behaves like A beta 1 to 42 in terms of A beta aggregation propensity. Now, confirming this in a co-sedimentation assay, in collaboration with Jochen Walter and Satish Kumar, we found that if you would look at the mixture of amyloid beta peptides alone, together with ASC or ASC alone, most of that would actually be present at time point 0 in the supernatant and completely found back after 6 hours in the pellet. Now, this indicates that ASC would drag the entire amyloid beta peptide into a kind of artificial plaque-like structure, which prompted experiments in mice that showed if we do co-immunoprecipitation for ASC and A beta, there would be co-binding in transgenic mice. And importantly, when we looked by immunohistochemistry, we saw that the core of the plaque would be made of ASC, and the surrounding 6E10 positive material would be A beta material. So that led to the hypothesis that ASC would be a seeding factor. So we employed another model very nicely introduced by Matthias Jucker and Larry Walker. And using that model of pathological A beta spread, we started at 3 months and looked at 8 months in this mouse model. And this is the next slide is probably the most important slide. So the model itself has a problem because synthetic amyloid beta is not seeding. And here that to us suggested that either the amyloid beta they used for injection was different to the synthetic amyloid beta, which can well be the case, or there are cofactors required. Now, this is an important experiment in my view because it shows you that if we inject APP-PS1 derived brain lysate versus wild-type brain lysate in opposing hypocampy, we get pathological spread as evidenced by Matthias Jucker's lab, published in Science in 2008, and we completely repeat and verify these data. Over here, you see that you have this massive increase in oligomers and fibrils and in total A beta as well. Now, we do the same experiment in non-injected mice. You see that wild-type brain lysate does not modify the experiment at all. And now we did the very same experiment in mice. We're just lacking ASC. And ASC has only one biological known function, which is immune signaling at the NAB inflammasome. But if we knock it out, we completely lose the spreading phenotype. And we also lose here, like in the test tube experiment, the occurrence of pathological oligomers and fibrils. Now, I didn't trust that experiment a lot, despite we published it well. So we started to generate a mouse, which is APP-PS1 transgenic, so we can label in blue amyloid beta by methoxy-XO4. We crossed them into CX3, CR1, EGFP mice, so all the microglia cells are green. And we had a knock-in ASC-M cherry. So we see all the ASC specks, which are being released by dying microglia cells as red dots. And what you see is our setup to photo and laser scanning microscopy. We use a non-anesthetized awake mice. They walk on this little treadmill. They're happy. They like it. And they conduct a spatial navigation task during this procedure. And we can actually visualize now microglia pyroptosis in this movie and the release of ASC specks. And you can already see that there is methoxy-XO4 binding surrounding these ASC specks. But if you take the green channel out in the next movie, please focus on the blue amyloid beta deposits. And you'll see already that the ASC here traps amyloid beta at the vessel wall. But please focus on the blue dots. These are A beta deposits, and you will find not a single one which does not contain a red ASC speck. So this is the next one over here, also the smaller ones. This is a non-injected mouse. It has never been touched. It lives a happy mouse life. Still, every core of every plaque is made of ASC. Now, this, to my end, proves that at least in the mouse models we're working with, the role for deposition is tightly connected to the occurrence of inflammation and microglial pyroptosis. Now, what about humans? As a neurologist, I'm more interested into the human side. And this is interesting because you see that co-immunoprecipitation here is actually showing binding of A beta to ASC in humans too. And then we set out in collaboration with Banner Health to look at a high number of samples. And we splitted the fluffy fiber compartment from the core of the plaque. And then looking at MCI cases, we found that ASC is already co-localized to amyloid beta in the core during MCI. The only difference towards AD is the occurrence of more A beta in the fluffy fiber compartment. And so all the plaques we can see in humans have an ASC core and 6E10 positive amyloid surrounding it. Now, Unfortunately, that's not a finding by my laboratory. If you see this microglia cemetery here, if you see this microglia cells here, there aren't any happy microglia cells. They undergo death. This is a frontal cortex section. These are released as specks. But Alzheimer and Franz Nissl in 1911 already painted this microglia suffering in a publication here. And you can find all these microglia corpses back. They painted already more than 100 years ago in this section. Now, one of the remaining questions we had was, is it a connection between A-beta deposition and tau fibril formation within neurons? And so is there a role for innate immunity? And proving this, we used a model by Guts and colleagues published in Science, and another injection model showing that if we inject APP-PS1 versus wild type lysates in these tau transgenic mice, would massively induce tau pathology here versus here, but not in the case we don't have ASC or NALP around. This is the quantification. To sum this very quickly up, is the mechanism is in the paper, so I don't have time to allude to this, but what we found is that microglia cells that would undergo pattern recognition ligation and release cytokines including IL-1 beta and IL-18 would, via the IL-1 receptor on hippocampal neurons, via MyD88 and MEC1/2, activate CAM kinase 2 alpha, leading to the hyperphosphorylation of tau and ultimately paired helical filaments. Now, this mechanism is active in mice, and we also provided evidence in humans. But why we think that this could be potentially interesting as a treatment? This is finding from a paper published earlier this year by Frédéric Brosson and my group, and what we used was the DELCODE cohort, prospective cohort of the German Center for Neurodegenerative Disease. And we have about 400 CSF samples of patients which we now follow up on an annual basis by MRI and neuropsychology. It turns out that people which belong to the healthy control group here, but are tau positive, have very high levels of tyro and very high levels of AXL, shattered products from microglial surfaces. And you can also see that this phenomenon burns out in a way that it doesn't become statistically significant here between Alzheimer cases that are tau negative and tau positive. Those are probably not real Alzheimer cases. But when we look to these, we find that the occurrence of high level of tyro and AXL is actually, in the early cases, connected to brain protection and not to degeneration. And these are the regions which were found to be less atrophic over time, over a follow-up of 5 years. We now have the 7 years data. You see that it's the temporal lobe, it's the neocortex, which is protected by high tyro and high AXL levels, in contrast to tau or A-beta, which is correlated with brain atrophy in these patients. Now, it's not only a preservation of the brain morphology, there's also preservation of cognition in these cases. You see that high tyro level or high AXL level patients do not have the same cognitive trajectory and don't show much decline over the subsequent years as compared to those with low levels. Now, the point is that we have been trying to identify the involved mechanism and looking to many innate immune signaling pathways. And the only mechanism we found was the suppression of the NALP inflammasome. What we see here is that tyro overexpressing cells or ligation of tyro is completely suppressing the occurrence of cleaved caspase-1, pro-IL-1 beta, and IL-1 beta. So the entire NALP inflammasome is massively suppressed in human microglia cells undergoing tyro ligation. Now, with this, I sum up. I hope I could convince you that the innate immune system is actively contributing to neurodegeneration. GWAS identified risk variants and lifestyle factors may modify neuroinflammation in the degenerating brain. The NALP3 inflammasome is activated in Alzheimer's disease and respective mouse models, and importantly, the pathway is highly conserved between both species. NAB inflammasome activation accounts for reduced phagocytic clearance of amyloid beta, seeding and spreading of A-beta pathology by ASC-SPEC seeding, and A-beta-induced tau pathology. The TAM receptor findings from DELCODE and the analysis from human microglia experiments using tyroligation suggest that inhibition of NAB3 is protective during early stages of neurodegeneration. And with this, I thank all the people of my group. Most of that work actually was performed during my time in Bonn at the German Center for Neurodegenerative Disease and at the University of Bonn. And I'm also thankful to my great collaborators and, of course, the funding associations paying all that work. And I'm thankful for your attention, and I hope you have some questions.
Thank you, Michael. Fascinating lecture. You've persuaded everyone that the traditional notion that neuroinflammation comes at the end of the process, at the end of the cascade, is probably the other way around.
I won't claim that. I'm not so sure if it's important to answer the N and HAC question. I think it's important to find drivers of disease. If we can identify mechanisms, and I believe that neuroinflammation is one, then we probably have ways to interfere and halt it. So the ASC-SPEC seeding, especially in the Moran models and in the in vivo model, which has never been touched, strongly suggests that the beginning of the seed, at least in this mouse model, is the death of a microglia cell releasing an ASC-SPEC. Potentially, it could also be an invading innate immune cell, but it has to be either a macrophage or a microglia cell. And there's no other way an ASC-SPEC gets there. And we really didn't touch these mice anyhow. They're living a happy life. So the finding that in 90% of human A-beta plaques, you find an ASC-SPEC in the core, and the amyloid beta surrounding it suggests that there might be a mechanism in place very similar to what we observe in the mouse model. And you could speculate now whether amyloid beta is our enemy or whether the ASC-SPEC formation is a good target.
I believe that we can no, we cannot allow questions because we are beyond time. Michael, thank you. So let's move on to the next speaker. Next speaker is Dr. Jeffrey Cummings, who also, I believe, does not need any introduction. But again, just for the form, let me remind you that Jeff Cummings is the person in the world who probably has the greatest visibility on treatment for Alzheimer's disease. Whenever an intervention for Alzheimer's, a clinical trial starts to show up in the scenario, Jeff Cummings is aware of it, and he dissects the design, the trial, and maps the trial on the larger scenario. Jeff Cummings was the director of the Mri Easton Center for Alzheimer's Disease at UCLA. Then he was director of the Cleveland Clinic Ruo Center in Las Vegas. And now despite he's no longer the director, he's extremely active in the field. And Jeff, please.
Thank you. It's always good to have your friends introduce you. That way you get these really nice introductions. Thank you, Giovanni. So it's my pleasure to be here. I've learned so much in the last day and starting this morning. So it's really been fantastic. I appreciate the invitation. So I'm going to talk about, at Giovanni's request, clinical outcomes in phase 2 and 3 clinical trials. And I'll go quickly through what we do, and I'll spend a little bit more time on what maybe we should do. Here are my disclosures, consult for a lot of companies. So I'll start with some orientation. I know not everybody here is an Alzheimer expert. I'll talk about phase 2 and then phase 3. going to emphasize the monoclonals because we are at a pivotal moment in the monoclonal development. And then I'll end by talking a little bit about how impressed I am with the Fondazione Prada and this convergence thinking approach. So just to say that I'm going to talk about the clinical measures, but the biomarkers cannot be dissociated from clinical measures, and we'll be hearing about the biomarkers next. Just a bit of orientation. Here's a paper that Philip Sheltons and I did a few years ago on the rights of drug development. So in order to succeed, the trial must have the right target, critical pathway, has to have the right drug, of course, with drug-like characteristics, the right biomarker, which we'll be hearing about later, the right population. I'll emphasize how that has proliferated recently, and it caused us or formed our reformulation of clinical trials. And then the right trial, which is well designed with appropriate outcomes. That's what we're discussing in today's lecture. Adequately powered, quality conduction, representative population. And I'll just make the point that this is drug development 101, and yet it's frequently violated. And if you went through the 140 clinical trials, or 172 clinical trials that are currently going on with 140 drugs in the pipeline for Alzheimer's disease, you would find that many of them could not check the boxes on the 5 rights, and they'll fail. Because you cannot get to a successful drug development program without knowing these things about your drug and its application in clinical trials. Here's the revised thinking about Alzheimer's disease. Of course, that we start with normal aging, and then those at risk will go on to preclinical AD, who have normal cognition but demonstrable abnormalities in the brain with amyloid imaging or CSF. That progresses to prodromal AD or mild cognitive impairment, which then progresses to AD dementia, changing our orientation. When I was in medical school, of course, Alzheimer's disease was dementia. Now we know that most of Alzheimer's disease is not dementia. It precedes the dementia portion of the illness. I'll just make the point in the bottom part of this slide that then we have different kinds of trials. So in a normal person who has not yet have amyloid in the brain, that would be a primary prevention trial. And this is really how we started the conference yesterday about the healthy lifestyle. That's primary prevention and extremely important. Then we can go to secondary prevention. That means, of course, that the person is asymptomatic. But we can begin to intervene to try to delay the onset of cognitive decline. So that's a new kind of trial that we haven't done before. Not a single one of those trials is completed yet because they just haven't been done. They are in process. I'll tell you about those. Then you have treatment trials in MCI to slow the progression to dementia, and then you have treatment trials in Alzheimer's disease dementia to try to slow progression. Well, what do we have to know at the end of phase 2? Well, we have to know that target engagement has occurred, or that there's proof of pharmacology at least. We have to have some sort of clinical indication. These are usually not now full statistically significant differences between drug and placebo. They're usually effect size and directional analyses. We have to know the doses. We have to know the safety. And from phase 1, we have to know the maximum tolerated or maximum feasible dose. This is the most common cause of anticipatable regret in a development program, is that you get to phase 2, it's a negative trial, and you don't know whether you didn't test the right dose or you didn't have the right drug. You must know blood-brain barrier penetration, and again, too often, drugs are advanced to phase 3 without this knowledge. The clinical measures then are determined by the population. Most phase 2 trials in disease-modifying agents are in mild cognitive impairment or mild AD dementia. That actually represents 40% of all trials currently going on, whereas symptomatic trials for cognitive enhancers are typically conducted in mild to moderate dementia. There's a difference in our approach here. We use the same clinical outcomes, but we approach them in slightly different ways. In a disease-modifying trial, you're dependent on biomarkers. In a cognitive-enhancing trial, you're depending on the outcome being the clinical measure itself. One of the things that we're starting to see in phase 2 are computerized assessments. These might be more sensitive. I think that has not been conclusively shown yet, but we do see that directional change. So in phase 2, if it's a cognitive enhancer, it's a short trial, 3 to 6 months. If it's a disease modifying, it's 12 to 18 months. You see that we use different tools for the global, the CIBIC Plus versus the CDR. We do use the ADAS-Cog. I'll come back to that later, although there are alternatives that are now being used, like the Neuropsychological Test Battery, or NTB. For function, we use either the ADCS-ADL as a common instrument, or increasingly the Amsterdam ADL as a more contemporary instrument. Here's an example of a very well-conducted phase 2 trial by Big Pharma. This is Eisai's drug, BAN2401. This is lecanemab. So I'm showing you the phase 2 trial. It was 2 weeks ago that the phase 3 trial read out. And you see a successful phase 3 built on a very well-considered phase 2. That's how you get to a successful phase 3. So what were the unique features of this particular trial? It was one of the first trials conducted in the MCI, mild AD dementia population, requiring positive amyloid PET. So a very well-defined population. It was large. 854 patients in phase 2 is a very large trial. A Bayesian dose-finding strategy was used. You can't really see this, and I won't go to it in too much detail, but they started off with 5 doses, not knowing how much of a monoclonal should be given. They wound up very consistently through the Bayesian analysis with the high dose being the best dose. So they were able to make very precise dose conclusions that were advanced to the successful phase 3. The target engagement was shown by a dramatic reduction on amyloid PET. In fact, 80% of patients had negative scans at the end of the trial. They would no longer meet a definition for Alzheimer's disease. They continued to progress, by the way, just at a slower rate. And a novel outcome measure, we're talking about outcomes in this talk, was used called the ADCOMS, or Alzheimer's Disease Composite Score, and we see many more of composite measures being used as outcomes in phase 2 and phase 3 as well. So what do we need to know at the end of phase 3? Well, I like this target product profile approach, and this is based on the idea that you know what the label needs to be in order to guide prescribers. So once you know what the label needs to be, you can then design your program backwards to meet the requirements of the label. So you start off with the target product profile, which is the description in the label, and then you look at your program backwards to make sure that at the end of your program, you have the label that you want. And that label, of course, is going to include indication, efficacy, safety, etc. It's typically divided into minimally acceptable and the ideal results. The sponsors sometimes add additional things like, what does a competitive landscape look like? So the outcome measures, they'll be determined by the population of the study. So this is one direction of evolution of outcome measures, is that we have new populations that we've never studied before. So we have new measures that we've never used before. That's a risk, but it's a risk we have to take. Most phase 3 studies are conducted in cognitively normal individuals who are positive, who have positive scans, in prevention trials, or mild cognitive impairment, mild AD dementia, or mild to moderate AD. So a word about prevention trials. They're new because we haven't even been able to define this population before. A risk here, well, they're large. I'll make that point first. About 1,500 people in many of the trials. They run 4 to 5 years, so unusually long for Alzheimer's disease trials. And they're usually conducted in phase 3 after assessment in a symptomatic population in phase 2. So you start off usually with mild cognitive impairment. If you're successful there, then you back it up into a prevention trial of a cognitively normal person. 2 examples of that are with ASI, where the Clarity program is a phase 3 program in MCI, and that is going to the A345 program, which is prevention. That's the lecanemab program. In the Roche program with gantenerumab, it's starting with a phase 3 graduate program, which will read out in just a few months, actually 1 month. And they have already started the Skyline trial, which is the prevention trial. So you see in both cases, start off with a symptomatic population. If it looks like it's successful, then you go to the prevention population. Wouldn't it be great if we could prevent Alzheimer's disease? That's a fantastic outcome. They're high risk. The best way to have a successful phase 3 is to just do what you did in phase 2 if it was successful. So here you're required to not do that. So I won't spend too much time with this. Again, the outcome measures, the global are different if you're in a symptomatic enhancing, cognitive enhancing program versus a disease modifying program. We use the same cognition and function instruments that I showed you in phase 2. Here's a terrifying figure from Philip's group, again, showing you the variability on 2 of the instruments. The CDR sum of boxes score on the left and the ADAS-Cog score on the right. So you see why such large numbers are necessary to get a successful clinical outcome. Patients are having all sorts of trajectories. In general, if they're responding, they're getting better. In general, if it's an unsuccessful drug, they're getting worse. But you see how much variability there is, much less in the biomarkers, which we'll see. So what are some challenges? Well, Philip's group, again, and I helped with this paper, recently published a paper called The Clinical Trial is as Good as Its Outcomes, as emphasizing the need to move to more contemporary outcomes. What do I mean by contemporary? Well, the mini-mental, which we used to define our populations, 1975, the CDR, 1982, the ADAS-Cog, 1984, and the ADCS-ADL, 1997. I think it's time for reconsideration. Those are older tools than most of you in this audience, so it's time to rethink it. We need to integrate better measures. Computerized measures are coming in. Digital technologies are starting to come in, though slowly. Patient-reported outcomes are necessary. We need to know more, particularly in the prevention trials. What do patients want? And we need to answer that question, and we need to make that the trial outcome. So I just want to emphasize the monoclonals because we're at such a moment with them. So aducanumab was approved on the basis of an accelerated approval that was based on, again, dramatic amyloid lowering. Lecanemab has applied for accelerated approval based on the trial that I showed you. They also just had a very positive Clarity readout on phase 3, so that they will go to the FDA for review in 2023, very soon. The gantenerumab program, Graduate 3, will read out, you see, in October or November of this year. So we're talking about in the next few weeks. So we'll know by the end of this year whether that monoclonal also succeeded. Donanemab in the Trailblazer 2 program will read out in April of 2023, again, very soon. And the other Trailblazer programs will be also reading out in 2023. They have also applied for accelerated approval. The solanezumab program is also progressing. That will be the first primary prevention trial. So the first DMT has been approved for Alzheimer's disease, aducanumab. It's the first DMT, it's the first monoclonal for any neurodegenerative disease. The trial participants were early. The MABs make extraordinary demands on healthcare systems, and they exacerbate our difficulty with global equity. So a few main points. Phase 2 and 3 are competence building and part of the development program. Phase 2 provides the foundational data. Phase 3 develops the final data. The outcomes have evolved to meet the population, but need reconsideration. They've not changed with many advancing ideas. We're at a pivotal moment in phase 3 development. So I just wanted to finish with this idea about the foundation. Alzheimer's disease, of course, is one of the greatest challenges facing humanity. We have to acknowledge that. And meaningful treatment will affect millions of people and will change the course of history. Neuroscience is the platform on which this historical transformation will occur, and we are looking at it now. Previously unimaginable progress is being made in understanding Alzheimer's disease and in the foundation for advances in treatment. And the kind of deep convergent thinking that we've seen in the last few days and we see in the entire Prada program of human brains model is required, and that's what we're seeing. So thank you for that.
Thank you, Jeff. Thank you, Jeff, for this overview. Certainly the field is moving forward and the acceleration is impressive, and we're picking the fruit of 20, 30 years of work.
30 years of work or more, maybe 100.
Maybe 100. There will be a discussion at the end. So let's move on to the next speaker. Next speaker will be Anthony Shapira. Professor Shapira, I don't know if I will be able to give you an introduction such as the one that I gave to the previous ones, because you're from the Parkinson community, but I'm sure that many of you know that Professor Shapira is heading the Department of Clinical and Movement Neuroscience at UCL, Queen Square, the historical Queen Square Institute of Neurology in London, and he is Professor of Neurology at the National Hospital for Neurology and Neurosurgery at UCL. He's an expert on the molecular clinical aspects of neurodegenerative diseases, and he will tell us about the targeting the glucocerebrosidase pathway, which is basically the ApoE of Parkinson's disease, right? Kind of.
We shall see.
Thank you.
Thank you very much for that introduction. And of course, I would like to start by thanking Professor Comi, the Scientific Committee, and the Prada Foundation for their very kind invitation to come and talk to you this morning. My topic is targeting the glucocerebrosidase pathway. And I'm going to start by reminding you that I'm going to shorten glucocerebrosidase to GBA, and how this may be a paradigm for slowing down Parkinson's disease. Well, this is a long story in some respects, but a short one in others. So, Dr. Gaucher was a French dermatologist who first described a patient with the disease, the lysosomal storage disorder, that was to bear his name 140 years ago. And then it was Roscoe Brady who really did all the hard work in the United States, identifying, characterizing all of the pathway for glucosphingolipids and ceramide and glucocerebrosidase included. And he, incidentally, is the father of enzyme replacement therapy. But it wasn't until 1996 that Ari Zimran, a Gaucher doctor in Israel, first described the association between Gaucher disease and Parkinson's disease. Now, Gaucher disease is an autosomal recessive disorder due to mutations in the Gba1 gene. And it's part of a pathway which is rich in neurological disease. And the blue arrow points out the GBA enzyme deficiency, but you can see all the other neurological disorders in this pathway. Gaucher disease is a multisystem disorder. It can affect the brain in what are called type 2 and type 3 disorders, but the more common type 1 Gaucher disease has hitherto not been described with CNS development, but obviously that needs realignment. Gaucher disease is, as I've implicated, treated with enzyme replacement therapy relatively successfully. So the link with Parkinson's disease, where does that come in? Well, I've indicated that there was the anecdotal reports by Ari Zimran, but the story between Gaucher disease and Parkinson's disease was nailed 13 years ago by Ellen Sidransky in this very nice multicenter analysis of the genetics of Parkinson's disease and the presence of GBA mutations, identifying Gba1 mutations as the most common association with Parkinson's disease. So let's look at that a little more closely now. Homozygous or biallelic mutations of Gba1 cause Gaucher disease. But the asymptomatic carriers of just one GBA mutation would, one assumes, be literally asymptomatic and certainly not get Gaucher disease. Well, that's all very straightforward, but what is interesting is that the risk of Parkinson's disease is virtually the same between homozygous biallelic cases of Gaucher and heterozygous asymptomatic carriers of Gba1 mutations. So in a way, the Gba1 mutation is autosomal dominant for Parkinson's disease, but with reduced penetrance. In fact, only 30% of those that carry a Gba1 mutation will develop Parkinson's disease by age 80. However, it's interesting that this is the same order of magnitude as the other autosomal dominant gene LRRK2 with variable penetrance. If you look at your Parkinson's disease clinic and you fully sequence the Gba1 gene, 10 to 15% of them will have variants. So this is by far the commonest genetic association with Parkinson's disease. And the same mutations are associated with dementia Lewy bodies and with REM behavior disorder. There are two particularly common mutations, if I can put it that way, the N370S and the L444P. The N370S is particularly common amongst the Ashkenazi Jewish community. And whereas in the Parkinson cases, generally 10 to 15% have Gba1 mutations, in the Ashkenazi Jewish community, 25% of Parkinson cases have Gba1 mutations. So what happens with Gba1 carriers? Asymptomatic individuals, they don't have Gaucher disease. They may have a family member with Gaucher disease, but they're normal for all intents and purposes, but they're at risk of Parkinson's disease. Approximately 20 to 30 times increased risk. And it's interesting that these individuals, before they develop the clinical features of Parkinson's disease, can exhibit some of the prodromal aspects of Parkinson's disease, such as impaired olfaction, subtle motor deficits, cognitive dysfunction, RBD, depression, and constipation. Once these individuals who are destined to get Parkinson's disease, do they look any different from so-called idiopathic Parkinson's disease? And the answer is not really. So they do get Parkinson's disease slightly earlier, generally about sort of 55 as opposed to 61, 62. And they do get more cognitive dysfunction. And their clinical progression tends to be more rapid. The pathology with Lewy body pathology is identical in terms of type and distribution. The imaging is the same. And the pharmacology is the same, their response to treatment. Turning from the clinical to the biochemical, the individuals with Gba1 mutations, when you look at their brains post-mortem, those who have developed Parkinson's disease have a significant reduction in Gba1 enzyme activity, Perhaps not surprising, of course. They've got a mutation in one of the alleles. But what is surprising is that Parkinson's disease cases with no Gba1 mutation also have a significant deficiency in the same enzyme. Not as severe, but clearly significant. And this basis of the enzyme being important to both Gba1 and non-Gba1 PD brings us to this very interesting reciprocal relationship between the enzyme activity, the enzyme produced by Gba1, so-called GCase, and synuclein levels. This reciprocal relationship, whereby any way you want to reduce the GCase activity, results in increased synuclein. And any way you reduce synuclein will result in, any way you increase synuclein will result in reduced enzyme activity. So this is a virtuous cycle, self-amplifying and perpetuating, and may well lie at the heart of the pathological process we see in Gba1 and non-Gba1 Parkinson's. So just in summary of Gba1 PD, it's a common risk factor, the commonest. 10 to 15% of PD cases have a Gba1 mutation. The penetrance about 30% by age 80. So why do some and others not develop Parkinson's disease? They have prodromal features. They have microglial activation in the brain before any evidence of clinical Parkinson's disease or dopaminergic deficit. And that's microglial activation is at least as determined by PET. They've got increased synuclein, increased spread of synuclein, earlier onset, more cognition, and faster progression. How does all this translate to therapeutics? Well, the Gba1 pathway is very amenable to being targeted to increase enzyme activity and thereby reduce synuclein levels. So here you see, oops, I'm sorry, let me go back one. Here you see gene therapy. It's a mutant gene, so by gene therapy techniques, usually using AAV, you can insert the normal wild-type gene, usually by intracerebral injection. These are in phase 1 clinical trial. And then you have pharmacological chaperone therapy. The mutant enzyme is trapped in the ER, and chaperone therapy will literally chaperone it to the lysosome where it's effective. These are currently in phase 2 and shortly in phase 3. HDAC inhibitors have yet to be developed for GBA. Enzyme replacement therapy will have to be modified because the current ERT is not brain penetrant. And substrate reduction therapy has failed in a phase 2 clinical trial. So let me bring you back to number 2 on this list, the chaperone therapy, because a repurposed drug called Ambroxol is a very effective GCase chaperone. And it is also a TFEB activator. TFEB is a transcription factor for the lysosomal pathway. So it has 2 important interactions: to increase enzyme activity and to increase lysosomal function. And in models from patients, fibroblasts, or alternatively Drosophila mutant models, or alternatively human dopaminergic derived iPSCs, or alternatively mouse models with Gba1 mutations, or non-human primate models, Ambroxol has a pretty impressive record of increasing the enzyme activity in the brain and reducing synuclein levels and spread in the brain. So this impressive portfolio of laboratory data led to this early proof of principle study published in 2020 using Ambroxol in patients with Gba1 mutations and Parkinson's disease and those without. And the endpoints of this study were safety of this drug in this particular population. CSF penetration was good, with 11% of the drug being present in the CSF compared to the blood, target engagement with an increase in CSF protein, and an increase in CSF alpha-synuclein, which is generally regarded as a good thing. And interestingly, the effects were seen in those with and without mutations. This positive proof of principle study has now led to the phase 3 study, the so-called ASPRO-PD study. And this study, which will have first patient in May of next year, will be a 2-year duration study. That is to say, those on active drug will be on that for 2 years. It's placebo-controlled, single dose. Primary outcome is parts 1 to 3 of UPDRS, 165 in each arm, and each arm randomized equally to those with and without Gba mutations, and a range of target engagement subsets in the CSF with a number of secondary endpoints, and Parkinson medications will be allowed. So this study will help define whether this particular pathway modulation and whether this particular drug can alter the course of Parkinson's disease in both a genetically defined and general Parkinson population. So I'll end there and thank you very much for your attention, and thank also all the people who've really done all the hard work and the sponsors of our research. Thank you very much.
Thank you, Professor Shapira. As I said, there will be discussion at the end. So the fight for finding drugs for risk factors for neurodegenerative diseases is extending to beyond Alzheimer's. And the next speaker will address another highly prevalent neurological disease, multiple sclerosis, and the speaker is Xavier Montalban. As you may have seen, Professor Comi has picked the big shots in their own domain. Xavier is the chair of the Department of Neurology and director of the Multiple Sclerosis Center in Catalonia, and the chief of the Neuroimmunology Research Group, and in the same research institute. Xavier is a physician, is a neurologist, and his interests include clinical trials and all clinical aspects of multiple sclerosis, from diagnosis to treatment. Xavier, please.
Thank you very much for the introduction. First of all, I would like to thank very much organizers for inviting me to be here with you. It's a real pleasure. Fondazione Prada and Professor Giancarlo Comi, thank you very much indeed. So I will be talking about the clinical outcomes that we use in phase 2 and 3 clinical trials in multiple sclerosis. This is my disclaimer slide. Let me just show first this is a slide that is coming up hopefully in Lancet Neurology. It's coming from the International Advisory Committee on Clinical Trials. As you can see here, I don't know if this is working. No. As you can see here, clinical expression of multiple sclerosis depends very much on the balance between injury and compensatory mechanisms. Many of them are quite similar to other neurodegenerative diseases as well. And these mechanisms are influenced by a number of factors such as infection, genetics, race, ethnicity, microbiome, comorbidities, etc. Of course, age is always an issue. I used to show this slide years ago, and this is coming from the classical natural history study showing that people with MS, 50% of them need some sort of help to walk after 15 years of disease. This is completely different nowadays. I think we have done our homework well, and we have now a number of different medications. I can give you that most of them are anti-inflammatory drugs. However, they have an impact on neurodegeneration. And the result of all these years is that the prognosis has changed quite a lot. This is a paper that we published last year. As you perhaps can see here, the probability for reaching an EDSS-3 higher at the age of 40 years has decreased very much, and it was 0.86 years ago, and now it's just 0.2. I also appreciate that the different diagnostic criteria play a role here with Will Rogers phenomena, but also early diagnosis and early treatment. So, which clinical endpoints or outcomes we have or we use in our trials? Let me show you one or two MRI outcomes. The first one is the gadolinium-enhanced lesions, or new or enlarging T lesions. This is the primary endpoint in all phase 2 relapsing-remitting MS trials. It's a key secondary endpoint in a number of phase 3 trials in relapsing MS as well. It's easy to, there you can see this, you can see this example with a number of gut lesions out there. They correlate quite well with clinical activity. They're easy to count. And this is the paradigmatic example of a phase 2 trial using the gadolinium-enhanced lesions as the primary outcome. This was the Natalizumab Trial, which is a humanized monoclonal antibody that targets the alpha-4 integrin, blocks migration of immune cells across the blood-brain barrier. As you can see in this slide, just as early as the first month, we observe a clear difference between the two doses of natalizumab and placebo. I think I don't know any positive phase 2 trial in relapsing multiple sclerosis which fails later on in a phase 3 trial. Perhaps Giancarlo and other people can correct me if I'm wrong. So let me just show you another slide about gadolinium-enhanced lesions as a primary endpoint in a phase 2 trial. This is tolebrutinib, which is a BTK inhibitor. And the difference here is that they just needed 3 months instead of the classical 6 months to have a positive result. And then, of course, is a key secondary endpoint in phase 3 relapsing MS. Just one example, I have been using different medications coming from different drug companies, of course. This is the dimethyl fumarate trials, define and confirm. As you can see, we obtain a 90% reduction with one of the doses in relation to placebo when we were counting gut lesions. What else we can measure using MRI? Brain volume. Typically, but not always, Sienna is the most used system. It's a fully automated method to determine absolute brain volume over time. So we run the program in different time points. We calculate the percentage brain volume change, and it's quite a robust measure for brain atrophy. So yeah, and there are a number of phase 2 trials in progressive MS that use brain atrophy as the primary endpoint. Here you have one example. This is the simvastatin trial in progressive MS that was positive. They measure the whole brain volume. Another one, the sprint MS using ibudilast, they showed a 48% slowing in rate of atrophy progression as well. And in fact, all these brain atrophy thing is coming from the phase 3 trial using fingolimod versus placebo years ago. They showed a clear beneficial effect of fingolimod in relation to brain volume change in comparison to placebo. And I think that was the first time that we realized how important it can be to measure brain volume. What else we have? We have others, but not now. We are very bad in measuring or identifying good outcomes for phase 2 trials in progressive MS. Perhaps chronic active lesions, cells, or peripheral rim could be one of them. Let's see. We need more experience. Clinical outcomes, relapses, annualized relapse rate. The primary endpoint always in relapsing multiple sclerosis, still after many, many years. Another example, this is teriflunomide versus placebo. We obtained a 31% reduction or 36% reduction in these two trials against placebo. And then if we compare teriflunomide against one monoclonal antibody as ofatumumab, which is an anti-CD20, the difference was 50%. So this is what we are using for relapsing remitting trial or for relapsing MS trials. What else? Disability. How we measure disability. We use the EDSS scale after I can't remember how many years, 30 years perhaps, is what we use in our clinical practice, not in many MS centers because it's not easy, you need time. And it's based mainly on neurological examination and walking capacity. So it's a primary endpoint in progressive MS trials, in phase 3 progressive MS trials. This is an example. This is the oratorio trial on primary progressive MS. We tried ocrelizumab versus placebo. The trial was positive, not very impressive, but that was positive. And ocrelizumab was the first drug approved ever for our patients with PPMS. We have another example, siponimod, which is a S1P modulator. They tried in secondary progressive MS, and the primary endpoint was the confirmed disability progression at 3 months. That was positive as well. I like this trial, the Biotin trial, because they were brave enough and they completely changed the paradigm and they measured the improvement of disability using EDSS or time 25-foot walk. And they were successful in the phase 2 trial. They call it sort of phase 3, but it was a phase 2. They were very positive, but they failed in the real phase 3 trial, unfortunately. What else we have for disability? The MS functional composite, which is a composite of time 25-foot walk, which is a walk-in test, the single-digit modality test, which is a cognitive test, and 9-hole peg test, which is measuring upper limb function. Was the primary endpoint in one phase 3 trial in secondary progressive MS, and some of the components are secondary endpoints in phase 3 trials. Let me just show you that one, the orange one by Jeff Cohen. He tried interferon beta 1A intramuscular in patients with SPMS, and they were successful. The problem they had is that they used the MS functional composite as the primary endpoint, and the FDA didn't believe them, so the drug was not approved. Another example is Informs, fingolimod in primary progressive MS. We used a composite endpoint using EDSS, but also 25-foot time walk and also 9-hole peg test. Any of them could be positive, but we, as you can see here, we failed as well. What about patient-reported outcomes? I think this is a hot topic we have nowadays in multiple sclerosis. I think that this is going to be more and more important. Let me just bring your attention to the PROMPTS initiative, which is a new global multi-stakeholder initiative on patient-reported outcome for multiple sclerosis, which is jointly led and coordinated by the European Charcot Foundation, specifically by the president, Professor Giancarlo Comi here, and the Multiple Sclerosis International Federation, with a key role of the Italian MS Society, and Paula is around here, I guess. Two examples of trials in MS using PROs. This is recent ones, ponesimod versus teriflunomide. They built up fatigue symptom and impact questionnaire, and it did work against teriflunomide, and that's important. It was a secondary endpoint, in fact. In the ozanimod trial, they had as a secondary endpoint the multiple sclerosis functional composite, but then they analyzed in a post-hoc way only the SDMT, which is a cognitive test, and results were positive, favoring those patients who were taking ozanimod. And what about NIDA? NIDA is no evidence of disease activity. That was introduced first by Eva Hadrova in 2009 in a Natalizumab trial. Typically, we use a NIDA-3, which means no relapses, no focal MRI activity, no EDSS progression. But you can complicate this as much as you wish with brain atrophy. That would be NIDA-4, or neurofilaments included. That would be NIDA-5. In this paper by Dalia Rothstein, when she was in Boston, she showed that NIDA is not easy to reach. And although 46% of patients treated were NIDA at year 1, only 7% of patients were NIDA at year 7. So it's not easy. And the predictive positive value is not bad, it's 78%. That means that if you are NIDA, probably your prognosis is going to be good for the next years, but the negative predictive value is not very good. So you can have some degree of activity and still having more or less a good prognosis. Let me go to the next one. This is what we published years ago as well. So perhaps a mild activity, very mild activity, some of you will not agree with this, is not going to produce any impact in the long term. And recently, again, Dalia Rothstein, this is a paper coming up in N2, showed that NIDA-4, adding brain atrophy, adds no advantage over NIDA-3. And there is one trial in which the primary endpoint is NIDA, which is the stenosis of ofatumumab versus first-line DMTs. What else we have? Is this enough? Probably not. So now we are very interested in the PIRA concept. So is disability progression independent of relapse activity? And we realized when we analyzed the OPERA's trials of ocrelizumab versus interferon beta-1a sub-Q high dose, we realized that patients didn't have relapses. Patients didn't have inflammatory activity in the MRI. However, a percentage of them, in fact, did progress. And most of the progression was due to the PIRA and not due to the relapse-associated worsening. So that means that the silent progression is present during the relapsing remitting phase of the disease. Very recently in Brain, Fred Ablin and co-workers published this paper. The green means PIRA. So even in relapsing remitting MS, in early MS, you can have that phenomena, PIRA phenomena. Of course, is greater in SPMS and primary progressive MS. And let me just show you work from my group, which is coming up in JAMA Neural. In our CIS cohort, we studied that, and we realized that, let me just see if I can do something, that difficult, that about 1/3 of patients with CIS, they do have PIRA. And in fact, 10% of the patients have PIRA during the first 3 years, so after the first symptom. So that means that you have a percentage of patients with SPMS during the first 3 years of disease. And the most important thing is that those patients who have PIRA, they have a bad prognosis in the long term. And this is a time to EDSS-6. So it could be a sort of a surrogate outcome for long-term disability. What else we have? We have more. When we are in the clinic and we are measuring the EDSS, this is just one snapshot that you have, but and perhaps in the best centers, we can see the patient every 6 months, occasionally every 3 months if the medication is a little bit complex, but in many occasions once per year. So we need more granular information. And then the digital technology comes in. Perhaps we could be able to identify subclinical disease activity and also progression of the disease. And the other important point, and talking again about PROs, is that we can ask patients with the smartphone about fatigue, about mood changes, about pain, about sexual dysfunction, etc. We ran this trial, the floodlight study, using a smartphone and an app measuring different things, PROs, test, and also passive monitoring. We did that with UCSF. And the adherence to active tests was quite high, 78%. The adherence to passive monitoring was very high, 90%. Although I have to say that that was under very much controlled circumstances. And also, what we saw is that the correlation between what we obtain from the app and what we measure in the clinic was really very much the same, and that was very reassuring. And this is my last slide. So still, we need better clinical and MRI endpoints, mainly for progressive MS. Combined scores allows integration of disease activity data. PROs must climb positions in the hierarchy of outcomes for sure. And this is our center in Barcelona. And I thank you very much for your attention.
Thank you, Xavier, for this overview of the MS field, which is by the dementia community often taken as an example, as a successful story to be followed. I cannot speak for the Parkinson's disease community, but this is one of the reasons why this type of events is so important, to exchange ideas across fields, across domains, which is usually not the case. We have our meetings in Alzheimer's, the meetings in Parkinson's, you have in MS, but it's not common to meet and discuss ideas. So I believe that this is very forward-looking from Fondazione Prada and Professor Comi to allow us to mix and intermingle. Thank you, Xavier. And I please, Xavier, you're supposed to sit here, and also the other speakers, Jeff, Professor Shapira. We are supposed to have the general discussion now, together with Vincenzo Silani as well. Vincenzo Silani is a well-known expert in ASL. Vincenzo is currently you're still at Axologico, yeah, right? Director of the Dino Ferrari Center for Neuromuscular, Neurodegenerative, and Cerebrovascular Disease. We're supposed to have a general discussion. So,
have a short presentation, 10-minute presentation, 7-minute presentation.
A 7-minute presentation, yes. Okay. And then another 7-minute presentation by Dr. Zhang, right? Okay. So, and at the end, we'll have a general discussion.
Sure.
Okay. Vincenzo.
So, thank you for the introduction, and thank you to Giancarlo and Prada Foundation. And talking about ALS on purpose, I just put this slide because this is the timeline of ALS and clinical trial. As you see, compared to the previous speaker, this is an empty timeline. You see my juvenile effort to change all the serum of the ALS patient, the plasma change. The rationale at that time was that something in the serum is toxic. It actually was a negative trial. Then you see the 1995 curve. This is the Riluzole, that just has been the first drug approved in clinical trials. And then at that time, with two good friends of mine, Hiroshi Mitsumoto and Benjamin Brooks, we decided to try to define why we failed for so many years. And actually, we failed probably because we didn't know enough about the rationale of the drug that we used. In other words, we didn't have enough rationale in preclinical settings. Then we didn't know enough about the pharmacology of the drug that we use in the trials. And then we needed to design better clinical trials. At that time in 2017, the Daveron story just came out. The Daveron, as you know, is the second drug approved by FDA in ALS, disputable data. But with Hiroshi and Benjamin, we decided to convene in Early House. Early House was a historical place in Virginia where the LS community used to meet and try to come out with new alternatives. And the revised Early House consensus guidelines were very useful in order to approach clinical trials. But in the last 2 weeks, the ALS community has been totally shocked about two news that just two approval, actually one approval related to the so-called Amylyx, and two drugs has been approved by FDA. And then there is the positive data related to tofersen in the SOD1 ALS patients. I'd like to say firstly that ALS is a very heterogeneous disease. Along the years, we know that the clinical onset is very heterogeneous. We have to consider the temporal frontal impairment of those patients. And this is crucial in order to define the survival of the patient. The pathogenesis is very heterogeneous, and ALS has more than 30 genes at play. C9, number one, is the most frequent gene in the sporadic and familiar cases, both in ALS and frontotemporal. Let's go in details related to the trial. This was a phase 2 clinical trial with those two molecules, sodium phenylbutyrate and hydroxydiol. And I can read from here, but basically the endpoint were the ALS functional rating scale that you can see over here. Oops. The functional rating scale and the survival, basically time to tracheostomy or survival, and the open label, and then the final open label trial use very, very generalized endpoint, like time to key event, time to death or tracheostomy, time to first hospitalization. You see how very generalized are those endpoints. Related to tofersen trial, there are two main issues here related to the decrease in the SOD1 activity in the CSF and the decrease in the neurofilaments in the serum. Those are two biomarkers that are very well characterized in these phase 3 trials. And there are also clinical endpoints that are being reached, namely the functional rating scale, namely the survival and vital capacity, and also the megascore dynamometers measures. The point that I like to make related to the tofersen study is that today our approach to ALS patients is completely changed. In other words, in sporadic and familiar cases, we have to run the SOD1 assay and just to define if the patient is mutated, because Biogen is offering an open label trial, basically open treatment, at least in this country, for all the patients. So coming on the main issue related to the outcome measures in this disease, as I said before, those are very common and some kind of primitive outcome measures. Functional rating scale, one of the several scales, time to tracheostomy, survival, but you see there is an absence of a cognitive behavioral dysfunction scale in most of the trials. This is crucial because this is a paper, but there are many papers related to the issue. This is a paper that has been done by Orla Hardiman in Ireland in 2011, saying basically that the patient with cognitive and behavioral changes live less. The survival is affected, in other words. And this is very important. There is a new issue coming, namely the effort of the ALS Society to come out with a concept of mild motor impairment. This is mainly related to the work of Michael Benatar. Basically, the ALS communities try to define a prodromal situation in ALS patients. This is very difficult compared to MCI, just to make a parallel. But biomarker can be utilized, and mycophenolate was clearly demonstrated that at least in the C9 and the SOD1 mutated patient, the neurofilament in the serum are going up 2-3 years before the onset of the disease. So biomarker can be utilized in the prodromal situation in order to define eventually the conversion to the clinical status. And neurofilament are in the LS community probably the best marker, also able to not just to validate the diagnosis, but also neurofilament can be used as a prognostic factors because a patient that have high neurofilament has less survival compared to patient that has a lower level of neurofilaments. I have to mention the dipeptide, also biomarker that actually with Petruccelli we defined many years ago. And the dipeptide is very useful, can be measured in the CSF, in the serum. The dipeptide is increased in preclinical settings, and they can utilize eventually as a biomarker for efficacy of treatment. And the interaction between this dipeptide and neurofilament has been adequately described. In other words, when the patient carrying a C9 mutation is developing the disease, has also increased the neurofilaments. Well, there is a desperate need to find a biomarker in neuroradiology, and we are focusing our attention on the accumulation of iron. Iron overload in the presental gyrus has a marker of upper motor neuron involvement, but there are many other efforts. And obviously, we are moving to digital biomarkers, and this is a very confusing area. In LS, it's not simple compared to Parkinson, for example, and so on. In long-term patient, we can use eye tracking and brain-computing interface in order to communicate with the patients, so we can have a long-term analysis, at least of the cognitive behavioral changes. Finally, I can escape the possibility we have with human bioassays. In our laboratory, we are just using IPS, as you know, like many other laboratories. But our effort was to try to have IPS from the blood. And actually, this is making the difference because we can obtain IPS many times as much as we like. And from IPS, as you know, we can obtain motor neurons and cortical neurons and organoids. And those are powerful bioassays we can use combined to the patient in order to focus and monitor our treatment eventually in the future. I'd like to finish to say, first of all, that we have prognostic models that we develop in ALS. This is a nice way to analyze the survival of the patient, predict the survival of the patient. And finally, I'd like to say the new area of ALS treatment is related to developing two platforms. One platform, the name is Trickles, is organized in Europe. And another platform is active in the United States. That means when the patient need to be treated, go to the platform. When the company want to treat the LS patient, go to the platform, and eventually placebo control they can share in the platform, and different trial can be eventually run in the same platform according to the different countries. And those are the people involved in Milan in our laboratory. Thank you.
Thank you, Vincenzo. Please take a seat. Now, the setup of this session is particular. The next speaker, I was looking for a Chinese-looking guy in the audience. I couldn't find him, and we will not find him because he will show up behind us. Here it is, Professor Nang Zhang from Tianjin Medical University. He will address frontotemporal dementia. The allotted time was 7 minutes, but I understand that you may need a bit longer. Is that correct, Dr. Zhang?
15 minutes, but I will try my best to shorten my presentation.
Thank you very much. Dr. Zank is an expert in the neuropsychology, neuroimaging, and biomarkers in dementia and neurodegenerative diseases. Please give us your lecture on frontotemporal dementia.
Thank you so much for introduction. Good morning, ladies and gentlemen. It's my pleasure to give you a little bit of current knowledge and advances in frontotemporal lobar degeneration. Here are my disclosures. Frontotemporal lobar degeneration is characterized by the unifying hallmark of atrophy of the frontal and or temporal lobes. However, aggregates of distinct types of misfolded proteins can be observed on a microscopic level. Currently, almost all frontotemporal dementia cases could be classified into 3 major molecular subgroups based on the disease-specific protein deposits. The most common histopathological subgroups are FTLD tau, FTL, and FTLD TDP, accounting for approximately 40% and 50% respectively, followed by FTLD fat, and consisting of fused in sarcoma, urine sarcoma protein, and tata-binding protein associated factor 2N, accounting for approximately 5 to 10%, and FTLD UPS, accounting for less than 1%. FTLD is a highly heritable disorder. Mutations in genes such as chromosome 9, open reading frame 72, progranulin, and microtubule-associated protein tau, MAPT, have been identified in about 25% of patients with frontotemporal dementia. Chromosome 9 and progranulin mutations are associated with TDP43 aggregation, whereas mutations in MAPT lead to tau aggregation. In recent years, mutations in an increasing number of genes have been associated with autosomal dominant frontotemporal dementia, such as BCP, DAR-TDP, DAR-DBP, TPK1, etc. Cumulatively, they account for less than 5% of all frontotemporal lobar dementia cases. The clinical disorder caused by FTLD are highly heterogeneous in their pathology and phenotypes, encompassing a group of neurodegenerative syndromes, including behavioral variant frontotemporal dementia and language predominant syndromes such as primary progressive aphasia, PPA, including the non-fluent agrammatic variant PPA and the semantic variant of PPA. Other syndromes that are part of this group are characterized by prominent motor symptoms such as cortical basal syndrome, progressive supranuclear palsy, and frontotemporal dementia with motor neuron disease. Most patients present with mixed behavior, executive function, language, and motor symptoms, but are diagnosed based on their most pronounced initial symptoms. For biomarkers in the past decade, neurofilament light chain, NFL, has become a biomarker of interest in FTLD, since it is a sensitive marker of neurodegeneration and its concentrations are correlated with the rate of clinical progression. In addition, NFL concentrations in plasma correlate well with those in cerebrospinal fluid. Moreover, NFL has been shown to predict future clinical progression during the prodromal stages of genetic FTLD. Although total tau concentration is not sensitive or specific for FTLD, amyloid beta and phosphorylated tau concentrations are useful in differentiating FTLD from Alzheimer's disease pathology. In our recent study, plasma concentrations of phosphorylated tau 181, NFL, and glial fibrillary acidic protein, GFAP, were measured with single molecule array method in a cohort including persons with Alzheimer's disease, subcortical ischemic vascular dementia, and FTLD, and cognitively unimpaired controls. Persons with FTLD showed higher levels of plasma NFL compared with controls, and lower levels of plasma phosphorylated tau 181 and GFAP compared with persons with Alzheimer's disease. Among the 3 biomarkers, NFL had the highest area under the curve of 4.8 in distinguishing persons with FTLD from controls, and GFAP had the highest area under the curve of almost 4.9 in distinguishing persons with Alzheimer's disease from those with FTLD. consensus diagnostic criteria for behavioral variant frontotemporal dementia and PPA include neuroimaging as the major biomarker to aid in clinical diagnosis, with an emphasis on MRI for atrophy, ECT for hyperperfusion, and FDG PET for hypermetabolism in the frontal and or temporal lobes. Amyloid PET imaging can be used to differentiate underlying Alzheimer's disease from non-Alzheimer's disease disorders in people presenting with frontotemporal dementia. Tau PET imaging was initially considered to be a potential major breakthrough in differentiating tauopathy from non-tauopathy among individuals with underlying FTLD, but this potential has not been substantiated. Although not yet used clinically, there are a number of additional informative neuroimaging modalities that can elucidate the pathophysiology underlying FTLD, including diffusion tensor imaging, resting state functional MRI, and auto spin labeling MRI. Now, I want to present some cases to show how we diagnose patients with FTLD and differentiated them from patients with Alzheimer's disease. Case 1 is a 70-year-old woman who developed apathy, inertia, and disinhibition, accompanied by executive dysfunction and memory impairment for 2 years, with an MMSE score of 26. And MRI and FDG PET showed mild atrophy and hypermetabolism in bilateral medial temporal lobes, and obvious atrophy and hypermetabolism in bilateral frontal lobes. PPET, which detects amyloid deposition, was positive, and Tau PET presented a deposition pattern typical for Alzheimer's disease, for example, precuneus and temporal-parietal cortex and frontal lobes, indicating Alzheimer's disease pathology or comorbidity. Case 2 is a 64-year-old man who developed impairments in memory and language for 2 years. With an MMS score of 16, spontaneous speech was characterized by slow rate with frequent pauses due to word-finding problems. Brain atrophy on MRI and hypermetabolism on FDG PET were mainly observed in the left temporal-parietal junction area. Both PIP and Tau PET imaging were positive, although off-target finding is also observed in basal ganglia and brainstem on Tau PET. This person was finally diagnosed with local panic variant PPA due to Alzheimer's disease. Case 3 is a 50-year-old woman developed progressive language disability with gradual onset for 1 year. She forgot the names of familiar friends and relatives and became incapable of understanding the meaning of simple words, such as computer or skyscraper, and common Chinese idioms, but was without any psychiatric or behavioral changes. Bilateral but left predominant atrophy of the anterior temporal lobes was observed on brain MRI. PPET was negative. Tau PET showed diffuse tracer banding in cerebral, frontal, temporal, and parietal lobes. Hypermetabolism mainly in the bilateral, temporal, and frontal cortex was also seen on FDG PET. This person was diagnosed with SV, a semantic variant PPA according to her symptom and imaging features, and was confirmed by gene testing to carry the MAPT P301L mutation. Semantic variant PPA is generally sporadic and caused by TDP-C pathology, with very few reports of tau pathology caused by MAPT mutations. This case was from a large family with the MAPT P301L mutation. All subjects in this family, including living and deceased subjects, showed semantic memory impairment in the early prominent symptom. In addition to the proband and the case, there are 7 family members carrying the MAPT mutation according to genetic testing, although there were no clinical symptoms reported by them. The other 9 participants who also underwent gene testing did not carry this mutation. The top line is representative imaging of the semantic variant PPA case. The middle line is representative imaging of a pre-symptomatic carrier with abnormal cognitive assessment. Although there is no visually obvious atrophy on brain MRI, focal hypermetabolism of bilateral frontal lobes and diffuse tau tracer banding in temporal and frontal lobes were observed on PET scans The bottom line is representative imaging of a presymptomatic carrier with normal cognitive assessment. No obvious atrophy, hypermetabolism, or tau accumulation were observed on brain MRI and PET scans. We further compare the differences in plasma biomarkers and cerebral blood flow measured with alter-spin labeling MRI between mutation carriers and non-carriers to explore the early biological changes in the thematic variant PPA phenotype that are caused by the MAPT mutation. There was no difference in NFL and GFAP between 9 mutation carriers and 9 non-carriers, although the levels of NFL and GFAP were higher in the 2 patients relative to both presymptomatic carriers and non-carriers. Interestingly, increased regional cerebral blood flow was observed in mutation carriers without adjusting for global value. After normalization for global value, both relatively decreased and relatively increased regional cerebral blood flow was observed in mutation carriers. At last, I want to very briefly introduce the Ftd Prevention Initiative. The aim of the Ftd Prevention Initiative is to bring together a worldwide cohort of FTLD and to promote clinical trials of new therapies. Okay, thank you for your attention.
Thank you, Dr. Zhang, for this overview of diagnostic and treatment issues in frontotemporal dementia. Now, we are supposed to have a general discussion now. It's not easy to identify a topic common to all what we heard. I propose that we do a roundtable on a topic that has been mentioned mainly in the last 2 talks, which is that of blood biomarkers. I believe that blood biomarkers will radically change the way we do the neurology of these diseases in the next future. I remember the first time I heard about trying to measure amyloid in the blood, it was 30 years ago by John Groudon. I don't know if is that correct? And it was a failure. He wasn't able to. But then there was a long sleep, right? 25, 30 years, and then 3 years ago, there was an impressive acceleration. Some of the fields that we've seen have already taken up, such as MS has already taken up the blood biomarkers for clinical trials. It might be interesting to hear about the other fields. Jeff, do you want to give us your vision, your long-term vision of how blood biomarkers will change the way we diagnose, treat, prognose, predict the development of Alzheimer's and the other dementias in the future?
Great. Thank you, Giovanni. And of course, we're going to have a session on biomarkers that will be very informative. Here's a radical vision of the future. There won't be any testing, right? It's so artificial that you have testing every few months and you make a bunch of conclusions on that. Combination of biomarkers and completely passive monitoring that is comprehensive will allow us to do without the EDSS or the ADAS-Cog or any other tool. What we want are continuous measures of the success of our interventions. And we have a long way to go to get there, but that would be a radical vision. The combination of biomarkers, so we see the biological impact, with completely natural, continuous, passive biomarkers that would allow us to know how our patients are doing.
Professor Shapira.
So in Parkinson's disease, we do not have a peripheral marker of disease. But there's a lot of work going on to identify one, and alpha-synuclein is an obvious target. I think that the development, let us suppose that alpha-synuclein does reach a level when we can use it as a marker of Parkinson's disease. What will be so important, it will be to apply that to genetically susceptible populations. So in Parkinson's disease, we have quite a number of individuals who are genetically at risk with genes that have variable penetrance. So if one can use those peripheral markers to identify, for instance, which the 30% of the Gba1 carriers that are going to get PD and those that don't, then those obviously will become eligible for preventative treatment if that's available. Importantly, I think these biomarkers that relate to underlying pathology, like alpha-synuclein levels, will be important in assessing therapeutic response of drugs, to sort of engaging the target, if you will, and identifying it peripherally and following up the progress of the patient, inevitably associated, more important, with the clinical outcomes as well. In Parkinson's disease, we do have some clinical prodromal markers which identify individuals at high risk, like REM, sleep behavior disorder, olfaction, and potentially combining that with some of the imaging techniques too. So if one could take those and add a peripheral marker to form a basket of at-risk markers to identify individuals who will develop Parkinson's disease, then I think that would be a great step forward and importantly, enable us to test our drugs, which, because it's a neurodegenerative disease, you have to have a readout after several years. It may bring that forward so we can determine whether that drug is efficacious or not.
So what you're saying is that you might have a first layer of screening with genetics, and then a second with peripheral markers with or without clinical signs.
Absolutely. That's the way I see it.
Xavier, tell us how the neurodegenerative field might benefit from the multiple sclerosis experience.
Well, I think during the last few years, I think there has been a tremendous amount of information on a number of blood biomarkers, but specifically on neurofilaments as well. And as you know, neurofilaments comes from axons and neurons, although probably inflammatory activity is the main driver for increasing neurofilaments. I think nowadays many MS centers around the world are using the levels of neurofilaments in their clinical practice already. It has some predictive value, but how is the disease going on? But also in clinical trials is now an important piece. Some of them as an exploratory element, but in one or two phase 2 progressive MS trials is the primary endpoint. So neurofilaments, I guess, in a number of health systems could even substitute occasionally the practice and performance of MRI.
Jeff, do you think that might happen in dementia and Alzheimer's as well?
Yeah, well, you know, in the U.S. we already have quite a few marketed biomarkers for AD. So you can buy the 42:40 ratio now for your patients. So it's coming into healthcare systems. p-tau217 looks very close, and NFL is already available to us as well. I don't see it being used in a very structured way yet. I mean, it's very kind of haphazard, clinician dependent, how competent they feel in interpreting the results. So I feel like there's a long way to go yet to define exactly how to use that information. But I did want to comment on one of Javier's part of the presentation, which was the combination biomarker, or the combination of biomarker and clinical outcomes that you're using for the no active disease definition. Because I think those kinds of composites could be used in other circumstances. And I think the statistics are also evolving for application to very complex composites like that. So that is some cross-disease learning that I think is really valuable.
And it's true that we will have Herman Zetterberger who will talk about the fluid biomarkers in a few minutes. But I was really interested to hear Vincenzo about TDP-43. Will we be able to measure Vincenzo and also Professor Zhang remotely? He's still somewhere in the ether.
Yeah, TDP-43 is obviously the marker we are looking for many years, and nobody on the planet, I think, is able to measure TDP-43, that basically is affecting more than 90% of familiar and sporadic ALS, plus, I would say, 40% of frontotemporal. So that's a major biomarker. But I think in ALS we are in a very opposite direction, in opposite situation compared to other diseases. Firstly, we don't have a preclinical, unfortunately, and we are really struggling to have a pre-symptomatic situation difficult to define, even at the clinical or neurophysiological terms. On the other side, we have more than 30 genes affecting the familiar cases, 15 of the sporadic cases, and C9 specifically. And we have a neurofilament gaining some kind of responsibility, even as an endpoint in clinical trials, because as I mentioned, the last 2 clinical trials have really neurofilament as an endpoint. But obviously, I think we need 2 or more combined biomarkers. I mean, the situation in Alzheimer, I think, is ideal because you can play with 3 markers. Here we are playing with 1 biomarker and eventually a dipeptide in C9. So it's a different situation, but I think it's some nice information and it contributed to neurodegeneration.
Professor Zhang, Professor Comi? Okay, Professor Zhang first and then Professor Comi.
I totally agree. From my point of view, of particular interest is the development of fluid assays that can detect markers of key protein opposites in FTLD. Findings on TDP-43 and tau real-time quaking-induced conversion seeding activity in cerebral spinal fluid and autopsied brain tissue of people with frontotemporal dementia appear encouraging. In addition, unlike Alzheimer's disease, the phenotype of patients with FTLD is very complex, including behavioral, language, and executive function, and so on. So I think we need to, we also need to propose a clinical evaluation for patients with FTLD to facilitate the diagnosis and evaluation in clinical trials.
Thank you, Professor Zhang. Professor Comi.
One common point that was being raised by almost all the speakers is that now that we are moving to the prodromal phase of the disease, it's becoming quite hard to use a clinical endpoint, even in phase 3. The lesson of MS is clear. The recent trials have shown that it is already in early diagnosis patients difficult to satisfy EDSS criteria in order to have enough patients having an increase of disability. So how can we then think about the future trials in Parkinson, ALS, MS, and Alzheimer? Because I suspect that we should move to trials of 5 years duration in order to have enough proportion of patients meeting the criteria for deterioration. So what is your view about this point?
So I'll jump in on that. It's interesting across all the diseases. I think we can see one possible way forward, which is the FDA's approval of aducanumab on the basis of amyloid lowering. So if we gain confidence in that, and it's been very controversial, but if we gain confidence in that, and if we think it's predictive of slowing cognitive decline, I think it could be an outcome that would substitute for a clinical outcome for these very early individuals. They're not patients yet, right? Because they have no symptoms. They're participants in trials that could eventually become a prevention for people at risk in the population.
Professor Shapira?
You've hit upon a very important point, Professor Comi, of course, and in Parkinson's disease, we are not yet at the stage where we can take a group of people that have prodromal features and know how many of them will develop Parkinson's disease in 10 years. Even if we add in genetic susceptibility, like a Gba1 gene, we still do not know what proportion would naturally develop PD. So putting them into an enriched clinical trial, we're not there yet. But perhaps with peripheral biomarkers and the complex basket of features, we might be at the stage where sometime in the future we can do that. And obviously they would be the most valuable cohort to test disease-modifying treatments.
I think that's an essential question, of course. I think in multiple sclerosis, you have two situations. One is in RIS, patients who have radiologically isolated syndrome, no symptoms at all, MRI very typical. I think that's easy. MRI gut lesions for phase 2 trials is the answer for that. Then I wonder why we need phase 3 trials with an annualized relapse rate as an outcome. So what you just need is a solid, independent risk management plan once a phase 2 trial is positive. On one hand, for progressive MS, I think situation is much more complex because we don't have a clear outcome for phase 2 trial. I think still there is a pitiful lack of that biomarker. But I guess that in the future we will be able to identify Doc Arnold's initiative and others, something that will allow us exposing few patients to the experimental molecule to say, okay, I think this deserves a study, why we don't go forward, and not for 3, 4, 5 years of clinical trial, 3, 4, 5 years of taking the drug, but with a solid, independent, that's essential, risk management plan. This is how I see the situation.
Vincenzo, what about prevention? The prevention scenario for a rare disease is not obvious, right?
No, in ALS, absolutely no. Even because the phenoconversion, the clinical phenoconversion in ALS is really quick, so it's very difficult to define. Probably we have to rely on mutated patients carrying, for example, C9 and follow families and pre-symptomatic and use C9 as a pre-selected population in order to pre-treat. But I don't think we have alternatives right now.
Professor Zhang, do you want to comment finally about frontotemporal prevention in frontotemporal dementias, which also seems unlikely to me? Professor Zhang, you still somewhere? No, non è più collegato, stavo facendo la domanda. Okay, so I believe that we can close here. I would just want to leave you with one point of reflection. Can we envision maybe a future where neurofilament light in the blood will be a surrogate outcome across the board, a surrogate endpoint across the board? For MS, it's already. And what about the others? Maybe it might be. So, food for thought, and now let's go have some food for stomachs as well, and drinks and maybe coffee. Thank you everybody for listening, and see you in 20 minutes.
Thank you very much indeed.
So, good morning, everybody. Professor Comi has arrived, so we can start with the next session, which is a very, very exciting session because it has already been introduced a little bit by the previous speakers in the previous session, how important biomarkers are across the field of neurodegeneration. I think that's the beauty of this conference, that we are discussing the whole field of neurodegeneration, AD, PD, MS, all the other neurodegenerative diseases have come across already yesterday and also today. And I think one binding element is there, the development or the presence already of biomarkers. I think that makes this session extra interesting. And of course, we have very interesting, very well-versed speakers in this session. So my name is Philip Scheltens. I'm from the Amsterdam University Medical Center. I also started my career in biomarkers. Actually, my whole career is in biomarkers, so that's the only thing I did. So let's start with the first speaker. A very close friend of mine, Giovanni Frisoni, originally worked in Brescia and has done a lot of things outside of Brescia, but then decided to move to Geneva. And although Switzerland is not a member of the EU, as you know, Giovanni is. He is the most sort of widely accepted lead of several EU-funded programs I know in the world. So it's a very pleasure for me to introduce my good friend Giovanni Frisoni, who will actually detail magnetic resonance imaging, the biomarker where it all starts with.
Thank you, Philip. I'm glad that you feel excited because I wasn't when I was given this topic on MRI, which is no longer one of the most exciting biomarkers. And I also have a hard time advocating for MRI. As a clinical person, I couldn't care less about MRI by itself. What I find of interest is the use of all the panel of biomarkers for what they can tell you, for what they can tell us, for what they can give us. And since Philip started before me to dig into the world of biomarkers with MRI, a lot happened afterwards. Anyway, I will try. I will touch three issues, three topics here where MRI can be integrated in clinical trials and the meaning of MRI, of what MRI can offer. Number one, obvious. MRI for surveillance of anti-amyloid monoclonal antibody treatment. Number two, the use of rates of brain atrophy by volumetric MRI as a marker of clinical disease progression and a surrogate endpoint of clinical trial. This is not really the latest news, but there's some new ways of looking at an old problem. And last, the use of MRI used in a very traditional way to exclude people whom we do not want in clinical trials, the slow progressors, those whose disease is so slow that we really do not want them because they are polluting the results in clinical trials, number one. And number two, even if the drug shows under testing shows positive, we will most likely not use the drug in these persons because they're progressing already slowly, naturally. So MRI for surveillance. This is one patient whom we treated in the aducanumab trial. In the open label extension, and this patient received 5 doses at 10 mg per kg until October. On October 2021, she received the 5th dose of aducanumab, and then MRI showed some images, something strange that you see here. This is vasogenic edema. It's localized vasogenic edema due to leakage of the water part of a plasma into the brain tissue. This is a gradient echo image where you don't see much, really. But after how long? 6 weeks, you started to see small black spots, which are small spots of microhemorrhages, where you had edema. The patient was all the time asymptomatic. This is not always the case. This is one of 13 patients whom we treated. This is to say that, of course, brain edema is not nice to have. It's not nice you to have. It's not nice for your patients, if you're a physician, that your patient have brain edema, because one, if it's localized, well, okay, fine, it's no big deal, kind of. But if it's spread more widely in the brain, it can be an issue. This is to say that if and when, hopefully positively, monoclonal antibodies will be used in the clinic, MRI will be key to monitor the adverse events such as ARIA. ARIA stands for amyloid-related imaging abnormalities. Apologies for not having mentioned that earlier on. E stands for edema. H stands for hemorrhage, of course. So MRI will be a cornerstone of monitoring adverse effects in treated patients, and not only at the beginning of the treatment, but which is where you usually have more frequently areas, but also during the treatment. These are the results from the lecanemab trial, the phase 3 Clarity lecanemab trial, where you can see that the area E and area H are fairly common. 20% any kind of area. Of course, it's the other way around. 20% in the treated, 9% in the placebo. But the percentage of symptomatic areas is relatively low. But of course, they should be, as soon as an area is detected, measures should be taken in terms of discontinuation of the perfusions. Second point, rates of brain atrophy by volumetric MRI as a marker of clinical disease progression, a disease tracking marker. I received a comment after the previous session was over that we're using biomarkers in a very wide, very large way, which is true. You can use biomarkers for many different aims. But you should be clear what you use it for. Here we're talking about using biomarker to track disease progression and possibly as a surrogate endpoint in clinical trials, such as neurofilament light in multiple sclerosis trial, which was mentioned by Xavier Montalban. Now let me bring you to the pathophysiology of where atrophy stays in the progression of the disease. This is a slide showing you what we developed recently as a concept of the pathophysiology of Alzheimer's broken down by 3 separate forms. The autosomal dominant AD, there's Randy Bateman in the room. It's quite kind of hard to mention autosomal dominant AD, but in the APOE4 related sporadic form and the APOE4 unrelated sporadic form. What we're proposing here is that the chain of events that we've always labeled as amyloid cascade is a cascade in autosomal dominant, but it may not be a cascade in APOE4 related sporadic, and even less in APOE4 unrelated sporadic Alzheimer's disease. What am I saying here? I'm saying that the way we've been using, the way we've been developing drugs and testing drugs until recently has been relying on a deterministic cascade of events, which is probably applicable in the familial form, where everything starts with amyloid, and that's sure. Then tau deposits, then you have neurodegeneration, neuronal loss, and synaptic loss, and then at the end of the story, you have clinical symptoms. And that's inevitable. It's a cascade. It's inevitable. You have the mutation, okay, you have amyloid, then you have tau, then. It's inevitable. And that's why, okay, I don't have it here, but you can see the black stickmen denote the notion that at the lifetime prevalence or the penetrance of the trait of the mutation is almost 100%. You have the mutation, you will have amyloid, you will have tau, and you will have symptoms. This is a cascade. And based on this notion, monoclonal antibodies were tested in symptomatic patients who are 15 years into this process. Why? Well, because if it's really a cascade, you block the cascade earlier on and everything else follows. So you should see a plateau. You should see the disease progression halt after a given time, which is not what we've seen in sporadic Alzheimer's disease. In sporadic, what we believe that the cascade is more like a chain, a loose chain. A loose chain starting from a genetic predisposition, APOE4, which predisposes to amyloid deposition, which is followed by tau deposition, and so on. And this chain of events is even looser when you don't have the APOE4 gene, which predisposes to amyloid deposition and everything else that follows. What does it imply? Well, it implies that, number one, that the lifetime prevalence of the clinical trait in APOE4-related sporadic AD is way lower than 100%. It's probably around 60%. And the lifetime prevalence here is 20%. Fine. But the other relevant implication for disease, for treatment and drug development is that anti-amyloid drugs should be more effective here than here. Amyloid drugs should be, if administered in already symptomatic patients, should be more effective in the E4 carriers, in the sporadic E4 carriers, than in the sporadic non-E4 carriers. And this is indeed what has been found in the phase 2 donanemab trial that was mentioned by Jeff Cummings earlier on. What they found is that the decrease of amyloid from baseline, let's see the leftmost part of the graph, that the greater decrease of amyloid, the better, the greater the decrease, the slowdown of the clinical progression. In other words, the clinical benefit. So the more you decrease amyloid, the better the clinical effect in the whole group. But if you break down by APOE4 carrier status, and I remind you that between 50 and 60% of all our Alzheimer's patients in the clinic are E4 carriers. If you break it down, you see that in APOE4 carriers there is no effect, while you can see an effect in carriers, which is exactly what is predicted by the what we call the probabilistic model. Now there's going to MRI, there's some predictions that we can do relative to a trial which is currently going on E44 homozygotes. E44 homozygotes are about between 5 and 10% of all Alzheimer's patients in memory clinics. So they're not super frequent, but they're not super rare either. And these guys have found this is a group of scientists who have launched a clinical trial with an anti-amyloid small molecule. And in the ADNI cohort, they found that rates of hippocampal atrophy and rates of cortical thinning were associated in the natural history of the disease. These are untreated patients. Okay, this is an ADNI observational study. They were related to the clinical progression measured with ADAS-Cog13 and a little bit with MMSE as well. So what does it say? It says that in E4 homozygotes, volumetric changes, atrophy changes on MRI track clinical disease progression. And the same group has recently presented the results of their clinical trial with this small molecule, which is an old molecule that's been reshaped, rebranded in a way, by adding a valine residue. And what they found is that the treated patients, the whole group had a response to phospho-tau 181 in plasma, and these were all E4. If you compare this response of tau to this anti-amyloid small molecule to historical changes of a phospho-tau, plasma phospho-tau in monoclonal antibody-treated patients, you see that who have all varieties of ApoE4, there's a much lower decrease of phospho-tau. Our model predicts that if you break down this population by ApoE4, you should see a greater effectiveness in E44, lower effectiveness in E4 homozygotes, and even lower in non-carriers. We will see if that's the case. In the last 4 minutes, I will address the issue of excluding slow progressors from clinical trials of disease modifiers. I'm going back to the notion of finding a signal in clinical trials. Even in E4 carriers, the signal, the benefit is not terribly strong. It's probably around between 25 and 30% slowing of disease progression. 25 to 30% is not something to jump on the chair for. So there's a reason to try to enrich the clinical trial population with patients who do progress to be able to see more clearly the effect of the intervention. And indeed, there's a population that we may wish to exclude, and these are the so-called late limbic predominant age-associated TDP-43 encephalopathy. It's a condition which used to be called hippocampal sclerosis. Now we know that the vast majority of these patients have pathological accumulation of TDP-43, which is the same protein involved in some cases of frontotemporal degeneration and in ALS. The TDP-43 in this condition deposits in all the patients, mainly in the medial temporal lobe, and specifically and very early on in the amygdala. And it can be really easily detected with MRI. You don't need to be a neuroradiologist, right, to figure out that something is wrong with this MRI. And so what we did is to look for the clinical features of these patients in our memory clinic population. This slide shows you the concept that we've reviewed all MRIs of 1,100 patients in the memory clinic, and we identified, we broke down our patients into, and I'll show you here, into those without medial temporal atrophy at all, in those with medial temporal atrophy but no amygdalar atrophy, those with medial temporal atrophy and moderate amygdalar atrophy, and those with medial temporal atrophy and amygdalar atrophy. And these are those that we labeled late, based on clinical findings. We don't have TDP43 biomarkers, of course. And in the next slide, I will show you the clinical progression. As you can see here, those who are progressing faster are those who have medial temporal atrophy and moderate amygdalar atrophy. Those who have medial temporal atrophy and severe amygdalar atrophy, paradoxically, they progress much slower. They progress as slow as those without medial temporal atrophy. And this is something that we know. TDP43 associated neurodegeneration in the medial temporal lobe, not the one of ALS and FTD, progressive, really slow. So I will leave you with my conclusion. Number one, MRI will be the mainstay of safety surveillance for anti-amyloid antibodies, monoclonal antibodies. That rates of brain atrophy is a valid marker of clinical disease progression, and it's a potentially valid confirmatory marker, but not for anti-amyloid antibodies. This we know from other studies. And the traditional volumetric MRI can be used to exclude the slow progressors from clinical trials on disease modifiers. And the last 7 seconds, I would like to leave you with this slide. We are at the Prada Foundation. It's a great place. It's a wonderful event. It's a place where we can exchange ideas, but we need the civil society to help us fill this gap. This is the gap between the cost of dementia, which is double that of cancer, and the funding for dementia research, which is between 10 and 12 times lower than dementia. I thank you for your attention, and I wish to acknowledge my co-workers for the last study that I mentioned. Thank you, everybody.
Giovanni, for keeping the time also, and of this last message is also well taken, I think. We go on. I mentioned MRI was very early on a biomarker. Can be discussed, perhaps FDG PET was actually probably the first biomarker tested in Alzheimer's disease, before that already. But that will be explained by Professor Daniele Perani. She is from, she's a neurologist and radiologist at the University Vita Salute San Raffaele here in Milan. Welcome.
Thank you, Philips. I am waiting for my presentation. Can see on my screen.
There it is.
Here we are. Okay, so we move to the, as you can see from the slide, to a tool which is very crucial in the assessment of molecular exploration of the brain, molecular exploration of the brain. I'm part of the scientific committee, and I prepare a workshop which was supported by Giancarlo as an important issue, Professor Comi, PET molecular neuroimaging neurodegenerative disease. So if the students, PhD and postdoc would like to see, there are very important issues on the role of FDG PET and PET in general in neurodegenerative condition, raising the importance in Alzheimer's disease, in the evaluation of brain reserve, which is very crucial in neurodegenerative condition, and also how artificial intelligence can support PET investigation. So I let you to look at the workshop registration. Some premises. PET is a well-recognized and unique tool for the in vivo assessment of brain metabolism, neurotransmission changes, protein load, and other molecular alterations such as neuroinflammation. The availability of PET is large, and it's important in research, in moving to clinical setting for diagnostic design, and also in the conduction of clinical trials. All the clinical trials use amyloid PET to select the patient. And so it's important for a prompt diagnosis, evaluating risk of progression. I'm going to show you some data about that, and maybe to target to see in vivo the effect of drugs on specific targets. In this position paper, we described the role of imaging, molecular imaging, towards the clinical and research aspects. And as you can see, FDG-PET is one of the important tools for the clinical setting also, whereas the evaluation of neuroinflammation is still completely in the research area. We can measure also, as I told you, neurotransmission systems, for example, the dopaminergic system in Parkinson's disease and in Parkinsonism. And one crucial aspect I always want to underline is that these molecular data need to be quantified or semi-quantified to be useful for research, of course, but also in clinical settings. So we have several methods, including at the end the molecular algorithm for binding potential, but we need to quantify our data or semi-quantify our data. So in neurodegenerative condition, we have FTG, we have the measurement of tau pathology inside the brain. We can measure with PET the large plaques in the brain, not the oligomers, just the large plaques. And alpha-synuclein traces are going on. We hope to reach a good goal in the near future. And then neuroinflammation with a sort of evaluation of microglia activation. So FDG-PET. FDG-PET is a funnel biomarker able to capture all the different pathological events, including tau, amyloid, neuroinflammation, that can produce perturbation in glucose metabolism because of synaptic dysfunction. Glucose metabolism is a measure of synaptic function. And so it's important to evaluate diseases, neurodegeneration, revealing specific patterns of degeneration, specific for each disease. I show you just a few shots of what I'm saying. The typical AD pattern, the typical AD pattern on top with the temporoparietal hypometabolism or the frontal hypometabolism in frontotemporal dementia, particularly the behavioral variant, Lewy body dementia with the involvement of the occipital lobe. These are patterns specific for each disease, and also in Parkinsonism. FDG-PET revealed its role in the differential diagnosis, so progressive supranuclear palsy, corticobasal degeneration, DLB, MSA, with the involvement of specific systems in the brain because they are dysfunctional. Here is an interesting results in Parkinson's disease where we evaluated a series of PD patients, sporadic, which presented or normal or very few premotor and subcortical hypometabolism, and other that already shown a pattern resembling Lewy body dementia or other kind of dementia. And in the long follow-up of these cases, we demonstrated that they develop dementia, only the cases who show large pattern of neurodegeneration. And this is another interesting study where we evaluated in the very early phase with Shayos alpha-synucleinopathy, the REM behavior disorders, where in almost all cases, apart 5%, there was an involvement of the occipital lobe, which show hypometabolism. You can see on the right single cases, and on the left the commonalities, the most important commonalities. So this is a marker of neurodegeneration in these prodromal phase. And now the follow-up is going on, and most of the patients will develop Lewy body dementia. What about amyloid PET? Amyloid PET can be measured in the brain, as you can see in the slide, in the part of the slide over there. And patient can have a large amount of amyloid deposition, or they can have nothing. And the images are quite clear. But there is a problem with amyloid that everybody knows, that amyloid can be present also in other diseases. It was largely demonstrated, and particularly also in normal elderly, up to the 40% after 75, 80 years. So this is a problem. It's a problem for the inclusion of cases in the preclinical phase, because they might not never develop dementia or Alzheimer's dementia. What about tau PET? Tau PET is emerging. This, I think, is a very important tool that we will have in the future. Can measure tau burden inside the neuron, because the traces can go inside the neuron. And there are interesting spreading visualization in cases with AD pathology, from the medial temporal region towards the lateral temporal and up to the other associative cortices, which really describe the diffusion of the tau pathology. And something which is very interesting is that tau pathology correlate with hypometabolism and not with amyloid load with a large plaque. And this was shown as the tau-mediated neuronal injury by tau pathology, and thus dysfunction, which really mimic, you see, the hypometabolism typical of AD cases. What about neuroinflammation? Only a few words about that. Neuroinflammation is a very, very important factor in neurodegenerative condition. With PET, we cannot measure the TSPO expression, which is an expression of the mitochondrial membrane, and it is possible. So the signal is not very high, but when neuroinflammation is present, we really can measure it with a specific mathematical model. And the cartoon you see over there are related to 1-MStic MSCI cases and an early onset AD, showing a huge amount of binding potential in the associative cortices. And my laboratory, my center, provide very important results on neuroinflammation, other disease, as you can see, Parkinson's disease, Lewy body dementia, prion disease, just to say that inflammation is a very, very common marker of the diseases, of neurodegenerative diseases, as we also shown in this descriptive meta-analysis, where we considered the different proteinopathy and the evidence with PET for amyloid in blue. You see, of course, the amyloidopathy are the most important correlate of amyloid in the brain, but amyloid is present also in alpha-synucleopathy or in tauopathy. And the tau is present in AD, but it's also present, of course, in the tauopathy and in other conditions. And neuroinflammation, that's what I am saying, is really across most of the diseases. This was well-known palette for the Alzheimer's definition with amyloid A, T, tau, and neurodegeneration. And for neurodegeneration, PET, FDG PET, is playing an important role, as well as some CSF tau measurement. And there are pros and cons of the ATN classification. In my opinion, tau and amyloid biomarkers are frequently, as I told you, observed as a copathology. So there is a disease, but as copathology is also amyloid. And also in several and other neurodegenerative diseases apart AD. So this biomarker positivity can be ambiguous. And in my opinion, it's particularly ambiguous in the asymptomatic individuals in the earliest disease phases. The important role of FDG PET is also to exclude neurodegeneration when very well examined and analyzed. So negativity, as Jagus described several years ago, since we have a high specificity for the diseases, a negative or normal scan exclude the presence of neurodegeneration. And this is very important as a time course of the examination of the cases. Why it's so important, the negativity? In this ADNI study, we took a lot of normal control cases and MCI cases who had FDG PET and amyloid PET. And we saw that the amyloid positive, either control or MCI, they decline more, the blue, than the negative. So amyloid has something related to progression, for sure. But looking at this population, more than 60% of normal and MCI amyloid positive, they did not progress. So we measure FDG PET in these cases, and we found that only the positivity of the PET predicted the progression. So the A plus, negative plus, and not the amyloid plus and negative neurodegeneration. And this was true for healthy control, who did not progress, and for MCI. I hope you can follow without the pointer, it doesn't work. So this is the reality. The positive scan, as you can see up there, were all the cases that in MCI and healthy control progress. The negative, they did not progress in the long, long follow-up of ADNI. So what's important for the for FDG PET? It works. Thank you. Sorry. Okay. Very well. Thank you very much. So it's important to consider it in this diagram that FDG PET and amyloid, this biomarker, can change before, long before the onset of clinical symptoms. So it's a challenge to use FDG PET in mild cognitive impairment, pre-MCI, and subjective cognitive decline. These are our studies. We perform a lot of, we publish a lot of evidence on that. What I want to underline is that the pattern I describe you in the full-blown dementia, they are present in the MCI condition when neuropsychological deficits are present. And the pattern predicts the kind of progression. And it's quite important. And this was repeated in a larger studies, 80 MCI included, where the pattern was positive, there was progression. A difference with the negative pattern, with a high specificity and high accuracy. Just an example of MCI subject with, of AD subject with different pattern according to the variant, the ID variant, posterior, logopenic, frontal variant, with the amyloid deposition, which didn't distinguish at all the variant. So behind mild cognitive impairment, there can be different diseases. And this is very important for the inclusion. And most of them can have positive amyloid, but different diseases. I go on with what Frisoni said before about this low progressor, the late, the so-called late. So we studied in a recent research a lot of amnestic MCI. They are clinically exactly the same, clinically, and this is a problem because they have a different progression. So we found at the beginning two populations, now you will know how I distinguish it, one who progressed, as you can appreciate by the neuropsychological, the data MCI and so on, and the other group who did not. So they were differentiated on the basis of the FDG PET. The progressors, they were perfectly adherent to the typical AD pattern, posterior regions. The not progressor, they just presented hypometabolism in the temporal median region. And as you can see, they were stable clinically, mental state examination, and along the time. Whereas these subjects, they are completely comparable, amnestic MCI, they progressed. So it's important to have a neurodegeneration sign. The CSF was not relevant, so very much mixed, whereas the other had a pathology quite evident, so CSF is very important too. This is a collaboration with the Geneva University Hospital, where we examined tau in this subject. And you can see that the temporal medial on top, they have almost nothing or very little tau in the temporal medial region, or nothing. Whereas the other, they all have a large amount of tau pathology in the medial temporal and in the associative temporal and parietal region. So also tau can really differentiate these two groups. Last data I'd like to present you, which is quite unique. It was recently published and was part of the project, an Italian project with Istituto Neurologico Besta, where we evaluate subjective cognitive decline cases and pre-MCI cognitive impairment, which means they are not full dysfunctional, but they have a lowering of the neuropsychological score, but still in the normal range. So they are preclinical or even normal persons. So the amyloid is in the majority low level, but also there was high amyloid load in this subject or intermediate. And the PET FDG is quite interesting because in this subject, 45% have normal function, normal metabolic function, but 55 had pattern. Most of them has the temporomedial pattern, other has the pattern characteristic of neurodegenerative condition, even if not so full exploded, but there was a pattern clearly recognizable. So in a chapter of a book, I try to define the discriminative power, and I think that FDG is important within and between differential diagnosis, and now tau is emerging as an important factor too, whereas amyloid is still looking at the published data in the literature at a difficult level to accept. So if the PET imaging neurodegeneration is important for the reason I told you, I don't want to go on too much, but the importance is a normal FDG PET scan will predict clinical stability. There are characteristic pattern of AD or other dementia, and so this is the importance for that. I took a slide of the last presentation yesterday afternoon, which point the importance of the role of synaptic disconnectivity in sporadic Alzheimer's disease and aging, and I appreciate it a lot because the FDG is really capturing this altered disconnectivity for whatever reason it is, but we see this phenomenon, and I think it's quite important to underline it. Last, other application in this ground, the study of connectomics. We are now merging with a multicentric study involving several PET centers in Europe that want to apply the studies of connectomes in neurodegenerative disease. We already published something on frontotemporal dementia and so on, Lewy body dementia. And what is interesting, you can also follow the neurotransmission pathway with FDG, because if the neurotransmission pathway is not working well, you will find alteration of the connectivity in this system. So we just study the Parkinsonian symptoms. To end up, I want to comment saying that molecular PET neuroimaging has enhanced our understanding of both the biology of Alzheimer's disease as well as the multifactorial etiology of other dementia. It is not proper equating the PET molecular biomarker to an effective 100% risk of progression to cognitive decline and dementia in normal controls and in subjective, for example, because if it's happened, there are a small percentage and it took a lot of time. FDG PET measures of brain metabolism, however, have shown high accuracy in diagnostic prognostic performance in MCI and dementia. A more comprehensive understanding of strength and caveats of these techniques, together with their optimal quantification and standardization procedure and their evidence-based clinical utility, will promote an informed use to take full advantage of these tools. And I thank you, my collaborators. Most of them were quoted in the paper that you will read, and the supporting. And thank you for your attention.
Thank you very much, Professor Parani, also for keeping the time and making a very strong message about the ATNN criteria, that you need both other letters outside of A as well. We'll go for a very exciting topic, which is actually body fluids, which is actually CSF and blood, and perhaps even more. And we'll hear about that by Hendrik Zetterberg, who sort of spends his life traveling between Gothenburg and London. And he is very well known for the work on the body fluids. And we're looking forward to Hendrik. What's new?
Thanks a lot. Thank you very much for inviting me to these exciting days. It's been super fun and also so good to have talks on different diseases. And I will show you a little bit about what we have learned from actually using biomarkers in biomarkers that were developed for Alzheimer's, for example, also in neuroinflammatory conditions, just to learn about how they work and learn about confounders and such things. And so I will give you I will stick to the neurodegenerative diseases mainly, and I will talk a little bit about the ATN concept. And but I would like to start with this slide. Yep. So this slide sort of summarizes what types of targeted biomarker assays that are available now. And these have been developed by different research groups based on some explorative work, but also based on targeted analysis of brain tissue and brain changes that relate to different types of neurodegenerative diseases. In this talk, I will focus on amyloid and tau and neurofilament light. If you design clinical trials now, it can be good to look also for markers that can be used exploratively in these trials, especially in phase 1 and 2, also if they are not fully established and standardized or so. So I will start with amyloid pathology, and I will start with this well-known thing that cerebrospinal fluid concentrations of the 42 amino acid long form of beta amyloid are lower if you do a lumbar CSF sampling. And this is thought to be because of retention of the protein in the brain parenchyma in growing plaques. This is one old slide where ventricular A-beta42 CSF levels were compared to plaque counts, and then you see this type of relationship between the number of plaques and the lowering of the CSF A-beta42 concentration. I have a slight suspicion that potentially microglia could be involved in this also. Activated microglia by amyloid could potentially also deplete the fluid a little bit from A-beta levels, especially the 42 amino acid long form. But this doesn't mean that it's not a plaque marker also, of course. We can compare with amyloid PET, as we heard, and then it looks like this, the relationship. So if you are in the upper left quadrant, you see that you have normal CSF A-beta42 concentrations and very little amyloid PET signal or no signal. And then if you are in the lower right, you're positive on both. If you're in the lower left quadrant, longitudinal studies indicate that you actually tend to move towards amyloid PET positivity. And it looks like CSF changes perhaps a few years before amyloid PET becomes positive. But I would say these are details, but still that's how the two markers compare. Of course, the fluids will never give you anatomic information. And here you see the obvious advantage of PET also when you evaluate the treatment effects of anti-amyloid treatments. There are confounders, and this is just one thing that we learned from our collaboration with our multiple sclerosis colleagues. The idea was a little bit to look at the effects of inflammation on A-beta peptides. And if you look in the C and D panels on this slide, you see that MS patients tend to have lower A-beta42 concentrations. And of course, they don't have amyloid pathology, although actually there have been reports indicating, based on this biomarker, that there might be some amyloid building up. But if you look in the A and B panels instead, then you see that also the shorter water-soluble forms of beta amyloid, those peptides are also reduced. And if you also measure the soluble APP fragments that are secreted into the CSF when alpha and beta secretases cleave the APP molecule, those are actually also lower. We have no clue about the mechanisms, but it's definitely not the pattern we see in Alzheimer's disease, because then you have a selective A-beta42 reduction. A very same phenomenon happens in normal pressure hydrocephalus. So here all A-beta peptides, and so these three A-beta peptides together with soluble APP, alpha and beta, are lower in normal pressure hydrocephalus. Originally, we thought that the A-beta42 reduction was because of buildup of the protein, because of CSF dynamics disturbances, but that's not the case. And the solution, of course, is to make a ratio here. So if you want the A-beta peptides to reflect amyloid pathology. One can make a ratio between A beta 42 and 40, or A beta 42 and 38, and then you get a very nice plaque pathology marker. So this is what it looks like. These are two different assays measuring the same thing. But in the A panel, you see on the red AUC, you see that A beta 42 by itself performs well. It's a good biomarker. But if you add in A beta 40 or 38 in a ratio, you get something which is almost completely concordant with amyloid PET positivity. And the B panel just chose another assay. And now there are several assays available. And in CSF, they correlate well. And we also have a reference material, so we can actually standardize assays so that we get the same absolute concentrations. And this is happening now. So in clinical chemistry, routine, across the world now, there is possibility of doing CSF A beta 42/40 concentrations, or the ratio. This is what it looks like in our lab. We used a Lumipulse fully automated assay that we run 3 or 4 times per week. And then we get this type of bimodal distribution of the biomarker. And this is from the validation paper. But these data are from, we just took all our data we have generated lately in the lab, and then we get a very similar pattern with a cut point which is very distinct and precise. So, and this is not, I work also as a general clinical chemist, and this is really an unusual, if we think about general clinical chemistry tests, bimodal distributions are quite unusual. This really indicates that there is something special with amyloid pathology, and that it's a distinct pathology. You either deposit or you don't deposit amyloid according to the CSF test. And you can actually determine very early on when you start to deposit. And this is shown in this graph. This is really a favorite study of where we've been involved measuring the fluids. So it's from the Madison cohort. It's called the RAP cohort. It's led by Sterling Johnson, and also these are data that Toby Betthauser has analyzed. These are people who are cognitively normal, but they have 1 or 2 family members with Alzheimer's disease. So they are a little bit enriched for ApoE4, of course, and they have high risk of developing Alzheimer's disease because of this family history, of course. But here you see that they have then enrolled in a study where they give repeated CSF samples and do repeated amyloid PET examinations. And in the left graph here, you see the CSF A beta 42/40 ratios. And you see that no one below 50 is abnormal. So all young people maintain A beta 42 in solution somehow. And I think that is fascinating, actually, that it's so distinct. But from the age of 50, people start to deposit, and they drop in the A beta 42/40 ratio. And it happens quite quickly. It's messy slides, so it really shows the data as they are. But within 1, 2, 3 years, people go from being normal to becoming abnormal. And then there is this floor effect, which probably makes sense from a biochemical standpoint, as the biofluid is sort of depleted of A beta 42 at a relatively constant rate, and new A beta is formed all the time. If you look at the amyloid PET signal, you see amyloid PET was not done between 40 and 50 because of cost reasons, and most would be negative also. But here you see also that people start to cross the threshold of positivity, and then they increase in a quite parallel fashion, which is fascinating, because then it means that when you have the amyloid cascade on your brain, it is a chemical process, and you can't do much about it by the way you live your life, which I think is interesting. And of course, amyloid cascades, the best way of stopping them is to remove the substrate. So that's what we hope the antibodies are doing, of course. Toby, he sorted people according to their slopes of the CSF A beta 42/40 change, and then he could define the time point 0 for onset of amyloid deposition. And then you see that amyloid PET starts to become positive a few years later. If we go to plasma, this has been a difficult, we have already talked about it a lot. It has been possible to measure A beta peptides in plasma for many years, but the assays have always been a little bit suboptimal. If the first assays we worked with, if we spiked in A beta, we could not recover it. Basically, it disappeared in the matrix of the complex plasma sample. And you see here, these are data from the old biomarker database that is available at Alz Forum. There is no pattern here for A beta 42, and the different assays didn't correlate well. But in 2017, this very important paper came from Randy Bateman's team, where an assay based on immunoprecipitation mass spectrometry was developed. And this assay has also been developed in a way that you extract all or most of beta amyloid from the plasma sample, and then you measure the 42/40 ratio by mass spec. And then you get this type of difference between amyloid PET positive and negative people with a nice group separation and also an AUC of almost 0.9. And thereafter, this paper came from a Japanese-Australian collaboration, also using a mass spec-based method, but using an inverted ratio, 40/42, and then you see this type of increase instead. So basically, I think these two studies really corroborate each other, and we have also developed a test where we also use mass spectrometry, and we used it on the INSIGHT 46 cohort in the UK, where people were cognitively normal, 20% were amyloid PET positive, and this could be detected by the IPMS method for plasma beta with an AUC of around 0.8. So it is really a test that starts to look replicable and reproducible and useful, actually. But mass spec, of course, it's a tough assay to run. It's you can scale it up, but it's tough. So we have worked with Roche to try to develop an automated immunoassay, and here you see raw data from that collaboration on the most validated form of the assay, which is performed on the Cobas instrument, a fully automated assay. The technician puts in the sample in the machine, and then a beta 42/40 concentrations come out. And then you see this type of group separation with amyloid PET people to the left, and negative people to the left and positive people to the right. And you see this type of group separation, but with this assay, we can't really use it for individual diagnostic purposes. What is possible with this type of assay is to enrich a population for amyloid positivity. And this is, of course, super relevant if you want to design clinical trials and reduce costs. Because if one then would like to do a pre-screening measuring a beta 42/40 ratio, accepting that not all will be positive, accepting that you might also include a couple of negative people and you will miss some positive people also, then it's fine if you confirm the result with this type of assay. But I think Randy will show data on that also we are approaching usefulness of a plasma test by itself if you also consider ApoE4 and age and some other parameters. So this has really started to take off in a quite strong way. And then one could say also, if it looks like on this slide, would we use, I will show you some plasma phospho-tau data. It really is like this, that if we move into the preclinical phase of Alzheimer's disease, then a beta 42/40 ratio performs a little bit better than most of the phospho-tau tests. So there is really room to continue to work with this type of relatively difficult marker, but still. Here I just wanted to highlight the challenge, which also is relevant if you design a clinical trial and want to use plasma beta for pre-screening. In the B panel, you see the CSF beta 42/40 ratio, a reduction of 50%, little overlap, quite easy to maintain such an assay stable over time to detect this difference. But to the left, in the A panel, you see that the reduction in A beta 42/40 ratio is small. So, and this means that you have to be super careful when you pick your laboratory, because the laboratory has to be able to measure this type of change, especially if you do pre-screening week by week, including patients to your big clinical trial. The laboratory has to be able to keep the assay super stable during the recruitment time. And this works, but you have to select the lab carefully. So if we think about the use of fluid amyloid biomarkers in clinical trials, pre-screening for amyloid enrichment, plasma beta 42/40 ratio, it works, but be careful with assay selection. Diagnosis, if you want to verify amyloid positivity, then definitely CSF beta 42/40 ratio is what most would recommend at the moment. But we might approach a performance of a plasma test too. And then detection of treatment effects. This depends on the mechanism of action of the drug. So if your drug is an anti-amyloid antibody and that drug will be present in the sample you will measure A beta, you have to check if that interferes with the measurement. And it could also be that the drug changes the half-life of your biomarker. And that's actually relevant for all biomarkers that you select for your clinical trials. Make sure what happens to the biomarker measurements in the presence of your drug. So that's an important phase 0 and phase 1 activity to establish that. Now, let's move to tau. Phospho-tau is increased in lumbar CSF, that we all know. We also know for many years that the levels of tau, the increase we see in total tau and phospho-tau is relatively age-specific. So other neurodegenerative diseases where there is also tangle pathology are typically negative for the fluid-based tau markers. This could mean that the amount of pathology is a little bit less if we talk about tau phosphorylation as a brain pathology than in Alzheimer's disease, where actually phosphorylation of tau it's very widespread and abundant. So it might be an amount issue, but it could also be related to something that I find super interesting, and it's been shown by many researchers now, both in animal models and cell models and in patients, that when you get amyloid pathology, neurons that are close to that pathology will react by phosphorylating and secreting tau. It almost looks like an amyloid-induced pruning mechanism. Hibernation was on a slide yesterday, and hibernation also goes with tau phosphorylation. Down-regulation of neuronal activity goes with tau phosphorylation. The highest phospho-tau levels you can see in humans are in the newborns, where there is massive synaptic pruning going on, physiological synaptic pruning. So that's how I sort of think about the phosphorylation of tau, that it's an amyloid-induced event. Neurons phosphorylate tau lets go of the microtubuli, and then it's secreted also. So you have total tau, phospho-tau increase in amyloid-positive people. Total tau can increase in some conditions, as you know. Stroke, of course, then you have a release from dying cells. Creutzfeldt-Jakob disease and extracellular amyloid will also increase total tau, but not phospho-tau with most of the assays. A little bit with 217, I think, and perhaps some of the... Yeah, there are so many phosphoforms. But that's very interesting, I think. So this also means that phospho-tau becomes such an interesting marker in the clinical trials of anti-amyloid treatments, because then if you detoxify amyloid, phospho-tau should go down, as we have seen it does now. So it's, there are many phosphoforms of tau. I won't go into details here, but tau has so many phosphorylation sites, and many can be measured in CSF. You can sort the different phosphoforms a little bit on the basis of the fold change in different disease stages, and it appears like the phosphoforms increase a little bit differently in terms of how early they increase in biofluids, with 231 and 217 typically being quite early markers. And 217 also has a very interesting stepwise increase the more advanced the disease gets, whereas 231, for example, plateaus. So one has to be careful with I think in clinical trials we should measure many phosphoforms of tau. Here's an example of what happens when amyloid starts to accumulate with plasma phospho-tau 231. So already with a little amyloid in the brain, you get this type of increase. We can combine phospho-tau with cognitive tests that are really basic, and then reach an AUC of 0.9. So this is sort of the easiest test, and then you reach approximately 0.9 when you have patients with subjective cognitive impairment, mild cognitive impairment, and then this is a test for how well the prediction of Alzheimer's dementia is 4 years later. Here you see donanemab effects on phospho-tau 217, a reduction after just 12 to 24 weeks. In aducanumab, it was also a similar effect on phospho-tau 181. So we can use it for pre-screening for amyloid and tau enrichment, plasma phospho-tau. We could use it also if we want to verify the effect. Then, of course, CSF phospho-tau exists, and plasma may be as good, actually. And then we have detection of treatment effects with plasma and CSF phospho-tau. So that also looks quite promising. Now I will spend a few minutes on neurodegeneration. 1 minute, exactly. I'll turn off my alarm here because neurofilament light, as you know, increases in most conditions with neurodegeneration. And here's just a busy slide showing the different diseases where increases have been seen. And the diseases with the most strong increase are HIV-associated neurocognitive dysfunction, frontotemporal dementia, ALS, but then you also see it quite clearly elevated in vascular dementia. If you have high NFL, you lose, typically in MCI studies, you lose more cognition. You lose more brain volume, as we have seen, and it's a responsive marker to treatments, as we heard from our MS colleagues. Here is spinal muscular atrophy kids, treated with antisense oligonucleotide-mediated stabilization of the deficient transcript. And then you see very rapid normalization of NFL when neurodegeneration stops. In Huntington, it has been less clear, but in Huntington you also see increases that have been related to toxicity. So I would like to highlight the possibility of using NFL almost as a safety marker in parallel with being able to see a reduction, a positive effect by treatment. Lecanemab had very noisy data on NFL, but there could be a stabilization. We will hear much more, I think, in a couple of weeks if we are lucky, and perhaps the biomarker data will be presented later on at ADPD when this congress happens in my hometown Gothenburg, so please come. It correlates, you have nice correlations between plasma and CSF, and that it really indicates that also the dynamics between CSF and plasma after acute injury are similar. And I think actually that I will close with that and just pinpoint, mention then that pre-screening for neurodegeneration, then you can definitely use plasma NFL. Diagnosis verification, CSF NFL, but plasma might be as good. Detection of a treatment effect, both plasma and CSF NFL will work. If there is a disease with a clear biomarker elevation, of course, and then safety. And there's Altforum, where you can look at all these other biomarkers that I've been mentioning across neurodegenerative diseases. Thank you.
Thank you very much, Henrik. So I invite all the speakers to sit next to me here, but before we start the discussion, and please, you have all the questions from the first three speakers already in your head, but you can only ask them when the panel is here convened. So please keep your questions to yourself. There are a lot of questions I could ask actually, but that's not my task here. So we have first two short discussions, and we'll start with Professor Hans-Peter Hartung. He comes from the MS field, and he is a professor at the Heinrich Heine University, Düsseldorf in Germany, and he will give his perspective on the biomarkers in MS.
Yes.
Okay. Well, thank you very much, Philip, Professor Comi, organizers and Prada Foundation. My heartfelt thanks for inviting me to this fantastic meeting. So switching gears again to multiple sclerosis and briefly discussing the role of body fluid biomarkers who basically subserve the same functions as the other type of biomarkers do, and they are listed here. And if we were to reliably integrate them into our management process, we would certainly be able to come closer to precision medicine also in multiple sclerosis. Now, diagnostic biomarker, we'll probably hear more. MOG antibodies, aquaporin 4 antibodies have allowed us to differentiate MS from NMOSD and MOGAD, and I will provide a couple of examples for the other functions of the fluid phase biomarkers. Let's first keep in mind that obviously we want to be together close to the act of crime as we can, but there are practical limitations to this. So most of the body fluid biomarkers investigated in more recent times are from blood or serum. This is a very nice, now slightly outdated review published by Comabella and Montalban 8 years ago, listing based on the better immunological understanding of MS, the multitude of incriminated, presumed biomarkers. Some haven't stood the test of time. There are reasons both related to technology as well as statistics. But as we heard earlier, the thing that has really emerged as the most robust body fluid biomarker has been neurofilament light chain, and this has been alluded to several times here. For example, just to show how the levels of NFL and another, I think, important biomarker, chitinase, determines the further evolution of the disease. Here, patients with this so-called radiologically isolated syndrome have been followed as they developed CIS, clinically isolated syndrome, or MS, and it has become clear that with high baseline values of both NFL and chitinase, the time to this conversion was shortened. To gauge disease activity, SNFL has proven to be useful. Here is a study that looked retrospectively at baseline SNFL levels and the occurrence of radiologic activity in the preceding year, and the fifth quintile high SNFL baseline values allowed to detect with high probability retrospectively MR activity and mirrors of disease activity. And the authors suggested that one might replace follow-up annual MRIs by just measuring SNFL. Here we see look at the prediction of the disease course worsening in terms of disability and brain atrophy. Again, those patients with highest SNFL ended up with the highest disability as reflected by EDSS, or the evolution in this series over up to 12 years of an increased brain volume loss. So if we try to summarize what we can gain from applying this specific body fluid marker SNFL towards the management of multiple sclerosis. Number one, it may be useful in the diagnostic process. Number two, it may allow to gauge inflammatory disease activity, prognosticate the disease course, and importantly, judge treatment response, and finally also make predictions. This is a summary of papers that looked at the treatment response to disease-modifying therapies in MS in terms of SNFL levels, and just in summary, all these DMTs that work in relapsing MS did diminish SNFL levels. In progressive MS, this has also been studied in the various trials are indicated, and interestingly in the INFORMS trial of fingolimod in primary progressive MS, where the clinical outcome was not met, there was an impact of fingolimod on SNFL levels at 24 months. This is from the anti-CD20 monoclonal antibody trials in RRMS and PPMS, opera and oratorio, and uniformly you could see that ocrelizumab did decline levels of SNFL. Another example from siponimod and fingolimod, highlighting the difference between SPMS and RMS. Patients with SPMS have higher levels of NFL at baseline than the relapsing remitting patients at the same age. And this is more conspicuous here, where you see that medium levels of SNFL are more associated with SPMS, higher than with RRMS. And again, siponimod as a treatment for SPMS was associated with lower SNFL levels. ASCEND was a trial in SPMS of natalizumab. It failed its primary clinical outcome, but again, SNFL measurements detected an impact of natalizumab. As you can see, SNFL concentrations were significantly lower. And coming back to an important paper from Sweden, which studied CSF, NFL, again showing for the first time the response to DMT reflected in a decline in the levels, and importantly, as will be discussed later, I'm sure, GFAP also as a potential body fluid marker. Now, sort of disappointingly, when assessing neurofilaments as biomarkers of neuroprotection in 2 recent trials, the outcomes were negative. Those 2 trials, simvastatin had been mentioned before, and ibudilast were positive in terms of slowing brain volume loss, but there was no reflection of any therapeutic impact on SNFL levels. Finally, SGFAP has also been studied more recently and shown to be higher in progression versus acute relapsing multiple sclerosis. And finally, as I mentioned, kinases. So let me conclude. Numerous body fluid biomarkers have been examined in MS. AQP4 and MOG antibodies allow the differential diagnosis of NMOSD and MOGAD. OCB and is diagnostic in CSF. That was discovered in the late 1950s and appears to be prognostic, in particular the IgG and IgM. NFL has emerged as the most robust body fluid biomarker, which can be measured reliably with high sensitivity, not only in CSF, but very importantly, for practical reasons in blood. These measurements increase diagnostic accuracy, predict disease cause, and when measured longitudinally, foretell disease activity and evolving disability. And multiple studies in RMS demonstrated NFL responsiveness to DTMT. Treatment response by body fluid biomarkers may be integrated in aggregate disease activity outcomes such as NIDA, NEPAD, and service trial outcome. They may alert to subclinical disease activities, silent progression or superimposed inflammation to prompt further evaluation and aid in DMT monitoring and adaptation. Still, I think future research is required before implementing them into clinical management. Thank you very much.
Thank you very much for this concise overview of the biomarkers in MS. And we'll quickly go further before you are allowed to ask questions to Professor Fudong Shi from the Department of Neurology and University General Hospital in Beijing, and I guess he will be virtual. Is that true?
Okay.
Thank you.
There he is.
Can you hear me and see my screen? Yeah. I'm trying to share the screen. So. So can you hear me?
Yeah, we can hear you, but we don't see the slides yet. So we see you, your office, the curtains, the books, I think.
It does show that I'm sharing my. So.
Something's happening. Yeah.
Voice or video?
Let me share again. It says something is wrong with Zoom, but I'm trying to share again. So can you see it now?
No.
All right.
So the unfortunate thing is that we can't hear Professor Dongxi giving his presentation. The fortunate thing is that we now have some time for discussion, because we also have a keynote lecture following this discussion. So I invite the audience to come up with your pressing questions to any of the speakers here next to me. Who? There must be, because it's so... Yeah, go ahead. Michael Schwartz, you're the first. Short questions, short answers.
It's a comment. So should it be short or long?
Short.
Okay. So the comment is that I think that we have spoken a lot this morning about biomarker, fluid biomarker, and I think we should distinguish between biomarker for early diagnostic and biomarker that can be a marker for disease modification, which are two different issues, and they can come early. We don't know the nature of this marker, but this is what we need to search for.
Okay, a fair comment. Any comment back from the panel?
I think sometimes I think about biomarkers more like markers of something happening in the tissue, and that the use could be both of the things you mentioned. But of course that can be discussed, and it's important to. What I would like to say that it's super important to check what happens to the biomarker as such when you measure it in the context of a treatment.
Okay, we have a question over there. First, yes, there, and then you are second, and then Giancarlo is third. Sorry.
Yeah, can I? Okay, just a quick question.
Yeah, go.
Okay, just was impressed that fluorodesoxyglucose looks a very promising biomarker for most of dementias. This is my question is. Does it mean that glucose hypometabolism may represent a common denominator for most dementias?
That's for you.
Yes, sure. As I presented, the hypometabolism is the result of several pathological conditions that disturbs the synapses. So the typical specific pattern that we saw in the different diseases, that's render the FDG PET a biomarker, because it's not for all the diseases, but it's with specific pattern, even in the early disease phases. So it's specificity and sensitivity, both things.
Okay. You?
So when we look at the distribution of the patients in your clinical trials, they're pretty scattered. They're not clearly responders or not responders, even in the imaging side. And so I wonder, it reminds me a lot of some of the cancer patients and how they respond to therapy, and the fact that we developed and learned to add other criteria and parameters to stratify these patients. Is there any approach in your field in which what seems to be a completely scattered pattern is instead something where some order can be brought when other criteria and other variable, and what's the trend?
I can answer the question, but the panel should. Giovanni?
I believe that from what I've shown, ApoE is going to be a variable that should be always taken into account in clinical trials, and it should be, I believe that it should be an a priori stratifying variable, not a posteriori as it's being used now. And this is for sporadic Alzheimer's disease is probably the most relevant biological factor.
I think to summarize the whole issue of the biomarkers is that it allows us to do precision medicine as we do in oncology. So we are just keeping up with your field, actually. Giancarlo.
To Dr. Zottenberg and Hartung, how much of the contribution to the neurofilaments is due to the ongoing inflammatory activity? How much is expression of the degeneration process? Is there a way to try to come up with this estimation?
The short answer is that it's not known, but it would be important to figure that out somehow. And exactly how to do that, I do not know, but it's very clear now from studies that are being presented in your field that in progressive MS, NFL somehow doesn't really work as well as I had thought originally, and instead GFAP shows up over and over as more related to progression of that type of MS. So basically, either one tries to figure out why this is the case for NFL, perhaps also develop new neurofilament light tests, new neurofilament tests like alpha-internexin and such things, or we start to use GFAP and some other markers in parallel.
Randy.
Yeah. So on several of the different diseases, the biomarkers oftentimes exceed the clinical final state of the patient. So the response to, for example, amyloid plaque removal can be 80-90% back the way towards normal, and still a 25-30% slowing of the clinical disease. Same for multiple sclerosis, same for other diseases that have clear clinical benefit, but the biomarker response is really exceeding that. So I guess two questions. Are there biomarkers that don't do that, that match much more closely the clinical progression rate of the disease from the patient's perspective, not radiographic, not other biomarkers, or why is that?
Who wants to respond? You?
Refer this to...
we all refer everything to
I think like this, I actually like that there is this disconnect, because to me, at least in the neurodegenerative space, it makes sense, because you have clinical symptoms because of network disturbances and breakdown of networks. And then there is this type of disconnect between onset of these pathologies and the network disturbances. So I am hopeful that we will, of course now I'm perhaps a bit over optimistic here, but I really think that we will see that these biomarker changes, if we initiate the treatments before these network disturbances, the biomarker normalization will be predictive of less symptoms in the future. So that's the disconnect. I would really like to creep down into the brain and see what happens in these. I mean, imagine the plaques and the networks around the plaques and all the tau and all the microglia and astrocytes. It must be a mess. So I'm not surprised that there is this type of disconnect. And I think still that the biomarker changes are, I really hope, I actually think that they will eventually predict.
But just an additional comment that the cognition goes to the dysfunction. So even in the follow-up and the evaluation, the follow-up of treatment, I think that the measurement of neuronal function is crucial, and to see if it can reduce or even goes over, related to the improvement of cognition.
Giovanni?
You're referring to the biomarkers? The biomarkers that you may referring to are those that are closer to clinical symptoms, such as neurotransmitter activity. But the closer to the symptoms, the closer to the patient health-related outcome, but the further from the pathophysiology, the real the upstream cause of the disease. So it depends on what you need the biomarker for.
This one goes to Henrik, but also to Daniela and Giovanni. So what's your recipe in terms of longitudinal assessments? How important are longitudinal repeated measurements, and how we are going to sell this to the public and to the insurances in terms of costs? We have really a big problem in Germany, also in Luxembourg, to justify the costs just for a simple lumbar puncture and CSF determination of all these great markers. But we have to probably measure over decades in a sequential manner for early detection and intervention. So how important are repeated measures?
Well, Michael, if I can go first, I believe that what we need in our field is a kind of Framingham study. We need a long preclinical longitudinal study with frequent measurements that allow us to measure the changes accurately and sensitively, the changes over time of a number of biomarkers, and figure out the time when the measurement is more effective to screen patients for secondary progression. This is something that I really believe we should do in our community. Something like Diane has done for the familial Alzheimer's disease. We should do it for sporadic Alzheimer's.
Can I just, Hans-Peter, in the MS field, it's quite common to do repeat MR scans. I mean, you're completely sort of happy with that. I mean,
with MR, yes, we never liked CSF, repeat CSF as much as our Swedish colleagues, but
the Dutch.
Yeah.
No one does. Okay. Quick.
Yes, very fast. This is crucial for research, as Giovanni said, but in the clinical application, the cost is very, very high using molecular imaging such as PET, but in any case, amyloid PET is used for the inclusion. I think that sometimes it would be better to have a CSF measurement of amyloid status and perhaps look at the neurodegeneration absent or present.
And I could just briefly mention that, of course, we hope that the blood biomarkers will make it possible to do repeat tests in the clinic. And in the context of a therapy, it will be much easier to advocate for that, I think.
I think the blood-based biomarker are the solution here because they will be cheap. I have a question for Professor Lassman. Short question?
Yeah, just a very short question. To what extent the neurofilament biomarker in the serum is actually diluted by things which are not related to the disease process? And I give you the example of progressive multiple sclerosis. About half of the patients, or even more, have secondary pathology in the peripheral nervous system with Wallerian degeneration and so on, which is expected to actually give higher neurofilament levels in the blood than anywhere. So in that case, probably there should be discrepancies then with CSF.
Yeah, and that's completely true. We see it now. We have done plasma NFL in clinical routine now over 3 years, and we see also discordant CSF and plasma NFL results. And this is exactly what you describe. Pure peripheral neuropathies typically have high plasma NFL and normal lumbar CSF, for example, lumbar CSF levels. And peripheral neuropathies increase NFL, so that's an important thing to consider. And some other things are also confounding the results a little bit. Kidney disease for unknown reasons, active cardiovascular disease also makes the biomarkers a little bit more shaky, but not. Phosphatide looks reasonably okay.
Is that also the case for plasma p-tau?
It is, epidemiological studies have just been published indicating that there are such effects. But a few weeks ago, Oskar Hansson's team published a study where they actually tried to address if these somatic, or what you should call it, comorbidities, if the influences destroy the biomarker's diagnostic performance, but that's not the case. So it's like large sample collections, small but statistically significant changes, interesting to explore. And I forgot to say that what we need to do, of course, is that neurofilament light we have to know about the peripheral contribution, and we should develop an alpha-internexin test. That is definitely what we are trying to do that, but we are not there yet.
Next year.
That's a CNS-specific form.
Yeah. Last question from you.
Yeah, a very short question to Hans-Peter, maybe more a comment. You gave a very nice overview of the different biomarkers in MS, but you didn't allude to the neurophysiological markers. And I think that in the emerging field of repair, it's important to add neurophysiology, the visual evoked potential, which is very closely related to repair of myelin and conduction. So what do you think of that?
Absolutely right. I was just dutifully responding to the task of restricting my comments to body fluid markers, but I think it's been very clear, particularly also in the progressive stage of the disease, that neurophysiological assessments provide invaluable contributions in assessing that. Yeah.
Okay,
absolutely
so we
right.
agree.
Yeah.
Thank you very much. So Randy, before we give you the keynote, we have to go back to China because in order to be collegial, he is now online and he wants to address us at the end of the discussion on the biomarkers in MSOD and MOGAD. Professor Dongxi, are you there?
Yes, I am.
Welcome.
So, can you hear me?
Yes.
Yeah. Okay. I apologize for the technical issue. That I'm very honored to be invited by Dr. Comi and the committee to talk about biomarkers in NMO and MOGAD. So first slide, please. So my practice and the research are based in Tianjin General Hospital and Beijing Tianda Hospital. It's only 30 minutes by the bullet train. Next slide, please. So we recognize NMO and MOGIC as separate disease entity relatively recently when we discovered the antibody to AQP4 is discovered. And then around 2013, there was a proposed separate entity of MOGIC for individuals that we initially characterized as NMO that without antibody to AQP4. And subsequently, the antibody to MOG was discovered, and some of the pathological properties are proposed. Next slide, please. So we see that the target antigen in these two conditions are very different. In NMO, we see astrocytes bearing AQP4 being attacked by the IgG4 antibody. On the other hand, oligodendrocyte in the MOGIC was a target for autoimmune attack. So there is a subsequent pathology which is shown in the next slide. So there is similarity of subsequent pathology, including demyelination, which is secondary to oligodendrocyte damage and astrocytopathy, and eventually all conditions will probably lack axonal damage, like what is happening in multiple sclerosis. Next slide, please. In terms of biomarker, currently we are focusing on detecting the antibody to AQP4 and MOG, which is very important for diagnosis and different diagnosis. GFAP has been recognized recently as a potential marker for the disease progression. On the other hand, the roles of NFL, and in some cases, the TREM2 is being studied, and some evidence emerged that it may have a role, which need validation. Next slide, please. So the dilemma for detecting MOG antibody and as well as other antibodies is very pressing, because the level of antibodies are very kinetic. They tend to be high during acute attack, but become normal or negative after the attack. So in that case, a higher cutoff for seropositivity or use of specific secondary antibody is very much needed to enhance the specificity. We all know that cell-based assays show excellent agreement with each other for higher positive and negative samples. However, in samples with low positivity or borderline antibody, the results are very ambiguous and in some cases discordant. So we have to somehow improve the detection methods. In our case, we use DSA amplification system because it's important for both for diagnosis and for clinical trial as well. Next slide, please. We are adopting the TeraMat signal amplification methods in an attempt to enlarge the signal to 3 times while preserving the conformation of antigen. Its value is being tested, and we also run sample from Yale in parallel so that we can see in such a fashion we can find clinical correlation as well as the sensitivity and specificity. Next slide, please. So we have a study ongoing trying to compare this TSA amplification versus conventional CBA, if it does have a role in amplifying the antibody signal for those with borderline or lower titer. In that case, it will be very useful. So the study is ongoing. Next slide, please. NFL and GFAP levels being detected in 2 studies listed here. There is agreement between the studies. The levels are very high. They become higher during relapses than in the remission. The level between GFAP and NFL ratio is probably has some potential diagnostic value. They are changing, we all know, but the value of these markers needs to be validated in large cohort, apparently. Next slide, please. So I'm going to talk about the remaining minutes, some of the new marker discovery and its validation of its role. We try to make use of our clinical imaging resources that are being established in our two hospitals in Beijing and Tianjin. Next slide, please. So one approach we are doing is we are trying to detect the exosome because they carry signal of cells that being destroyed or have a pathological feature. In doing so, such signal can be detected in the peripheral blood, may reflecting the changes of the brain. Next slide, please. So this is we know that exosome has many functions in pathology and pathophysiology, and in conjunction with GFAP, we can easily differentiate its origin in terms of where it comes from, in example, astrocytes. Next slide, please. So we saw that there was a greater proportion of proteins that carried by exosome that are upregulated or downregulated. And when you look at these proteins, and they all come down to immune reactions, cell death, and metabolic signaling. So it comes very close to pathology of NMO as we are currently recognizing. So on the other hand, new proteins like ApoE that has not been previously described playing any role in NMO has been emerging from this type of assay. Next slide, please. So when summarizing the proteins, we see changes. There was TGF-beta and complement activation protein. Interestingly, we verify some of these proteins with our other approach. We're focusing on 100 CXCL10. They seem to have some correlation with disease activity. Again, we are validating this finding in large cohorts of patients. Next slide, please. So I'm going to talk about ApoE a little bit because it's pretty intriguing for our group to look at the presence of ApoE that carried by these vesicles in the peripheral blood. This is a flow cytometry. You can clearly see it's elevated, and also compared to healthy individual, it's rather apparent elevation. Next slide, please. So when we inject a portion of this ApoE into the mouse model of NMO that being induced by infusion of IgG isolated from patients, we see the reduction in lesion size as well as the reduced number of proliferating microglia. So it seems to have something to do with glial activation and interaction with microglia and astrocytes, which is being investigated. Next slide, please.
Professor Dong, I really have to ask you to finish because we are running really out of time. Can you?
All right. Okay. Okay. So this is the chain. Next slide, please. We see the level as well as preliminary association with the clinical presentation. Next slide, please. I think I'm going to just skip the imaging data on 17. Next, please. So there is some recognition of our group in terms of mock imaging data, but next two, please. And on 17, they're presenting next slides. Very unique cortical lesions that are not previously revealed by 3T. Next, please. Next. And I'm going to skip the rest slides. I'm going to make one point here that imaging, well, the marker can be seen in a very high dimensional approach, like for example, 10x single-cell sequencing. By doing so, we recognize compartmental specificity of immune cells. We discover new biomarkers as well as some markers that relate to new therapy. So we're actually conducting 2 trials related to rituximab refractory patients. In those patients, the buff appears elevated, so we're using Biomarkers to try to have a single arm small trial going on those trials. Similarly, we have discovered that BTK is very dysregulated in B cell in the NMO, so it's again we are organizing a large trial sponsored by Sinomab to try to see BTKI has any place in therapy of NMO. I think that's what I'm going to make. Thank you very much. I'm sorry for the technical issues, and hopefully going to Milan next time.
Thank you very much. Greetings from Milan, and thank you very much. So now we are up for the last part of this morning for the keynote lecture from Randy Bateman. He's from Washington University and better known as Diane. It's not his first name. It's actually not even the children's name. It's the thing he set up with the Dominant Inherited Alzheimer's Disease Network, also with the trial part in it. And it's really groundbreaking work. And I think he is the best person in order to enlighten us on new clinical trial designs. And for those who are craving for lunch, I mean, the reward is even better if you just wait a while. And that's while you are listening to Randy Bateman.
Okay, we've heard for a couple of days that prevention is very important in Alzheimer's disease, and even though I have a lot of information to tell you about, I'd like to ask the organizers to turn up the lights just a bit. If everybody could just stand and stretch a little bit. Sitting is the new smoking, I hear. Go ahead, stand, please. We've been sitting in this hall all morning and haven't had quite a chance to stretch. There we go. Everybody just stretch it out. And let's do the little calisthenics, breathe a little bit. All right. And can we have the lights up? It's a beautiful day outside in Milan. No, up, brighter. It's gorgeous outside in Milan, and we're in this jet black hall. It's dark. All right, I'm going to move quick because I'm the only thing between you and lunch, and we've just had a few snacks. I really appreciate being invited to this venue, and this morning has been a highlight. Yesterday was great to lay the groundwork about prevention and everything else, but this venue is important because we have colleagues from other disease areas to share our ideas with, our thinking with, and that's where the magic happens. It doesn't happen from me telling you what I know. It happens between us, what happens when we communicate and dialogue. And so I really appreciate that part of the meeting. I hope we continue that. I'm going to cover a broad area, but I'm going to focus on a case example. Please don't go to sleep. I know they keep turning down the lights on me, but I don't know why. Clinical trials designed to accelerate therapeutic delivery. And I want to share with you what I think is one of the most exciting times in the history of Alzheimer's disease. We are on not the verge, we have passed that threshold. We are now in an era of changing the course of Alzheimer's disease. And many of us are already thinking about the next steps. What can we do next? What can we do more? How do we have a bigger impact? But I don't want to lose track of the fact that we now have conclusive proof, in my mind, phase 3 trials, well-run phase 3 trials that clearly demonstrate for the first time in the history of this disease we can change the course of it. And so my presentation is going to focus on how do we accelerate that? How do we leverage that? What else can we do? And actually, most of what I'm talking about has already been mentioned, sprinkled throughout the meeting. So I don't think I'll say a lot that's new, but I will try to highlight these points. I want to start with the history of treatment development for Alzheimer's disease. Alzheimer's, as you all know, was originally identified in 1906 and described by Dr. Lois Alzheimer. However, it's been known throughout history. It's been with us as long as we've probably been modern humans. It's just been rare because we haven't lived long enough for many of us to experience it. But you'll see that on this timeline, there's a lot of developments that occur, but really the first set of specific drugs for Alzheimer's, thank you for the lights. The first set of specific drugs for Alzheimer's disease really came in the 1990s, and these are neurotransmitter modulators. These change the way the neurons communicate. It was fantastic we had something to treat the symptoms by some modest amount, but it didn't change the course of the disease. And it was the discovery of the molecular biology of the disease, the proteins that make up the pathology, the genetics behind that led to the ability to develop treatments that could target that pathology. And I'll try to show you how we've developed to reach these first impressive thresholds of being able to nearly completely remove one of the pathologies and do it in a way that has a clinical cognitive impact that's meaningful to patients and to family members. It's not a cure, but I'll point out there are relatively very few cures in medicine. And in fact, amongst even our most successful diseases, multiple sclerosis and other diseases that we treat, we're in that ballpark. We're in the same ballpark as the successes. So for Alzheimer's folks, way to go. I mean, this is great. We're no longer the untreatable disease, right? We're there with our colleagues. I think this is fantastic. Now the race is on. Can we go faster? Can we do more? I put this here just to highlight this 2022, what I think is now conclusive proof. We've seen this coming in multiple trials, phase 2 studies, interrupted phase 3 trials, but this really, I think, conclusively demonstrates it. I understand that some of this presentation may be open to the public, and they may wonder, what is Alzheimer's disease and dementia? So I try to start with the basics. Dementia is anything that causes impairment in memory and thinking, and Alzheimer's disease is one kind of dementia. It is the most common one because we're all living so much longer these days, but there are many other things that contribute to cognitive impairment and dementia. We need to keep that in mind. What is the challenge? It's, as I said, the most common form, but I had to add this in last week. Before I wrote no cure, and no disease-modifying treatments, but I changed it. But now, disease-modifying treatments currently for Alzheimer's disease, which was previously universally fatal, we may be on the way to actually delaying people dying from Alzheimer's so they can live their lives and hopefully pass away from something else that's not as terrible as Alzheimer's disease. The personal and societal impact is enormous. 30 million people around the world, 6th leading cause of death. In the U.S., it costs us a little over $200 billion, or a lot over $200 billion to our medical systems, and that strains everything, including medical care for other diseases, and it's only getting much, much worse. And so we've got to reverse this trend. We've got to do something about it. This is the organ that it affects, the brain, and you can see on the left half a brain that's ravaged by Alzheimer's disease. It's shrunk and compared to the right half, the other hemisphere from someone else. And this happens to be a mutation carrier that I'll talk about. And when you look at the brain under a microscope, you see the characteristic plaques and tangles. And so this is a neuritic plaque, and much of the biomarkers we were talking about, that Henrik and others were talking about, generate from this plaque. And here is a flame-shaped tau tangle right in a neuron sitting in there. And there may be ways for us to measure that. But these are the two pathologies of Alzheimer's disease that we need to track. Now, not as many people are familiar with the fact that Alzheimer's used to not be a disease of older people. That was called senility, and if you had an older onset senility, it by definition was not Alzheimer's. You could only get Alzheimer's if it was early onset, less than 65 or 60. Alzheimer's first patients were in their 50s. Auguste Dieter may have had a mutation that led to her Alzheimer's and maybe someone else in her family. And these mutations cause Alzheimer's disease, and they've been incredibly instructive for us as a field. So I'm going to focus on this most informative patient population that tells us about Alzheimer's, and I'm going to use that as a case example about how we might accelerate clinical trials for Alzheimer's and other diseases. Some people might ask, well, is this really the same or similar? It's not the same. It's a different form of Alzheimer's. It's caused by a mutation. The age of onset typically is much younger, in the 30s, 40s, and 50s. There can be some nuanced difference. For example, they have more amyloid plaques, more tangles than sporadic Alzheimer's disease. Sporadic Alzheimer's disease oftentimes has other co-pathologies. But the clinical presentation, the cognitive symptoms, the pathology of plaques and tangles, the hypometabolism, the areas of atrophy, our biochemical biomarkers all tell us it's the same disease, it's a form of the disease. I'm going to take 2 important minutes to share with you the perspective of a patient and a family, because this, I believe, is the foundation upon which all research should rest. And I just want to share this. I hope the sound works through the audio booth. Let's try. Here we go.
My father has the genetic mutation that gave me a 50-50 shot at getting the gene as well. My sister got tested, she's negative. I lost the roll of the dice on that, and I found out a few months after she did that I had the genetic mutation. I came to St. Louis, Missouri to be a part of the Diane study at Washington University. Every year I get the same set of tests, which they can look back at my baseline visit and compare information as far as changes. Am I showing more symptoms? Am I not? All the test results, all that's kept blind. Once a month I have a visit from a home nurse that comes from Seattle to my home in Manson. And I get an administration of the drug, and then they monitor my vital signs to make sure there's no allergic reactions or anything else. For me, coming out of having no control over this disease at all, being involved in the drug study, no matter what they needed, was my only form of control. I've hugged almost all the people who are involved in this study, and it's care and concern from them that makes me realize that we're all part of the same team. It will change your life being involved in this. I'm not in medicine, I can't find the disease, but I have a body full of information that I can help somebody else find a cure. I have a 5-year-old daughter, and now she's at risk for the disease as well. I would love for her to know that this was a problem in the past. It has to be beat.
And Brian really captures the sentiments of these families that are cursed with this mutation that's inherited down through the generations, that it has to be beat. And I think we can beat it. The technology, the capabilities, what we can do is growing. And I think that we have a chance to actually stop the disease, in at least some people. So this is a map of the network. This is the dominantly inherited Alzheimer's network. It's comprised of 2 major studies. One is an observational study where we follow people over time, and the other we try to intervene to treat or prevent the disease from progressing in those individuals. I'm going to show you some of the biology behind this, and then show you some of the treatment attempts that we're taking and how we're trying to accelerate treatment development as an example. Shown in this video, you'll see on the left-hand side, this is PIB-PET and amyloid PET scan, and also on the top, these are years, 18, 17, 16 years before the amyloid plaques are really beginning to accumulate. And you'll see the plaques continue to accumulate until they reach symptom onset right at 0 years. Now, the FDG PET, 7 years before, is decreasing, and the atrophy is setting in 5 years before. Now at symptom onset, the plaques are fully established. The brain continues to have decreased metabolism, continues to shrink beyond that point. And what we now know is that there are phases or stages to the disease. And this disease is a 30-plus year disease. It actually starts with 2/3 of the disease progressing before the first symptoms are noticed by the patient or the families. And the last third of the disease, the last 10 years, being the clinical progression through dementia and ultimately leading to death. In that symptomatic phase, that's where the tau tangles are really predominant, and they are growing substantially right at symptom onset all the way through till the end stage of the disease. The plaques, as I showed, really begin about 15 to 20 years before the first symptom onset. That's when they're growing. And before that, we call that pre-pathology. We don't have plaques or tangles, but there's still an opportunity to intervene because we know that there can be some subtle biochemical changes that can be detected. We call that primary prevention. Primary being before pathology, secondary prevention with pathology, and then symptomatic trials. Almost all trials to date, including the successful one that just came out, have been run in this symptomatic phase. And as others have noted, we have prevention trials that are underway. So Dr. Cummings showed this, and I'll show you some of the prevention trials in these trials and why we're doing prevention, why we think we need to do that. But as the last session clearly emphasized, one of the solutions to accelerating clinical trials is to have biological measures. And in order to implement advanced clinical trials, to speed it up for both screening and readout of what the drugs are doing, we need better biomarkers. One of our greatest challenges is that PET and CSF is not easily accessible around the world. In the US, in Sweden, in some parts of Europe, it is. In many parts of the world, it is not. We're running a global study here. So we need to have things like blood-based biomarkers, and these will transform our trials in this, but especially for sporadic Alzheimer's disease, where we need better representation of the entire population we are aiming to treat. Some people ask, well, why bother testing for Alzheimer's disease? If you screen someone and they turn out they're positive, you have to tell them that. Why would we tell them they have Alzheimer's disease? What other disease do we not tell them what the diagnosis is? Is it multiple sclerosis or Parkinson's? Why would we tell them? There are clear reasons why we tell people. It turns out there is a mountain of data, studies, and evidence demonstrating that informing someone of their accurate diagnosis leads to lower anxiety, better management, better care. It's what patients want, it's what families want, and it's what's good for the medical system. So I have to say I get irritated when people ask this question. I'm like, why is Alzheimer's disease so different than any other disease? And in the history of diagnostic development, this was very nicely reviewed. We are now in an era where there are many blood-based biomarkers, and they are accurate, and they can be used. Henrik showed some of the data on the plasma amyloid beta 42 to 40. Here's a recent publication across 3 different cohorts showing the same cutoffs can be used in different continents and different clinical studies. The accuracy is pretty good. It's about 0.85 or so, and if you add ApoE to it, you can get up to 0.9. That actually is as good as most clinical tests in use today, but it's not as good as our best. Our best are CSF and PET. It can predict changes in amyloid before PET becomes positive, several years before. And in trials, we are already using these biomarkers to slash the time it takes to find participants who are eligible for the study. The cost in the trial drops by millions. That certainly is important. But what's more important is the time that we save in starting and finishing these trials. If we can finish a trial a year or a year and a half faster, that is an enormous benefit for the field and our therapeutic development. And it also, by the way, saves tens of millions for the trial. So it's really important. I show in these graphs, you see on the left 2 the time and the cost. On the left 2 are not using blood-based biomarkers as the primary screen, but on the right, it cuts that in half. And you can literally take off a year, year and a half off a trial recruitment by using blood-based biomarkers. These are being used now in prevention trials, and symptomatic trials have now implemented them as part of the screening effort. So these are being used today in clinical trials. They're also being used to diagnose in the clinics, but to a much lesser extent, I think, because we don't yet have treatments that depend on them. I think that's likely to change soon. How good can blood tests be? Here's a recent report. This is from Oscar Hansen's group. He compared different p-tau assays in his cohort, and he found that using the best p-tau assays, you can actually get as good as CSF compared to PET. That's Henrik's presentation showed CSF was about 0.93, 0.95. Here it is, AUC 0.95 for a blood test for amyloid plaques. So I think we're there. This, by the way, is coming out in more publications, has been replicated in multiple studies. I think we're going to be there. We're within a stone's throw now. This question of implementation and getting it out there. There are, by the way, 2 major companies that have this running. and are offering this at different levels using mass spectrometry. It can be done in the hundreds of thousands to millions of tests per year. So I don't think the technology is the limitation. It's the implementation. But we would have that same issue with PET or CSF. And so I think this is going to be a matter that will happen. Now I want to get back to the biology and how else can we accelerate clinical trials. Remember the map that I showed you about the changes in the mutation carriers? If we map each of these biochemical changes, starting with the amyloid 42 to 40, and within a few years, the p-taus are changing, p-tau 181, p-tau 217, p-tau 231. Then other p-taus, p-tau 205, NFL begins to change, total tau changes. That's years later. And then years after that, the tangles really begin to grow during the symptomatic phase. You can use this panel of biomarkers now to triangulate exactly where someone is at in their stage of disease. In the mutation carriers, we take advantage of the fact we can predict when they're going to get sick. If Brian's dad got sick when he was 45, Brian can expect the symptoms to come on at 45, plus or minus 3 to 5 years. We soon, I predict, will be able to do this for most people. We will be able to run a panel of biomarkers and predict how close one is to symptom onset and use that to enroll people in prevention trials and use that information to assign different treatments to. So disease-modifying therapies and preventions have had a storied history. I think there's been a lot of controversy over the years. There's been the amyloid wars and the Taoist and the Baptist and the journalist, and everybody gets involved and everybody has a strong opinion. The opinions get stronger the less data or information they have, which is the opposite of science, right? A scientist's opinion gets stronger when her or his data gets more and more. And so the data was there. We've had the data for a long time, and it's growing, and it's completely consistent. So I think, as I said, I really think we are on our way. You can see through the years the efforts that were made, the disappointments, but people really stuck with the program. They kept going, and now I consider that we have really our first successes. And so we're showing clinical benefit, but there's so much more to do, right? We want to slow down the disease even more. We want to stop it. We want to prevent it. And so this is the current landscape of where things are at. We shouldn't forget about prevention. There's a reason I had you all stand up and stretch a little bit. By the way, it's not aerobic exercise. That you need to do 3 times a week, 30 minutes at a time. Aerobic exercise, strong evidence for. So we need exercise. We need good diet. We need cognitive engagement, education. We need all these things to prevent the risk of coming on. But with all that, the people who live the best lives are still at risk and will still get Alzheimer's. So we have to have more than that. We have to be able to intervene. These are all the different therapeutic targets. Look how many and different kinds are in development. So the portfolio of drugs in development is diversifying. These challenges have been reviewed by other speakers, so I won't dwell on them. But ultimately, the only way to answer these questions is doing well-designed clinical trials, having those answers, and using those answers to inform the next clinical trial. And this is what we do in the clinical trial space. I just want to mention that, that recent findings of these slowed cognitive and clinical declines with amyloid plaque removals is one of these clues. Before, when we trivially removed the amyloid plaques by 5%, it didn't do anything. There was no difference. When we lowered down soluble A beta production by 80 or 90% in symptomatic AD, no benefit at all. But when you remove nearly all of the detectable amyloid plaques by PET scan, you consistently have a clinical cognitive benefit. That is a message for us. We have to remove this pathology to nearly normal levels by PET to see this effect. As I asked the question, there's a mismatch. We remove 80 or 90%. Why don't we 80 or 90% of the way stop the disease? Because there are other processes going on probably. And the question then becomes, can we get more mileage out of targeting amyloid? Do we need to target other things like tau and inflammation? And so we need to do this. In order to do this, everybody has to come together. This is my family. We're on a Alzheimer's Association walk to raise some funds to help with research and care of Alzheimer's patients. But it takes a larger family. And so that larger family is all the stakeholders that are trying to stop the disease. These are patient advocacy groups that have contributed to these trials, pharmaceutical companies that contribute expertise, time, their best drugs, money, governmental funds from the National Institutes of Health and others to support these kinds of research efforts, and then in-kind support from other stakeholders that are trying to help out. And in all these stakeholders coming together, we formed what was called the Gba1 Trials Unit, a group that brought everybody together to try to design these trials. And I'll show you that our first attempts were squarely focused on amyloid. We were targeting amyloid plaques, we're targeting soluble amyloid, and targeted the production of amyloid. We launched the trial with actually 3 drugs. The beta secretase inhibitor was stopped early due to safety reasons. We continued the trial with the 2 other drugs. One was an antibody to soluble amyloid beta. The other was an antibody to plaques. We enrolled these individuals from the families, and we randomized them to receive one drug or the other drug or placebo. And that was in 2012 we launched that trial. We read out the trial in 2019. That's 7 years. A 5-year prevention trial, 4 to 5 year prevention trial takes 7 years. Why? It takes several years to find and enroll people. So think about that. We could have got, had we had better tests, had things set up, we could potentially move faster. Here are the bottom line results of that trial. If you take a look at the top, that's the same individual. That person has no symptoms of Alzheimer's disease. They have a head full of amyloid plaques. At the end of the study, the plaques are gone. At the bottom is a symptomatic individual. That person has a head full of amyloid plaques. At the end of the study, substantial plaque removal, but not to normal levels. And what we found was that all these biochemical directions showed that when we removed the plaques with these antibodies, we had the 42:40 ratio improve, we had the total tau levels improve, we had p-tau 181 improve, and we had NFL improve by a small margin. So 40%, 30%, and only 10% on the NFL. But we did not see a clinical difference. These people still clinically, those who were advanced symptomatic, mild to moderate Alzheimer's disease continued to advance. And those that were asymptomatic all did great. They stayed asymptomatic. So we don't know yet what this will do in the asymptomatic phase. So we extended this trial and we're running it out to see if we remove amyloid plaques in asymptomatic people, do they ever progress? Do they ever have the onset of dementia? And that's ongoing right now. This is one of my favorite gifts I received from Amsterdam. I wear them around the house. Thank you, Philip. And I love it because it has this fight Alzheimer's on one sock and it has POW on the other side. So it reminds me that we should be bold and proactive in our fight against this terrible disease. And in order to do that, we have to pull out all the stops, bring out all the guns. And in addition to symptomatic and secondary prevention and primary prevention, going much earlier, trying to stop the disease from coming on, we have to think about what can we do to really impact the biology of the disease. And so we're looking at tau treatments now. Genetic treatments like ASOs and siRNAs, small molecule aggregation inhibitors, and other tau monoclonal antibodies, and using these in combination together to have the biggest impact for the patient. So I just want to mention that there is a biomarker that correlates very highly with clinical and cognitive progression in Alzheimer's disease, and it's tau PET. As we measure tangles through tau PET, it remarkably has remarkable associations with that phase of the disease. So I think it's a worthy target to go after. And as I said, combination therapy. So we've just launched the first combination trial in Alzheimer's targeting tau and amyloid. And in this case, people will receive both drugs at the same time. In one case, they'll receive the amyloid first, then they'll receive the tau, and the other group will receive tau first and then the amyloid. And the goal there is to try to hit both targets at the same time for a bigger effect. And we're going earlier, much earlier. Dr. Eric McDade is leading the primary prevention in the Gba1 2, and the goal there is to go 25 years before symptom onset. Some of these people will be as young as 18 years old. and they're going to be on an intervention that's going to try to prevent them from developing amyloid plaques in the first place. And we call this the ultimate trial because we believe that if anything will for sure cure the disease, it's got to be this. If we can prevent these people from getting amyloid plaques, we don't see how it's possible for these people to develop disease, but we'll follow them over time. You remember that cascade of events. We'll follow those year by year to see, do we interrupt the entire process? Do we stop the other things from coming on? And so this is the timeline at the top. You see 2012 to 2028. We're squarely in the middle here. We have ongoing open label extensions and combination trials. We're preparing to launch our next tau drug arms, and primary prevention is now launching. Next week is our startup meeting here in Europe in Barcelona, so we're super excited by this. But I want to end by just pointing out that this is possible. Not only have we had an impact in the symptomatic phase of the disease, in other diseases it is possible to delay the onset not by months or years, but by decades. My Italian half of the family has a rare form of familial hypercholesterolemia. My uncle died when he was 30. My granddad died when he was 60 of major heart attacks. And today, my other uncle is alive at 70. He just finished an African safari and has had no cardiovascular symptoms. He has the same, by the way, the same hypercholesterolemia. But the discovery and the development of treatments to lower cholesterol, in this case the HMG-CoA reductase inhibitors, has added decades of prevention for people at the highest risk, like these mutation carriers, but also for the rest of us, preventing cardiovascular and stroke. And so this, I think, is the generalization. This is the end game to which we can deliver the promise of how to stop Alzheimer's if we can prevent the pathology of Alzheimer's from progressing. So I made it in the nick of time. In my last few seconds, I just want to give some thanks to all the research teams, my lab, the DIAN, Dominantly Inherited Network, and the trials unit, and the many centers that make all this possible, as well as the families and the patients. And I'll leave this up here for people who are interested in information. Thank you. Is there, I know you all want lunch. Is there any burning questions? Did I have a misstatement, or do you want to clarify something? Or do we all want to eat? Yes. George Freidenberg.
Earlier characterized by someone else that the lecanemab trial was a marginal improvement. You thought it was a dramatic improvement. Who's right?
Well, okay, so George, George Freidenberg, Us Against Alzheimer's Disease, asked the question. I characterized the lecanemab trial as a significant improvement. And there have been press releases and publications and things like that where people question the value of it. But I describe it this way. In the CDR sum of boxes, when I talk to patients, well, your CDR is 0.5 or it's 1, I tell people, I describe what the box is. You can independently go shopping, balance your checkbook, do things like that. Or you can't do it independently. You have to have someone with you to help you do those activities. That's the difference between a 0.5 and a 1. Most of my patients and most of my families have told me they find that very clinically meaningful. So from the patient-family perspective, I think it is clinically meaningful to do that. Furthermore, the evidence suggests that it's separating greater over time, so greater gains could be gained in the future. And I think if that 0.5 then turns into 1, 1.5, and 2, we're talking about a substantial effect that, again, matches and rivals that of all of our other diseases that we're treating. Diabetes, heart, everything else that we treat is in that range. So I think it's a huge win. Does anyone want to, let me do real quick, show of hands. Who thinks slowing down Alzheimer's disease by 27%, gaining that 0.5 box score over 18 months, if you yourself had it or one of your loved ones, would you want to take that treatment or not? Just a quick show of hands. Okay. Who, hands down, who, almost everybody raised their hand, but who wouldn't? Who wouldn't want that? Oh, come on, there's got to be one person in here that doesn't want it. The side effects, that's right. And there are side effects. And so the ARIA, right? And there's or the expense or the trouble, you got to get injections and you got infusions, and it's not a fair question because all of that's inclusive. But it turns out the people who actually have symptoms of side effects is a few percent. And so, but it's an unfair question because you don't have it, or your family, well, maybe your family member does, but you have to be sitting in an office with a doctor who explains it all to you. And then you have to think about it, and you have to say, okay, infusions every 2 weeks. But my only point wasn't for everybody to make a decision and finalize it. My only point was that many of us think that that's important. And it is a substantial, this is a milestone time in our field. And I just want us to recognize that. Because, you know, I think there's balanced reporting out there, but I think there's less than the exuberant enthusiasm that actually I think we should have at this point. This is good. All right.
Thank you very much.
which is really crossing the boundaries among different neurodegenerative diseases, something really novel and something that is very important to do. The first talk is entitled Early Phase Trials in Alzheimer's Disease, Dawn of a New Age by Kath Mummery. Kath is a consultant neurologist and honorary associate professor at Queen Square. She's head of clinical trials at the Dementia Research Center at UCL and has particular interest in early phase studies and genetic therapies and done some very groundbreaking investigations in her career. So, Kath.
Thank you. Thank you for the lovely introduction. I'm sure we're going to have people catching up with us because lunch has been very short. So I'll get started, though, because we've got a lot to get through. So I'm going to talk about early phase trials. First thing I want to say, though, is thank you so much for inviting me. This has been a tremendous day and a half so far. I've learned a huge amount. And having the basic science here and the epidemiology, as well as clinical trials, across all those diseases is really quite extraordinary, and I hope we do a lot more of this. I'm an optimist and a clinician first and foremost, and like Randy, I do think that we're at the beginning of a new age. There is a question mark on my slide, which maybe is because I'm slightly more cautious, but I don't think so. I think it's just that I chickened out. So what I want to do first of all is just briefly go back to where we came from and then give you a few examples of some of the early phase clinical trials that we've been doing that really are changing the boundaries in what we're able to do in Alzheimer's in particular. And I'll go back first of all, way back when Sir John was a whippersnapper. John Hardy and Martin Rosser were hunting for the gene that caused familial Alzheimer's disease. And one of the first advances in Alzheimer's disease that really changed what we thought about it was caused directly by a young woman called Carol Jennings. And she'd noticed in her family that there were too many people dying young and seemed to be dying of dementia. And she realized this was more than coincidence and chose to write to John and Martin about her family. That decision that she took and that bravery she showed to write to them in the late 80s and say, there's something going on in my family, and then to inveigle all of her relatives to have blood tests, directly led to that first genetic discovery. So I think she's an extraordinary woman and advocate for Alzheimer's. And this is her with my friend and colleague, Nick. And Carol, throughout her life, has been a powerful advocate for Alzheimer's disease research and looking for treatments. So it was a real privilege and fitting that we could offer her a place in the first ever treatment trial in those at risk of Alzheimer's disease. She was my first patient into that first treatment trial, which Randy described earlier. That was the DIAN-TU first arm, and she was the first patient in the UK that went into it. Her family have continued to be involved in research in general and in the DIAN-TU network. In fact, her son has just gone into the latest arm of DIAN-TU. So this is why I do trials. This family are an incredible privilege, and other families like them, to work with. And to be able to go from genetic discovery to finding a potential treatment in Carol's lifetime is really quite extraordinary. So we've heard quite a lot from Randy and others about the changes in the past couple of weeks from the point of view of lecanemab and the data. I'm with him. I think this is significant, and I think it's hugely exciting, even if it is a modest benefit so far, if the data and the details bear that out. But before this, we had a huge number of phase 3 trials that were on the back of early phase studies. And the question is, well, why were they then negative if these early phase studies were done in the right way? And I think we need to think about that design, which has come up before from people like Jeff, how we make sure that those later phase studies are given the best chance of being positive. We've got a lot to learn from MS, where they were describing how very few phase 3 trials are negative after positive phase 2s. So I don't need to talk to this audience about targets in AD. We had an incredible discussion yesterday about a large number of potential candidates, and there will be a number of those that are on the verge of going into phase 1 studies and others which are being explored. But just briefly, we've looked for decades now at ways to try and treat amyloid, and until recently that was negative, but we now have the first evidence that we can change the course of the disease, however modestly. I think that that's massively significant. We are now looking at different ways, not just immunotherapy, which was dominant for many, many years. So in early phase trials now, we currently have an siRNA against APP, trying to knock that down in phase 1 trials in young onset Alzheimer's disease. The secondary pathology in Alzheimer's disease of tau is obviously the next target that people have been focusing on in more recent years, and I'll focus more on this later in the talk, but I just want to mention the immune pathway and microglial response because there's been so much talk about this over the past day and a half. It's fantastically exciting, the different methods and some of the things that I've learned about over the past part of the conference. Michael Schwartz described the peripheral modulation of the immune pathway, enhancing the response within the brain. And that's a study that is literally starting in the next few weeks in Alzheimer's disease, looking to see what effect that has and how safe it is, which is very exciting. And there's been a phase 1 going into phase 2 trial looking at TREM modulation with a monoclonal antibody for the past year and a half, which is also ongoing. But as I said, I'm going to focus mainly on therapies that have been targeting tau. And as we know, tau has a critical role in mediation of amyloid toxicity. Matt Brandy said earlier, when you get symptoms, you have a head full of amyloid. But the tau pathology correlates directly, both with severity and topographical distribution, with cognitive impairment. And in addition, we can see prion-like seeding and spreading that propagates tau pathology, and we know from silk work, again, that Randy has done and others, that tau production correlates with the presence of amyloid. And so if we can suppress Alzheimer's and tau in Alzheimer's, we can potentially prevent and reverse that tau spreading and aggregation and prevent that amyloid-induced toxicity. And critically, it's a different therapeutic window to the one that amyloid gives us, where I think, as John has mentioned before, we really need to be going earlier, John Hardy, than we are at the moment. There have been multiple strategies targeting tau over the years, and again, as with amyloid, immunotherapy has been the dominant approach. Both active and passive, primarily passive. There are other approaches. The other one I want to focus on is tau reduction using gene silencing against MAPTRX. What you look at in this pie chart, not a pie chart, but if you look at this diagram, you'll see that there are very, very few, only one drug that's got to phase 3. There are far more in phase 2 than there were when I last drew this slide, which was 2 years ago, but a number of those have fallen off, and they are primarily immunotherapies. But there are more in phase 2 than there were then, which I think is encouraging. So as I said, I'll just talk about 2 immunotherapy drugs and 1 tau reduction drug. One thing to say, actually, is if I was giving this talk a few years ago, it would not be me selecting the number of treatments that I'm doing just now. It would be talking about everything, and it wouldn't take 20 minutes. So things have changed a lot. Active immunization. Talking to an audience who will know more about this than me, but the legacy of AN1792 was talked about yesterday with the nervousness that it gave the community in trials, at least, because of the development of meningoencephalitis in phase 2, the fact that phase 2 study was stopped. For a long time, certainly in amyloid, people did not want to repeat that experience, but it is now being looked at against tau. Again, the preclinical work in transgenic mice showed peripheral immunization against tau, reduced brain accumulation of tau, and showed cognitive and behavioral change associated with that reduction. And so clinical work, there are 2 active vaccines currently being explored. One is ADVAC1, which is slightly more advanced than the other, which is AC Immune. Preclinically, this induces antibodies targeting path tau. It reduces aggregation of tau, and the phase 2 was read out in 2021 by Novak et al. It showed the drug was safe in mild Alzheimer's disease. It showed that it developed a robust antibody response that was sustained. And from the biological point of view, there was a reduction in NFL. There was some reduction in CSF tau, but only in one moiety rather than all of them. There was no change in cognitive or functional outcomes. And one of the reasons I show you this is because I think it's really disappointing how quickly or slowly we've learnt across the trials, for example, in terms of who goes into them. So in this trial, post hoc, they looked at who went in, and only 2/3 of the patients had AD pathology criteria. And this is a very recent trial. We should have learnt this years ago, that we need to be ensuring they're path-specific. Post hoc, they suggested that if they were path-specific, they showed a clinical positivity, but clearly we need to see phase 3 data to see whether that's real or not. Turning from active to passive immunotherapy, so clearly these drugs bind to extracellular tau, mop up the extracellular tau, and there have been a number of approaches trying to do this. There's uncertainty over the path species to go for, so there's been a wide range of strategies from epitope-specific to promiscuous pan-tau antibodies. There have been a number of negative trials, some of the ones you saw on that previous diagram, in PSP, but also in AD. And so perhaps people were getting a little disheartened around immunotherapy in AD against tau. My colleague, Rohan De Silva, in partnership with Eisai, has developed a new anti-tau antibody, E2814, and this targets a path-specific seeding-competent part of tau, and we hope, or they hope, that that will make it more active. The preclinical work has been very encouraging, and Eisai did a first-in-human healthy volunteer study, which was phase 1, and that showed a significant reduction in the CSF tau levels by up to 60%, that was sustained. And we started an open-label study, phase 1b, in moderate autosomal dominant Alzheimer's disease a year ago. This is an 18-month multiple ascending dose study. First patient we got in, this is being led by UCL, this is September 2021. There's 7 in the cohort. This is a tiny study across 3 sites. They get the antibody infused monthly. The doses are ascending in each individual over time, and we're using CSF to target, to determine target engagement. So far, it's been running for a year. There's been no worrying safety signal. The study is ongoing. And what's important is that study's in moderate autosomal dominant Alzheimer's disease. And if we come back to the DIAN-TU trial, that next-gen anti-tau arm that we've discussed, Randy mentioned, I mentioned earlier, these results will inform that phase 2/3 much larger arm in terms of firstly, target engagement, and secondly, safety. And we know that there may be differences in this population. So having the same population with this drug used in is really important to optimize the dose requirements in this precious study. In addition, and it's no less important, moderate autosomal dominant Alzheimer's disease patients don't have a chance to go into any studies normally. They're not eligible. And these families are desperate to do something. You heard on the video early that Brian was talking about taking back some control. That's how he felt when he was in trials, and that's what all of our trial participants say. And this enables people to feel that they are involved, they're taking some form of control. I think that's really important in what we do. So turning from immunotherapies to genetic therapies. There's been an explosion of therapies in certain areas recently. This is about spinal muscular atrophy, which was mentioned yesterday. And for diseases like this one, it really is a revolution in treatments. 3 licensed therapies in gene therapy is extraordinary. And just to revisit a graph, which I'm sure many of you have seen before, this shows the patients across multiple trials. So this here is, can you see that? Is that working? Marvellous. The 10 minutes rehearsal beforehand worked for all of those that are giving talks now. So this is the placebo group. Effectively, these infantile SMA children die by the age of 2. If you give them this drug, consistently they improve their motor milestones. Importantly, if you give them this drug pre-symptomatically, and they've been prenatally diagnosed, you give it at birth, their motor milestones normalize. So you've gone from children dying at 2 to being able to run around with their siblings with relatively normal motor function years later. And that is extraordinary. Now, if we can do anything like that in Alzheimer's disease, that would be unbelievable. But clearly, that's something that is a hope at the moment. So there are expanding number of CNS gene targets now for these intrathecally delivered ASOs. I've talked about SMA just now. Huntington's disease, I think, was mentioned yesterday, so that got to phase 3 with tominersen. Currently, that's paused, waiting further analysis, so there's no positive result there yet. But you did hear earlier about tofersen in SOD1 ALS, and that had initially shown biomarker benefit after 6 months, and after 12 months, it's shown clinical benefit. So that is really encouraging. And that will be taken forwards. There are some very early phase studies in some forms of genetic FTD, genetic Parkinson's disease, the SCARS and prion disease. But I want to focus on the MAPT ASO in Alzheimer's disease, which I've been involved with since the beginning. So we've talked about immunotherapy and mopping up proteins, either actively or passively. So going upstream and trying to prevent production of the protein in the first place by gene silencing is clearly another way to go. This gene, if this is the MAPT gene that produces tau, the Ionis MAPT-Rx, now known as BIB080, is a synthetic oligonucleotide that binds to the messenger RNA, leading to its destruction and therefore preventing production and reducing the amounts of tau. If we're uncertain which species of tau are pathological, then suppressing all forms in all compartments is a pragmatic solution. Preclinical models suggest that reduction by 50% is sufficient, and so that's what was aimed for in the study. The preclinical work prior to this was very elegant work done by Sarah de Vos in Tim Miller's lab, and effectively they showed in transgenic mice that accumulated tau that if you gave the ASO versus the scrambled ASO, you reduced accumulated tau. Not only that, and I haven't got it on here, I apologize, but you also reversed the pre-existing deposited tau. And associated with that, there was an increased survival rate, and there was normalization of nesting behavior in the mice treated with the active ASO versus the scrambled. And so on the back of that, in 2017, phase 1 first-in-human placebo-controlled trial was started with this drug. And this feels a very long time ago now, actually. I mean, it's only 5 years ago, but the idea of giving a genetic therapy to Alzheimer's disease at that point did sound slightly bonkers. And we dosed the first patient in October 2017, completed the trial in 2020. There were 46 individuals that went into the study. It was a multiple ascending dose cohort study, so there were 4 cohorts, A, B, C, and D. The first 3 got intrathecal doses monthly. The last cohort got doses 3 monthly due to the information gained from the first 3 cohorts, and they all had 6-month follow-up. They then went into an open-label extension. A lot of people worry about the invasiveness of a monthly intrathecal injection. Every single one of those patients, certainly in my cohort, wanted to go into the open-label extension and continued in it until it was closed. So for them, there wasn't an issue with having this process done. Primary outcome was safety, and secondary outcome was CSF levels of tau and p-tau. PET wasn't available when we started this study. So the results. Safety results, no SAEs in the active group. There were 2 in placebo. And in terms of the AEs, the majority of those were headache, post-lumbar puncture syndrome, procedural pain. There was very little in the way of worrying signal from the point of view of anything related to drug. No patient was discontinued as a result of an AE. And in terms of target engagement, let me just orientate you to this graph. So the graph on the left is change from baseline in CSF total tau. Graph on the right is change from baseline CSF phospho-tau. And the lines are black is placebo, and then the doses as they increase are yellow, orange, blue, and purple. And just to be clear, the blue is the 3-monthly dose. So they actually had a higher dose, but the overall dose was lower. It was midway between orange and purple. The arrowheads here are the doses that they were given, and this is the follow-up period. So what we found was a dose and time-related reduction in CSF tau, both t-tau and p-tau, and that was sustained through the follow-up period, although they'd stopped dosing. So it's the first anti-tau drug we felt to show reduction in tau in sufficient levels to potentially slow cognitive decline. It was safe, and so an 18-month phase 2 trial has just started, which, watch this space. So, in conclusion, what are the next steps? Well, I think that the proof of concept, mechanistic, short, small phase 1 trials are absolutely critical. We have to learn about our pathology, positive patients. I can't believe we're still putting people in because they look like Alzheimer's disease, not because they've got the pathology. And we need these to translate from the fantastic basic science that we've been hearing about into what's meaningful for patients and into the later phase trials. Critically, we need to ensure that we've got target engagement, and we need to ensure that we've got doses right, as somebody else mentioned, not worry about whether we got the dose right or not after the trial has failed. We have got an expanding portfolio of treatment targets, which is important because if we have an amyloid treatment but it's only working modestly and early, we need to be able to treat at different stages, and we need to be able to consider combination therapies, which may well be an anti-tau therapy in combination with an anti-amyloid, or it may be an anti-inflammatory, or it may be 2 anti-amyloids. We don't know yet. There's an awful lot of work to do. There are multiple other targets being explored that I haven't had time to talk about. Combination therapies are not something that we've really had to think about until now. It's been hard enough treating with one at a time, although there have been a few cases in the past where people were looking at BACE inhibitors along with an amyloid immunotherapy. We really need to map out the interactions if we're going to start using an anti-amyloid therapy, which we may well be doing next year, alongside a different therapy. We don't have those interactions or the biological effects of having two drugs ready. We also need to have a really close reciprocal relationship with basic science. So when a study, and this was mentioned by Prof. Larson yesterday, when a study is negative, it's equally important that we go back and explore why that was. And I don't think we're given the data to be able to do that. I think we need to use that immense accumulation of data rather than just say, well, the study didn't work. And I'm sure we would all be keen to do that. It's getting pharma perhaps to play the game with us. And finally, something we really don't do very well is post-mortem in patients that are treated in trials. I know Randy has set up so that we try and do that in the DINTU trial, and there are other trials that we're now starting that with, but it's critical that we look at these people's brains who have been on a drug and understand physically the effects of that and interactions of that with either comorbidities or other things. We don't understand that well enough. Finally, coming back to the patient, because that's where I start and finish, this is a quote from one of my trial participants who was at risk of ADAD and was in the trial for a good 4.5 years before he decided that he needed to go and do his bucket list before anything actually happened to him. So this is what he said: "It's made a real difference to me being involved in the trial. For many years I felt useless and unable to have any effect on a disease that has been rife in my family. I have lost so many, and unfortunately there will be a few more that will suffer the same fate. Early onset Alzheimer's is cruel. Being part of the trial and having such wonderful support has helped me grow stronger. It's enabled me to deal with many skeletons in my closet and leave them behind. The work that you do goes much further and deeper than a cure." And that is why I do trials. And finally, just to say thank you to everybody that I work with in the team, and most important part of the team, the participants and their partners. Thanks for listening.
Thank you. The next talk is entitled Parkinson's Disease by Per Svenningsson. He is a professor and senior consultant in neurology at the Karolinska Institute. His research interests are in Parkinson's disease, in looking at both motor and non-motor aspects, and his studies are in both preclinical models and from patients with the goal to identify novel targets for improved diagnostic and disease-modifying therapies.
Thank you very much. And I would also like to start by thanking the organizers and especially Professor Comi for the invitation to this fantastic meeting and for me speaking on Parkinson's disease pharmacological therapies. So I will initially briefly talk about current treatment of Parkinson's disease and then about drug development in Parkinson's disease. There are two pathological hallmarks in Parkinson's disease. There is the loss of the nigrostriatal dopamine neurons, and there is the presence of the alpha-synuclein-enriched Lewy bodies. So in the clinics, we study the dopamine loss by PET and SPECT examinations, and these are PET images from my patients using that PET. And you can see that there is a loss of the dopamine transporter initially at terminal level in stratum, and at later stages also at the cell body level in substantia nigra pars compacta. Since I'm a Swede, I always include a slide on the pioneering work of Arvid Carlsson, because he founded all the dopamine replacement therapies that we use today. So in his pioneering work, he used rabbits and he gave them reserpine. And reserpine made the rabbits paralyzed, and then he gave them various precursors of the serotonin, tryptophan, but also L-dopa, and he found that L-dopa restored locomotion. Arvid Carlsson also found that L-dopa generates dopamine, and that dopamine is a neurotransmitter by itself. Nowadays, we also have several therapies that reduce the enzymatic degradation of dopamine, and also several different agonists that stimulate especially dopamine D2 receptors to offer our patients. As you know, Arvid Carlsson was awarded the Nobel Prize in Physiology of Medicine in year 2000 for his breakthrough work. I could not find an original photo from that prize ceremony, so I made this collage. And as some of you see, Arvid Carlsson is a little bit bigger than in real life. We have one more sensational therapy in Parkinson's disease that we can offer our patients. That's deep brain stimulation. I will hear more about this from Professor Moro and Professor Bergman. So here you can see an example of a film of a patient that has deep brain stimulation and severe rest tremor. And when it turns off the stimulation electrode, he has terrible tremor that he can then contract with deep brain stimulation. But we'll hear more about that later today. So this is a summary of the current treatments that we have for Parkinson's disease. We have more than 15 different symptomatic therapies, most of them then restoring dopamine transmission, and then we also have DBS. And we combine these different treatments for our patients in combinations that we think are optimal for each patient. However, we need to develop even better therapies, both better symptomatic therapies and disease-modifying therapies. There are more than 90 clinical trials to try to improve symptomatic therapies against both non-motor symptoms and motor symptoms of Parkinson's disease. And I will show you some data that we have on trying to find better treatments for a side effect of levodopa called levodopa-induced dyskinesias. So just to give you some background. So this is a patient that is coming to the clinic in off state. So she has not taken her, and now both films started unfortunately at the same time. But if you look at the film when she comes to the clinic in off state, you see that she's very, very bradykinetic and walk with very, very small steps and very reduced arm swings. Then these patients get her levodopa treatment that is shown on the other film. And you see that she has a therapeutic effect of levodopa, so she walked much, much better. But she also developed this hyperkinetic condition that we call levodopa-induced dyskinesia, and this can be quite bothersome for the patients. So what we need to our challenge is to try to find a therapy that have the therapeutic effect of levodopa, but that contracts these dyskinetic manifestations. So over the years, several groups have used different experimental models and shown that there is a maladaptive plasticity of the serotonergic system in these levodopa-induced dyskinesias. Under normal conditions, when you take levodopa is decarboxylated to dopamine in dopamine neurons and released in a controlled fashion, where the released dopamine is, synaptic dopamine is taken up by the dopamine transporter and also acts on D2 receptors that act as autoreceptors and counteract further release of dopamine. So you have a controlled release of dopamine by L-dopa. But in the Parkinsonian condition, where you have fewer dopaminergic terminals, a substantial amount of the levodopa is taken up by serotonergic terminals and being formed, decarboxylated into what we call false dopamine. And this false dopamine is released in a much more uncontrolled fashion because there is no dopamine transporter D2 receptors on the serotonergic terminals, and this then generates dyskinesias. So we and several other groups have found that if you dampen or reduce the activity of the serotonin neurons, either by reducing the activity of the firing of the neurons with the 5-HT1A agonist, or if you reduce the release of false dopamine from the serotonergic terminals by stimulating the 5-HT1B receptors, you can dampen this uncontrolled release and also contract L-dopa-induced dyskinesias in animal models. So together with colleagues in Lund, we performed then a clinical trial based on this experimental data using a compound called L-toprazine, which is a combined 5-HT1A, 5-HT1B agonist. So we performed a double-blind randomized placebo-controlled dose-finding study of L-toprazine against levodopa-induced dyskinesias. So the patients came to us in the morning and got an L-dopa challenge dose together with placebo or a dose of L-toprazine. And we measured then dyskinesia using the clinical dyskinesia rating scale, but we also measured the anti-Parkinsonian effect of levodopa using UPDRS, which is the motor score that we use in clinical practice. As a secondary objective, we also used another dyskinesia scale called Rush Dyskinesia Scale. And the results are very encouraging. So both the 5 and 7.5 milligram dose of eltoprazine significantly reduced dyskinesias compared to placebo. And that was true both when we used the clinical dyskinesia rating scale and the Rush dyskinesia rating scale. Eltoprazine had no negative effects on the therapeutic action of levodopa and very few side effects. So we hope that eltoprazine or other serotonergic compounds will, in a not too far future, come into clinical use to treat these L-dopa-induced dyskinesias for our patients. But the even bigger and more important breakthrough for PD treatment would be to find a disease-modifying therapy. And as we have heard during several talks in this symposium, the progression of Parkinson's disease is very much driven by the spread and propagation of alpha-synuclein aggregates or Lewy body pathology. Here you can see a slide describing how clinical symptoms correlate with the BRCA staging of how alpha-synuclein in red is spread throughout the brain. Initially, BRCA found alpha-synuclein aggregates in gastrointestinal tract, olfactory bulb, or in the brainstem. Then the alpha-synuclein aggregates propagates and engages the dopamine neurons, and the patients develop their motor symptoms and get their diagnosis. But this propagation of alpha-synuclein then continues and gives rise to a lot of motor and non-motor complications. So large parts of the brain have Lewy bodies, and actually up to 80% of our patients with Parkinson's disease develop dementia. So there are many approaches now being tested in clinical trials interfering with alpha-synuclein. There are small molecules inhibiting alpha-synuclein aggregation and misfolding. There is alpha-synuclein antibody immunization. There are ASOs, but also gene-based therapies to reduce alpha-synuclein levels. And there are small molecules that stimulates autophagy or stimulates lysosomes to degrade alpha-synuclein. Most of these studies are ongoing, and those that have been completed have not shown very positive data. But we still need to wait here to see whether any of these approaches will have a strong effect on the disease progression. So alpha-synuclein plays a central role in the molecular mechanism involved in Parkinson pathogenesis, but there are many other mechanisms also involved. Over the past years, we have studied one molecule that has shown neuroprotection in animal models called CDNF. So it's a neurotrophic factor that protects dopamine neurons, both in the neurotoxic doxydopamine model, but also in alpha-synuclein-based models. It was discovered by Mart Sarma and colleagues in Helsinki, and Mart Sarma has worked on this, understanding the molecular mechanism of this compound for several years. And he has found when he gives it into putamen that it's retrogradely transported to the cell nucleus and that it reduces ER stress and neuroinflammation. More recently, he has found that CDNF binds to the same binding site as GPR78 or BIP, which is one of the critical molecules in the unfolded protein response pathway. And CDNF contracts the action of BIP or GPR78, especially under stressed conditions. So we have conducted a clinical trial with CDNF, a very advanced and invasive trial, I have to admit and say. So we used convection-enhanced delivery to give the full-length protein via 4 catheters into putamen. First part of the study, main study part, was placebo-controlled, so we have a vehicle group. Then in the extension study, we gave all the participants CDNF at different doses, and then we are right now conducting a follow-up study. It's important to mention that we used the participants in this study were patients with advanced Parkinson's disease. As you can see, the average disease duration was more than 10 years. And this is, of course, not ideal when you want to study a neuroprotective compound. But we thought for ethical reasons that we could not conduct this very invasive therapy in patients at earlier disease stage, since it's the first time it's administered to humans, actually. The data, when we looked at our motor scores in UPDRS, shows not a significant, but a trend for an improvement using the mid-dose of CDNF. We also used various devices in this trial, including accelerometers, and when we studied then bradykinesia or hypokinesia with this accelerometer, we found again that the mid-dose of CDNF seems to counteract PD progression. There were a heterogeneity in the response among the participants, but we have some what we think is responders with this therapy, and here's an example. So this is a patient that was at Hernian-Jaw stage 2 with a disease duration of 10 years. He had a very good L-DOPA response, off time approximately 6 hours per day. He initially received placebo and then active treatment with CDNF, first at low and then at mid doses. And as you can see, he improved on UPDRS off, and he actually developed dyskinesia, so we had to also to lower his dopaminergic therapy during the trial. Since there was a heterogeneity in the response among the patients were genotyped after the completion of this study, and it turns out that this patient has the most common LRRK2 mutation, G29S. And we also had actually one patient with a Gba1 mutation enrolled in the study, and we learned that afterwards, and both those patients actually responded to this therapy. So it may be that the CDNF is good for the treatment of a subtype of patients with Parkinson's disease. Martsarma and Herantis have now developed, they have identified the binding region of CDNF to GPR78 and generated brain-penetrating peptides. And they have shown in animals that if you give these peptides systemically, that it enters the brain, and it also protects dopamine neurons and reduces neuroinflammation in an alpha-synuclein-based model. So we think that if we can start to work with this compound, we can give it to many more patients, and we don't need to use this invasive approach, and hopefully also later on at earlier stages. And phase 1 trials are planned to start beginning of next year. During the last final minutes of my talk, I will talk briefly about another mechanism that we also heard a lot about during previous talks in this symposium, namely how you can activate the microglia to have neuroprotective effects. And one such compound that we also heard about, or another target that we heard about, is the GLP-1 receptors. And there is data from Ted Dawson showing that a brain-penetrating GLP-1 agonist that he developed, NLY01, inhibits activated microglia and thereby inhibits the activation of astrocytes into the toxic A1 phenotype. And this compound is now in clinical phase 2 trials in Parkinson patients. There are several other GLP-1 agonists also in clinical trials. against Parkinson's disease at different phases. And especially, there is a lot of interest in a compound called exenatide that is already used in type 2 diabetes. So in England, there is a large phase 3 trial being conducted with exenatide. And the rationale for doing this large phase 3 trial is the data from the phase 2 trial that Foltine and colleagues generated and published in Lancet a few years ago. So they found when they gave exenatide to patients with mild to moderate Parkinson's disease that they could have a significant slowing of the disease progression. One thing that we noticed that the effect is relatively rapid, so there may actually be a symptomatic effect of exenatide, but that's something that we learn now that they're conducting a phase 3 trial, and we will receive the results from that trial. So to summarize, there are more than 15 symptomatic PD therapies, including DBS, that we'll hear more about in clinical practice that we can offer our patients. There are more than 90 novel symptomatic therapies in drug development. In our center, we have shown that deltopracin show promise as an antidyskinetic therapy in PD. There are more than 50 possible disease-modifying therapies in drug development, and we have studied CDNF, and our data indicate, or also taken together with the preclinical data, indicate that it may contract PD progression, at least in a subtype of PD patients. There are results coming from clinical trials using approaches to interfere with alpha-synuclein very soon, so we'll learn more about the success of those approaches. There are also clinical trials towards subtypes of PD patients. We heard Professor Shapira's lecture earlier today on Gba1 and Ambroxol, but there are also specific trials for LRRK2 PD patients. And finally, GLP-1 agonism shows promise in slowing down PD progression, and we will soon know if this holds up. So finally, I'd like to acknowledge collaborators that have participated in this work. So thank you very much for your attention.
Thank you. So now the next speaker is Steve Hauser. Is that...
Good afternoon. I'm Steve Hauser, speaking to you from San Francisco and the Weil Institute for Neurosciences at the University of California, San Francisco. The Weil Institute is an umbrella organization that links together the clinical with the basic neurosciences, with a goal to speed development of much-needed therapies for the 1 billion people each year who are afflicted with a brain disease. My talk this afternoon will be on the advances in pharmacologic management of multiple sclerosis, with a specific focus on the role of B cells and therapies targeted to this class of white blood cell. As you've already heard, multiple sclerosis is a chronic multifocal CNS disorder. Myelin loss is characteristic and is immune-mediated. There is secondary neurodegeneration driving disability. There's a genetic predisposition with more than 230 variants identified that contribute to MS risk. There's a strong influence of environment with infectious agents such as Epstein-Barr virus and Akkermansia bacterium identified as associated with MS risk. The prevalence appears to have increased sharply, and our treatments are increasingly effective. When I was a trainee in the 1970s and a patient came in with suspected MS, we would ask the staff to dress the patient in a bathing suit, and we placed the patient in hot tubs that were located on all neurology services at the time, and if their symptoms worsened and their spinal fluid showed evidence of oligoclonal bands, these increased amounts of antibodies in the spinal fluid not present in peripheral blood, we would say that they had MS. The MRI wasn't even a rumor. We had no treatments except for steroids, and these barely worked. And moreover, there was a therapeutic nihilism in the field and a belief that we could never develop useful therapies for people with MS. Today, therapeutics for MS can be confusing because there are so many excellent choices for our patients. Here is my personal summary of the safety and efficacy of these drugs. Ideally, we want a therapy in the upper right-hand corner of this cartoon, something that is highly effective and very safe. Beginning in the 1990s were the injectable therapies, immunologically nonspecific, very safe, but also not very efficacious. In the early 2000s, a second line of therapy were brought to the marketplace, both oral therapies and the first monoclonal antibody, Natalizumab, which was much more effective. Also, a third line of therapeutics, some of these treatments not approved by any regulatory body, which appeared to be quite effective, but also had a low safety profile. Natalizumab was taken off of the market when cases of the viral brain infection PML, or progressive multifocal leukoencephalopathy, were identified. But fortunately, with the development of a blood test that identified patients at low risk for natalizumab PML complications, the drug could be returned safely to the marketplace for those patients at low risk. Our therapeutic decision-making was guided by what we call treat to target. We would begin with a safe first-line therapy when patients had breakthrough disease. We then advance therapy. More recently, development of B-cell-based therapeutics were brought to the market. Ocrelizumab, ofatumumab, and off-label rituximab. And these drugs did two things. First, they showed us the importance of B-cell therapeutics and also reinterpreted the effects of other effective MS drugs as possibly also working through B lymphocytes. And second, these represented a highly effective and relatively safe form of therapy that could change our treatment paradigm so that these now could become first-line therapeutics. I'm now going to show the pivotal trial data from one of these B-cell therapies, ocrelizumab, to show you the magnitude of the effect. Patients coming into the study had MRI evidence of nearly 2 active inflammatory MS lesions in the brain. This was a double-blind study in which patients were treated with either ocrelizumab, the B-cell therapeutic, or the platform therapy, interferon beta-1a. Interferon beta-1a is an effective MS therapeutic, reducing the inflammation on the MRI scan by about 75%. But look at this. The effect of ocrelizumab was a greater than 99% reduction in brain inflammation. I would argue that this is the strongest effect seen against any therapeutic in modern medical treatment trial history. More than 99% of an effect size. We now have data that go out more than 9 years, and ocrelizumab remains safe and effective over the course of this time, and patients who were initially treated with interferon and then moved to ocrelizumab capture the benefits of this drug over time. A most important finding from the long-term extension studies of B-cell therapies for patients with MS has been its continued safety. The CD20 antigen is present only on B cells in the middle phases of their differentiation cycle. CD20 is absent in the earliest B cell precursors, the stem cells and pro-B cells, and also absent from the terminally differentiated antibody-producing plasma cells. This means that with CD20 therapeutics, B-cell reconstitution is preserved, as is long-term memory. In addition, our therapies target mostly B cells in the peripheral blood and not the majority of B cells that are present in lymph nodes and spleen. This probably together explains the long-term safety of this therapy. Thus far, I've spoken mostly about the relapsing form of MS, MS which is characterized by periodic attacks of inflammation. I've not spoken about the progressive neurodegenerative form of the disease. Most people with relapsing MS evolve to develop progressive disease over time. It was my hope that by shutting down MS inflammation so completely, we would no longer see progression. But this was not to be the case. When we looked at progression in our patients with relapsing MS in the ocrelizumab trials, those patients treated with interferon as expected were worsening about 6% of them per year. Fortunately, this was reduced by about 40% to approximately 4% per year in patients treated with ocrelizumab, but not reduced entirely. And, as you've already heard, when ocrelizumab was tested in primary progressive MS, benefits were also seen, but these were also just partial, approximately a 30% reduction from the 15% of worsening present per year in patients with primary progressive form of multiple sclerosis. So what this told us is that there is an underlying progression, even in patients with early relapsing MS, and that this progressive element of the disease is treatable, but only partially so with our B-cell therapeutics. What is going on? Understanding that progression occurs across the MS continuum is a very important lesson that we've learned from the clinical trials and only could have learned from the clinical trials. It brought us back to the laboratory to ask what is happening in our patients. In fact, in the MS world, we've learned as much from our bedside experiments as we have from our bench laboratory experiments. And it's by bringing these two worlds together that progress has been most effectively made. I think that this is a model of bringing the bench and bedside together that is so important if we're to make discoveries that improve lives of patients not only with MS, but for the 1 billion people each year who suffer from brain disease of all forms. We've learned in MS that progression is, yes, driven by inflammation in the myelin-rich white matter tissue, but there are also other pathologies that we were previously unaware of. Most importantly, we now know that in the meninges, in the tissue lining the outside of the brain, there are B lymphocyte-rich inflammatory follicles that form and persist. We never knew that these were present. In addition, some old MS lesions remain chronically active, percolating, with borders of macrophage-like phagocytic cells called microglia in the brain, and these cause progressive loss of the axon guide wires that nerves use to collaborate and communicate with each other. Furthermore, there is a gradual destruction and thinning of the myelin membranes in the entire brains of people with MS. And finally, there is age-related worsening that we see in all of us, of course. A very important feature is that every time that we see a B-cell rich follicle in the meninges of the brain, there is a degeneration of the nerve cells lying just below, something still largely invisible to our current MRI scans, and this is why it was missed for so long. So we now understand that progression is present throughout the disease course and is mediated by multiple different pathologies. And all of these pathologies, we believe, are improved, but only partially so, by our B-cell therapeutics, just as anti-CD20 therapies are only partially depleting in the lymph node and spleen. This is why they are so safe. They are also only partially effective against these residual cells which are lodged in and just outside of the nervous system. Here you can see what is happening to B cells at different stages of differentiation in the peripheral blood in red and in the spinal fluid in blue. And I just want to show you that most of the B cells in our peripheral blood are naive, early, unstimulated B cells. Whereas in the spinal fluid, most of the B cells are activated, antigen experienced, and ready to produce antibody plasmablasts. So there is a huge difference between peripheral blood and spinal fluid. Angry, activated B cells are moving into the nervous system in our patients. And here I'll make the point just very briefly that the activated antigen-stimulated B cells in the nervous system are making the oligoclonal bands that we detected way back in the early days, and that these cells are also chronic and persistent. They are recirculating between the spinal fluid, the nervous system, and the peripheral blood, and they are, we believe, being activated and continually stimulated in both compartments. When we initially conceptualized the development of B-cell therapeutics for MS, we were targeting what we thought were autoantibodies that were culprits in MS. B cells differentiate, as you've already heard, into plasma cells, which are engines for antibody production. A single plasma cell will make many thousands of antibody molecules per minute. We thought that those antibody molecules are deposited on myelin membrane, perhaps on nerve cells, triggering both complement and other cells to destroy these tissues. But in addition, it was subsequently recognized that antigen presentation may in fact be the true culprit in MS. B cells don't secrete antibody, but they have antibody expressed on its surface. These antibodies can scavenge substances in the environment and then present those back on HLA molecules to T cells. And if the T cells recognize the same substance or the same structure, those T cells are then activated and can carry out their autoimmune function. Beautiful work by Roland Martin's lab indicates that it's not only exogenous substances, but it may be endogenous molecules like the HLA molecule itself that B cells present, and that these molecules are seen by T cells, which are also cross-reactive to substances like Epstein-Barr virus, Akkermansia bacterium, further stimulating those cells, and that a target may be a molecule called RAS-GRP2 or RAS-GREP2, which is ubiquitously expressed. And as you've heard before, it's likely that these meningeal follicles living just outside the surface of the brain are secreting cytokines and other chemicals which may be also neurodestructive. And now returning to the bedside, the clinical as well as the biologic data argue that early institution of high efficacy therapy is the best therapeutic option for many patients with early MS. This is the best way to prevent the accumulation of long-term disability. These data are not only found in the long-term extension studies of B-cell therapies and natalizumab, but also now in at least 5 published experiences of real-world data demonstrating the benefits of early high-efficacy therapy rather than beginning with a less effective treatment and treating to target. So in conclusion, the progress against MS has been truly extraordinary, with major gains in long-term outlook for our patients. B lymphocytes are clearly central actors in the pathogenic cascade. Progression is present in all MS patients across the disease continuum, likely due to chronic CNS inflammation that's trapped behind an intact blood-brain barrier. For most patients, highly effective therapy should be initiated as soon as feasible. And finally, more effective immunotherapies against progression, I'm confident, are on the way. And in closing, I'd like to thank the Fondazione Prada and Professor Comi for honoring me and my institution with a place at this important initiative. It is only through visionary global efforts like this that we are likely to speed the development of therapies, preventions, and cures for sufferers of brain disease. Many thanks.
Maybe I'll do the introduction now for Steve. I counted, Steve and I go back 40 years. We're postdocs together at Harvard Medical School with Howard Weiner. So Steve is the Robert Fishman Distinguished Professor of Neurology at UCSF and Director of the Wheel Institute. He was Chair of Department of Neurology at UCSF for over 25 years. And I think more than any other person on earth, Steve gets credit for really pushing and developing ocrelizumab as a treatment for MS. So it's really been an honor to be a colleague. And we're also co-founders of the International MS Genetics Consortium. So it's really been an honor in my own life to be such a close friend and colleague as Steve. Okay, so we now move on to, now we're going to show that one slide for me, the PDF I sent. We can do that for a second before we move to the general discussion. It's arriving. So I just want to make a shout out. We're celebrating the 70th anniversary of the Yale Department of Neurology with a symposia. We actually tried to link it with this, but the dates didn't work out, called Progress in Human Immunology, MS Research. It's sold out in New Haven right now, but it's open to Zoom for as many participants. The keynote speakers, Laura Glimcher, President Dana-Farber, Arlene Sharp, who discovered PDL1, Vijay Kutu, who discovered TIM3, Michael Brenner. discover gamma delta T cells. And the following day, have a distinguished group of MS speakers, Jack Antel, who's here, a number of my former students, Amipa Orfil Dieger, Kai Wukafenek, and my mentor, Howard Weiner. And anyone will, if it's okay with Prada, will distribute this to anyone's interested in joining us for Thursday or Friday, but it should be a very special symposia. So thank you. So now move on to the last topic, the general discussion. And first we have Adriana Chio from the University of Torino in Turin. And Professor Chio is professor of neurology, medical director of neurology at the University Hospital in Turin. His research has provided unique insight into ALS, and he's identified a number of the ALS-related genes, including the C9orf72 and KIF5A. So we're honored to have you here.
Thank you, and I want to thank Comi for having invited me and the organizing me, and the Prada Foundation. So my duty now is to talk about ALS. Le frecce. Okay. To talking about therapy in ALS. So there are a lot of therapies that are under study in ALS, probably more than 50. So the time is short, so I'm not going to give a list of therapies, but to give you some suggestions about what happens in ALS. But first of all, something is important about ALS. It's a rare disease. The incidence is only 3 per 100,000, and the prevalence 10 per 100,000. This is very important because we have 50 therapies that are under study, and there is now a lack of patients to be included in studies in a lot of centers around the world. It's a progressive course. The epidemiology of ALS is interesting because we know a lot about the epidemiology in Europe and in North America. We have really few data about other countries. Again, we have a bias of information about ALS, and we also do not know how is the course of the disease in a different population. There are indications that in some population the course could be quite different from the European Caucasian one. ALS was considered a disorder motor system, upper and lower motor neurons, but now is conceptualized as a complex multi-system disorder. We know after the year 2000 that there is an important cognitive impairment. About 50% of patients have a cognitive impairment, and 15% of these are frontotemporal dementia. But most importantly, we know that now that there are also some metabolic component. In particular, the lipids are considered quite important in the genesis of the disease, and just on the contrary, diabetes is considered a protective toward the disease. But this part is really starting to be understood now. We know that a lot of genes have been discovered, more than 30, and they represent about 70% of the cases that are considered familiar, and 5 to 10% of the cases that are considered sporadic ALS. However, the heritability of the disease is much higher, it's probably more than 50%. We performed a study that's been published recently in our population-based series. We found that about 30% of patients had at least one mutation of one of the genes that have been proposed in ALS. And some of these are already actionable for therapy. So probably the genetic component is much more important, and you see the light green, these are sporadic patients. Most of the cases that have mutation are apparently sporadic. But genetics is very important because it's the best way we had to obtain information about the mechanism related to ALS. There is a huge heterogeneity of pathological mechanism. I'm not going to list all of them, but they go from impaired DNA damage repair to nuclear transport, aberrant metabolism of RNA, and so on. This is very important because it's the reason why we have a lot of different trials, because all these possible mechanisms, almost all of them, are can be targeted by therapy. And so there are different therapies that are in the study in phase 1, phase 2, and very few in phase 3. But most important, it's likely that more mechanisms act simultaneously or in sequence in the same patient. So probably a single drug is unlikely to be active in ALS. There are also other obstacles to the therapy to find an effective therapy for ALS. So biological understanding is quite wide, but a little bit confusing. We think that there is a lack of effective preclinical models. SOD1 model, the mouse, is very good, but is very good for SOD1. The clinical and biological heterogeneity is very important, as similarly to other diseases. I've seen a very important indication about, for example, in Alzheimer's disease. And there are huge pitfalls in trial design. In particular, there is an absence of a clear defined biomarker, but light neurofilament could be the right biomarker. That's one point that we can consider. So we can go to available therapies, but it's important to see that we have now 3 therapies that had been marketed for ALS, or are going to be marketed for ALS. The first and the oldest is Riluzole, and it's active on one mechanism that is one of the first proposed for ALS, which is the toxicity of excitatory neurotransmitters, and also called neurotoxicity. The effect is quite low. It's only 10% of increase of survival. And there is no indication that there is an effect on the clinical status of patients, which is interesting because we can separate survival compared to clinical status, at least as what we know. Riluzole is very important because it's the first drug that has broken the therapeutic nihilism for ALS. Anyway, the second one appeared only 18 years later. It is Edaravone. Edaravone is mainly a scavenger of free radicals. The effect in this case has been found only on the LS-FRS scale, which is a functional scale of the disease. You see the difference of treated patients, which is the red line, and not treated patients, placebo patients is the blue, is very small, but the trial was performed only for 6 months, and there was not a analysis of the patients after this period. There was not an open label extension. Second mechanism, second drug, not really satisfying. It's not been marketed in Europe, but anyway, there is something there. Third drug is very recent because only a few days ago, AMX 0035 has been marketed by FDA. It's a double drug, sodium phenylbutyrate and TATCA, with 2 different mechanisms. It's interesting to say, that TATCA has been studied in Italy with a positive trial. Phase 2 trial is ongoing now, a phase 3 trial. So the combination is not clear because sodium phenylbutyrate was studied in ALS in a trial and it was negative, completely negative. Anyway, the effect again is is ALS-FRS, and after 24 weeks, the effect is slightly better than Edaravone. You can see the difference between the two curves. What is interesting is that both Edaravone and the AMX 0035 have been studied with an add-on with Riluzole. So we have 3 drugs in the last trial, and 2 drugs with Edaravone, which is useful because probably, as I told you, the mechanism related to the ALS in different phases of the disease are probably overlapping. So a multi-therapy is probably the best way to have a therapy, to have a treatment for ALS. So we can go to tofersen. Tofersen is a oligonucleotide, antisense oligonucleotide against SOD1. It's it has been proposed after the big success of Nusenesre in SMA, and you have already seen these graphs, but I want to point something about, not only about the effect, but we will go after, but about the biological effect, which is very important about the dynamic of the reversion of the pathological process. The effect on SOD1 reduction, this is a reduction of the protein, not of its activity, was found in the CSF after only 2 weeks, so quite early after the starting of the treatment. The effect on the neurofilament that are considered a good marker, very good marker on motor neuron degeneration, or neuron degeneration in general, appears after 12 weeks to 28 weeks. So it as a peak is here, sorry, after 28, you look at the green line, after 28 weeks. So you have the target, the reduction of the effect on the pathological protein, and then you start to have an effect of the neurons. So rescue, you save more neurons, what else, we don't know. And the same effect appears in the extension, open label extension. The blue line, these are patients treated with placebo, and from this point they are treated with the drug. So they have exactly the same effect after 6 months. The effect on the clinical aspects is particularly evident for spirometry, for the respiration, which is significant already at 28 weeks, but it appears too evident in the open label extension with an important reduction of the LSFRS and the strength of the patients. So the clinical effects appears after 28 weeks, which is quite logic. You have effect on the target, effect on the neurofilaments that indicate that you are rescuing motor neurons, and then you have the clinic. You need to have a collateral innervation or what else to have this. This is important because, in particular in US, after the pushing by the Patients Association, they want to do very short trials in ALS, which is probably not the case. We need more. We need a longer trial to see some effects, some clinical effects, because obviously we cannot base only on the biomarkers, but we still need to have something about clinic. What is interesting, we are talking about the possibility to make something in pre-symptomatic. A study, Atlas, is ongoing on pre-symptomatic patients with SOD1 mutation. Going to the end of my presentation. We are going to something more than in ALS, at least the personalized medicine, is the custom drug for just one patient. This is a bill approved by the Congress of the United States to allow the production of antisense against a single, for a single patient, against, which is Jassy Helmstead, is indicated as reporting the bill. She has a FAS mutation, which is one of the most aggressive gene in ALS. This drug was produced by Ionis just for the patient. They had the opportunity to see some effect, and so a trial started after that. The trial is ongoing. It's a phase 1 trial, really. It's not a phase 3. It's indicated. This is a mistake by the press release. Several patients are already on treatment. The problem, the fact that we are going to have one-person trial could be with one-person drug is very interesting from one point of view. Could be a problem in the future, in particular for rare disease. I don't imagine that something is happening with common disease, but maybe with some rare mutation in common disease could be also the case. So I want to end with my thanks to all the group in the ALS Center in Torino, where we participate to a lot of trials, and we hope that we are going to find something new in the treatment of ALS, but we are just happy for what happened the last year. Thank you.
Thank you. The last short talk of the session is by Alexandra Dur. from the Paris Brain Institute, the ICM at La Petite Salpêtrière. Professor Dora is head of neurogenetics in the Reference Center for Rare Diseases and specializes in spinal cerebellar degeneration. So, thank you.
Thank you very much, David, and thank you, all of you, being still here. And the short talks are not really short, so I try to be short. And thank you so much to the Fondazione Prada, because it's a beautiful place to be here and see so many people working on other diseases where we don't have our head in, and so we learn a lot. I'm at the Paris Brain Institute, where I can conduct the translational studies I like to do, because I'm a clinician in the campus of the Salpêtrière Hospital. And I would like to go back to the idea that the neurogenetic diseases need age, especially old age, to appear. So in an inherited neurodegeneration, the question is, if there is a pathogenic major variant, like in Huntington's disease, which gives rise to the disease in full penetrance, how come that this disease will appear 40, 50, 60 years later? Because it's not a switch on of the gene at one point in the age. It's there, it's doing its toxic role from the beginning. So what is happening, and I'm rather thinking about more compensation, or in the beginning we were talking, yesterday we were talking more about resilience and compensation, and I think that is something important. So I would, because Huntington's disease was not yet introduced, I introduce you to Sophie, who shows you the disease with chorea and dystonia and no speech in her case, and probably she would not be able to be included in any of the trials out there. Huntington's disease is due to an expanded CAG repeat in one of the two genes. So you have the coexistence of the abnormal one and the normal one. It's true that usually the age at onset is late, but you can see that you have nearly all the ages present. I mean, most of them are between 30 and 50 or a little bit later. What we have as a treatment options, thanks to the psychiatrists we're working with, we have a lot of neuroleptics. We have tetrabenazine, which is not working very well, but there are still treatment at this stage for the disease. The more interesting question now is, what is driving the onset of the disease? Because we need to know if we are able to estimate the onset of these diseases, we can go back, see what are the pathological changes before, and then intervene the earliest possible. So what we know in Huntington's disease is that the size of the CAG repeat very clearly is related to the age at onset of the disease, because the longer the CAG repeat, the earlier the onset. This we know for a long time. But also the nature of the CAG repeat is important, because if there's an interruption after the CAG repeat by a CAA, which is also encoding a polyglutamine, but if this is mutated to a CAG, the age at onset will be earlier, meaning that you need a pure stretch of CAG repeats to drive the onset of the disease. The second very important one, which has been shown in 2016 by Leslie Jones, who unfortunately disappeared very quickly, and she really did an immense work on the GWAS study in this residual age at onset that is not driven by the CAG repeat, and you can see that there's a major peak in the DNA repair genes or complex, and this is probably very important to drive the disease. In the relation to this, we were able to show, and this was possible only because we had 30 years of sampling of these patients for a long time, because to show somatic instability over time in blood, you need samples that were taken a long time ago and today. So seeing that it's with age, you have an unstable CAG repeat in the blood and in the brain probably, is an important driver of the disease. So what do we know in patients, we know from the study we did with Sarah Tabrizi and Vancouver, Blair Levitt, and Raymond Ross, that there's atrophy in the symptomatics that you can quantify very well, but we know also that in the pre-symptomatic stages, you have atrophy that is already present, and it's really smaller protein volume are characteristic of this pre-symptomatic stage. Again, we have neurofilament clearly different in the pre-symptomatics, far from the disease. They have no signs at all of the disease. And more importantly, perhaps, that's why I was saying that we didn't switch on the gene at the time, is that we showed in fetuses from, they were aged 13 weeks of pregnancy, that the cortex was already completely abnormal. There was abnormal localization of the mutant huntingtin that was more concentrated. You had a defect in the polarity of the cells and the differentiation of the neuronal precursors, and you had abnormal cellulogenesis. So the question is, if there is an abnormal neurodevelopment due to the presence of the CAG repeat, what happens that the disease is finally clinically visible only there? And I want to finish with a trial which didn't go very well, and Kes Mamori did allude to that before, because you can, I mean, from all the other diseases we heard of, tau and TDP43 and alpha-synuclein, you can, if you do a very protein-centered approach where you want to get rid of this aggregation, you can see here, you want to get rid of the abnormal protein that is accumulated in the cell. You can do an ISO, like in the what we saw before, with decreasing huntingtin, as you can see here on the slide, for the huntingtin protein by the antisense oligonucleotide. And it's very well dose-related. The more you inject, the lower you have the huntingtin. The problem was that including more than 800 patients in this trial, nearly 250 in each arm, injected every month or every second month, every 2 months, you can see that the placebo, which is in gray here, and you have the 2 months injected in yellow, and in blue, the most frequent, or the highest dose, the most frequent injected, you can see that the patient is going worse and worse, and that's why the trial was halted. So the question is, what is the reason for that? It's not really only to get rid of the protein that is accumulating in a stage of the disease where we are already having clinical signs, because perhaps the ISO1 itself is doing a lot of bad things. That could be, but it could also be that the presence of the normal protein or the interaction between the abnormal and the normal protein are important to maintain this compensation over time. So, as we can say, and as we all said before, treatment should be early, but we know that brain development is impaired, so probably treatment must be different in the beginning of the disease, in the middle, far from onset, or closer to onset. We know that the silent phase is not soundless, because biomarkers are clearly showing pathological events or dysfunction. And I really think that the brain is adapted for compensation to avoid overt signs. They are not developed for several decades over time. And Sandrine Imbert, who did this work in the neurodevelopmental field, recently showed that there's a synaptic defect, which is quite subtle in the newborn mice with the mutation. And if you treat them very early at this time to get rid of the effect of this synaptic dysfunction, you don't develop the disease later on in the mice. So this gives an idea that perhaps trying to treat very early, but not very long, at the right time, I think that is the more important message I want to say, that treatment should be early, it's more at the right time, is perhaps something we should pursue. And I stop here and I say thanks to my colleagues at the ICM.
Thank you. I'd like to invite all the speakers to come up to the table for the questions. Okay. How long should we go for? As long as we want. Okay. Thank you. Okay. I would like to start with a general comment. One theme that's really emerging, and Steve really showed this in MS, is the earlier you treat, the better the results. And we're starting a clinical trial at Yale, multi-center trial of patients with RIS. Now, RIS is multiple sclerosis, so that still may be too late. We clearly need to identify disease earlier than that. But the hope is we can prevent the neurodegenerative aspects induced by either T cells, amyloid, tau, pick your poison. But the earlier we treat, clearly the better the outcome. And I think that's going to be really the key to everything we do. Okay, questions? Yes, Catherine.
A question to Alexandra. Great talk. So it's very impressive to see the abnormality in the cortex in the newborn, even before newborn, in the embryonic brain, and which remains more or less silent, maybe with compensation. And I had maybe naive question. Do you know if the number of repeat, CAG repeat, is correlated to the amplitude of the abnormalities? And that might explain why it appears earlier?
It was quite difficult to get these fetuses,
sure,
but
sure.
the 7 fetuses where we could study the cortex, they had quite normal CAG repeats, meaning between 40 and 45, so not large ones at all.
Not large ones.
So, which gave us the idea, because we know that the disease is really driven by the size, that it will start quite late in life, and still, so we were not, we were so surprised when we saw, and even I'm not a biologist from the cell on the cellular level, it was so visible that we thought, oh no, they don't have the same CAG repeats. Yes, they have small ones. So I cannot tell you if in a juvenile case, for example, it's very large, probably there must be even more disrupted cortical layers, because these children are never normal in a way, because the juvenile cases they have a neurodevelopmental disease in a way, because they have intellectual deficiency and other difficulties. So it's really not the same disease when you have very large or normal.
Alexandra, do you think any common genetic variation, which does seem to affect Huntington's disease, may have affected your results? Could you look at that in regards to how they may have responded to the drugs?
You mean genetic variation in
common
outside,
one? Right, outside of the Huntington gene.
I mean, in the GWAS, there were several peaks, but the most important peak was in the DNA repair system, and it would be interesting to look at them to see if, because you still have a variation in when things happen. So even if we know for the CAG repeat and the instability, and there are other modifying factors that play a role, but how to look at them very early on would be interesting.
Michaela.
Again to Alexandra, very nice talk. In animal model of ALS, the SOD mice, and in animal model of 5X FAD mice, it was shown that when you breed the mice with SCID mice, you see earlier onset and more severe disease. It was shown very nicely by Stanley Appel in SOD mice and in 5X5 by a group in California. So my question, so again, I'm obsessive about the role of the immune system, but it was shown very elegantly. So I think that the question in Huntington is no much different from many other familiar diseases, that you're born with the mutation and the disease onset is later on in life. So it may be that there is accelerating aging of the immune system. That's my question.
It could be, but the normal function, which is a bit different for the other diseases I heard of during the meeting, the normal function of Huntington is very well studied. And there are a lot of different functions in the cell, but I mean, the transport mechanism or cell divisions, and it's a scaffold protein, so it's very important for a lot of cellular mechanism. For the immune system, I mean, why not? But it didn't seem to us, at least, that it was on the first row from the dysfunction or from the pathogenesis that would explain the disease. But again, it seemed to play a role in all of these diseases. That's what I learned here. So let's see.
Yeah, the immune system. Yep.
Well,
and then we'll do.
What is happening in MS, as you know, David, we are starting now to consider a combination therapy to target together the acute peripheral inflammation and the confined in CNS inflammation in some way. So then the question is, because this combination of factors has emerged in all the conditions, chronic condition, is there any evidence that there are some initiatives moving in this direction, Alzheimer, Parkinson, ALS, to combine together particularly a target on the immune component and a target on more the degenerative component, or whatever it is, based on the different pathologies?
Yes, please.
I mean, I think it's an excellent point, and it's something that we need to consider across all the neurodegenerative diseases, and the fact that we're sitting here together talking about the same thing indicates that, which is fantastic. We've got a lot to learn from MS in many areas. This is one. Randy mentioned in his talk that the DIAN-TU study is about to study a combination of an anti-amyloid and an anti-tau therapy. That's the first time we'll be using two different therapies, and because it's in the genetic population, we've got a much better handle and much better profiling on those patients. So that will be a good start, but there's an awful long way to go. I think you're right, and that's the direction we should be thinking about, but I think it's slowly catchy monkey, I guess. We don't want to put it all in one basket.
Other comments?
I can comment to... I don't know of any trial in Parkinson's disease that combine therapies, but I mentioned about this with the GLP-1 at the end, and I think that type of compounds may actually work on several different mechanisms. So now I said that it works on, based on the data of Ted Dawson on activated microglia, but we heard already yesterday that it also affects metabolic states, and there is also quite high abundance of these GLP-1 receptors on dopamine neurons themselves. So it may be that by using this compound you actually act on many different sites, and that's why it may be promising.
Well, in ALS we are trying to combine, obviously. We have no trials on aspects related to inflammation at the moment. We have trials related to the drugs acting on complement, which is not exactly the same, but there is something related. By the way, the second trial that I made when I was a young neurologist was with beta interferon in ALS, so it was, unfortunately, it was unsuccessful. You can imagine why.
Just briefly wanted to add something. If these drugs are approved by the FDA next year, we're inevitably going to be starting to do combination drug trials. So I think that's going to be a big change in the complexity and the way that our studies move forward.
And I would just add that if we can stop the initial event, that may be all we need to do if we can go early enough, before the degenerative phase begins. I'm very curious to see what happens with the clinical trial that Genentech has started, with a multi-center trial, using transferrin receptor hooked up to anti-CD20, which allows the antibody to cross the blood-brain barrier. And that may be critical, because if those B-cell follicles, getting back to Professor Lossman's discussion yesterday about the cortical events, which are clearly important and driving the disease, if we don't get rid of the cortical, these B-cell follicles, or whatever they're secreting, we may never be able to treat progressive disease in MS.
You made a very important point about compensation, because this is for Huntington, this is for ALS. I think that we have probably to concentrate on the mechanism of compensation. That's a solution of everything somehow. We had, for example, a family with FOV SOD1, so the most aggressive. Most of the family was affected except one. So the question was, what is so different in this person related to other ones? So maybe we have a solution under our eyes. We have to just to understand what's going on.
Monique Breteler yesterday said that too, that we have to get interested in people, they are not getting it.
Okay, here.
It was for Kat, but you just addressed my question about the FDA approvals, so that was a short one.
That was a fast approval. Okay.
And so I'm hearing the promissory notes of being able to go earlier and have a significant impact on other diseases. So in SMA and multiple sclerosis, heading earlier really seems to, and I think that's a general truism in medicine, isn't it? That you go earlier, you prevent, it's bound to have an impact. But what I've picked up on is maybe a cautionary note, and I wonder about the impact of the Huntington's trial. And you mentioned this about the effects of lowering down normal Huntington expression could have an untoward effect or unexpected effect. What does that tell us about other targeted therapies? For example, in Alzheimer's, there are programs that are considering lowering down normal expression of protein, tau, APP, these other things. What can we learn from the Huntington's trial that we might apply to other areas about lowering down normal physiologic function, as opposed to the SMA case where I think it's more specific for the mutated form?
Yes, I think we should learn a lot from that, because even in the phase 2A trial where we downregulated the HTT protein, you had NFL going up at distance of the injection of the ASO, and you had increased ventricles. And this was reproduced in the phase 3 trial where we took the highest dose to have this dose effect, because everybody was scared not to downregulate enough of the bad Huntington in the cortex, because we injected it intrathecally. And so everybody was saying to go into the striatum and to bring down the Huntington, it has to be the highest dose. Probably it would be really great that Roche and the others would try to understand why this happened and why people with the highest dose and the frequent injection got worse on motor, cognitive, on all the readouts we had. So probably you could say that the polyglutamine Huntington, it's not functioning. It's not a loss of function or a toxic whatever function you invent. It's just that it's working a little bit, but not enough. And the normal one is compensating in a way. But when you then pull down both of them, that's really bad. So the idea would be rather doing something else on another pathway, or increasing other more protective pathways than pulling down the Huntington, I think.
Alexandra, may I ask, can you comment also on the allele-specific ASOs? I mean, WAVE's trial and so on, when you just downregulate the mutated Huntington?
Yes, the WAVE trial, WAVE Life Sciences tried to develop allele-specific downregulation, which is more logical, but you need, because the mutation is, and the normal allele is the same, it's just a longer CAG, so recognizing this needs another SNP. So SNP1 and 2 did not allow it to downregulate enough Huntingtin. So the last SNP3 the wave life allowed to downregulate this time. So we will see, but it's a phase 2A. It's safety more than efficiency. So, but let's see.
Last question, Alex.
Well, it was more a comment in line with what has been said, because I think we have seen many genetic, dominant genetic disorders have gain of function, like for alpha-synuclein, Huntington, the spinocerebellar ataxias with expanded alleles. And we have considered that it's just a toxicity of the protein, but we haven't considered much that there could be also a loss of function. And if this is the case, because the function of the abnormal protein can be a gain, but also partly a loss. So if you reduce the level of abnormal protein, which has still some normal function, then you can induce a more severe disease because of the loss of function. So we have to consider this, and I wanted to ask Pierre whether that's not a problem that we could encounter in the ongoing studies on synuclein, for instance, which is very abundant, but we don't know exactly what happens in the human brain if you decrease its level that much.
No, it's a very good point. One aspect with synuclein, unlike Huntington's disease, it, at least in animal models, if you make a knockout of alpha-synuclein, you don't get that much phenotype. I mean, the Huntington knockout, the mice die. So that's at least an argument that it could be relatively safe to downregulate alpha-synuclein. But you're absolutely right that it's...
But is it not that it is too abundant? I mean, alpha-synuclein, if you try to measure it, it's in the CSF, it's everywhere.
Yeah, and it's also very high in erythrocytes. Yeah, so it has But again, the knockout mice at least have a quite mild phenotype. So that is at least an argument that I think it's worth trying in advanced cases of MSA or PD.
Maybe I'll just end by saying it's probably an autoimmune disease anyway, so it doesn't matter. Let's do the experiment. I want to thank all the speakers and the audience for a very lively discussion. Thank you.
Okay. Okay. Welcome back to this session, which is the last session for today before the closure. My name is Alberto Albanese. I have the pleasure to invite to the podium Professor Naldini. Professor Naldini is an expert on gene therapy. He's professor at San Raffaele Hospital and University with a strong expertise in the field of gene therapy. So thank you for being here.
Okay. And just to go on. But it's not working from here. It's blocked. That's right. Okay, so first of all, thank you so much for the invitation again, particularly to Giancarlo Comi. I have a disclaimer to make here because, of course, I cannot talk about gene therapy of the severe neurodegenerative diseases we have been discussing for the last couple of days. Unfortunately, we don't have a clear understanding, as I've learned even more here, of what would be the target to address with the gene therapy approach. But I think the point of my presentation will be to discuss the tools that we have developed for other brain diseases, which I think can inspire future development as we can better understand what the focus would be. Unfortunately, this is
La prima diapo, per favore.
I can see from here, but I think there is a riabilitare il contenuto. They have to do from there. Okay, if they do, they have to. Okay. That seems to be okay now. I think I can go from here. So we have a number of fundamental advances in medicine that can allow us to address severe disease with unmet need by new strategies. And I think they mostly are based on the capacity to manipulate the genes of a cell by introducing new genes or editing, specifically sequences, isolating and transplanting a stem cell for the regeneration of tissues, and also harnessing and modulating immunity. And if you put together these three pillars, you can really think of the potential to design new treatment for severe disease with unmet medical need. Now, currently, if you are approaching gene therapy strategies, you have a number of tools available, some which we have had for a long time, like essentially viral-derived vehicles to transfer genes into target cell. These are mostly derived from either lentiviruses or adeno-associated viruses. We have more recently in this armamentarium a number of tools to edit the genome based on Zinfinger, TALEN, and more recently and more widely CRISPR-Cas technology. We also have improved approaches which may allow to have single base changes with high precision, or even changes in the epigenetic features of a specific locus. If you look at the time since we have had these tools and also when they enter clinical testing, as I mentioned, we have the longest experience with the gene transfer technology, which you see on top, while there is a much shorter, in fact, experience, and only for a couple of years for the gene editing tools of oldest, let's say, age, while the rest are still in the R&D stage. Now, gene therapy can be deployed by an ex vivo approach in which you can manipulate a cell of interest and then transplant it back in the tissue. or directly in vivo by transferring your cargo, whether it's a gene or your editing tools. While in vivo direct gene transfer into the seems more appropriate for treating CNS diseases, it also necessarily confined to the delivery site and may not have a pervasive, a broad-acting strategy, while you can achieve instead by some of the other strategy ex vivo. And this is where I'd like to actually focus my presentation also, because the experience in our institute here in Milan on developing a stem cell based or an ex vivo gene therapy strategy to tackle a variety of diseases, including neurodegenerative disease of childhood. And I think that what we have learned from that approach should probably be relevant also for wider application. Now, stem cell gene transfer is essentially leverages on the long clinical experience of transplantation from an allogeneic donor. In gene therapy, typically the cells are harvested from the same patient, so it's an autologous product which is manipulated ex vivo for gene transfer or gene editing, and then is engrafted after a conditioning which makes space in the marrow for the cell which are infused back. If this is successful, we establish a source of gene-modified progeny cell of the different lineages which may distribute into the body. And intriguingly, some of these may also actually reach peripheral tissue, including the CNS, so the microglia and, of course, macrophages throughout the body. So the promise of this strategy has been to treat a variety of immunohematological disease, immunodeficiency, hemoglobinopathies, but also storage diseases which affect different organs, and potentially also cancer and infection. And we have now several hundred people treated worldwide by this treatment for different diseases, some of which have even reached now the stage of actually approved drug. And our institute here in Milan have contributed to at least 2 of these approved drugs, which are among the first ever in the world, which are now available to patients. Now, one of these is actually, as I mentioned, is a neurodegenerative, essentially a dysmyelinating disease of early childhood, in which there is a lysosomal deficiency preventing proper catabolism of myelin, essentially sulfatide. And the absence of this proper enzyme leads to accumulation of material in microglia, as you can imagine, but also in a variety of neuronal population, neurons, oligodendrocyte, and affecting most of the brain and becoming lethal, essentially, in the second childhood, about 10 years of age. Transplantation of donor stem cell for this storage disease is not routinely recommended because it's poorly efficacious. However, what we develop, a stem cell gene therapy approach, is based on the concept that when you condition a patient before transplanting stem cell, whether autologous or allogeneic, conditioning which is based on chemotherapy or also radiotherapy, deplete tissue sources of myeloid cell, including microglia progenitors. And so the infused cell, presumably the progenitor themselves, will home also in the brain to replace the depleted niches. And if these cells have been engineered to be functional by a vector gene transfer, they will not only reproduce functional microglia cell for clearance of the product, but as this is a lysosomal enzyme, part of this lysosomal enzyme, functional one, will escape the sorting into the lysosome and be released by the transduced cell into the microenvironment surrounding, to be captured by neighboring cell, which will then sort into their own lysosome. So this is a typical cross-correction approach. This is routinely used to treat storage disease by providing a recombinant enzyme, which was captured by different tissues, cell in the body. Of course, this is hardly doable for a brain disease. You would have to inject the lysosome enzyme every few days into the brain. However, if you establish a local source, you may allow essentially cross-correction of the population. And the key for our strategy, because I told you that stem cell transplant from a normal donor is not really efficacious, we model into the mice that because we are engineering the cell, if we actually made the cell by our vector transfer overexpressing the enzyme to supranormal level, we would have an improved actually biodistribution into the CNS, provided that, of course, this would not affect the biology of the cell, which we proved to be the case. So based on these studies, of course, we now more than almost nearly 10 years ago, we have started clinical trials, which led, as I said, to final approval of this treatment. And these are some of the long-term results of the first group of patients treated here that you can see into this slide. And you can see here, for instance, this is the level of essentially engineering of cell in the periphery. So it means 70% of hematopoietic cell, including monocytes, and the other are carry the vector. So these patients are essentially almost transgenic, if you want, in terms of the hematopoietic system. And this leads to an excess activity of the enzymes, if measured in the blood cell, as shown here. This would be the normal range. You see, most of these patients actually would, while they didn't have detectable activity before gene therapy, now they have over activity. Of course, this is not relevant for treating the disease. What is more rare is that if you look in the brain, and this is by sampling the cerebrospinal fluid, we do detect now bioavailable and functional enzymes in the normal range, essentially indicating that there has been, in fact, production locally as expected. And now this has translated in an amazing clinical benefit beyond even what we would expect based on our clinical studies. And this is only one of the parameters that I'm showing you. This is a gross motor function scale, which essentially monitor the acquisition of motor skill as children grow. This is age by year. The gray area would be the normal, essentially, motor acquisition of children. These are the children that we saw, and that we see actually in the clinic when they are already symptomatic. You can see this baby here, unfortunately, they are really severely affected, and as I said, they end up bedridden and unfortunately without any hope. We could not even think of treating such a brain damaged child. Of course, it would need much more than gene therapy, maybe what you will hear next, I think, in terms of cell replacement. However, we could treat the sibling of such patient. Unfortunately, in the same family, children with the same genotype were born at the time the diagnosis was made for the first one, and they were early in their disease progression, maybe very poorly symptomatic, such this brother of this girl here, in which you can see here now 7 years after treatment is essentially a normal child. As you can see also in the scale upper there. So that essentially early treatment prevent disease development even, and this has been true for the vast majority of the patient treated, as you can see in this graph here. Unless we treat when these symptoms are already established, like in a couple of cases which were essentially providing essentially try to test also the treatment in some sibling which are already symptomatic. But by the time that the transplant establishes itself, the disease rapidly progresses. So one of the questions, of course, you may ask is this high level of engineering of hematopoiesis, is it safe or not? Are you actually, early study of stem cell gene therapy provided, of course, benefit in some patients, but also adverse event, essentially leukemic outgrowth. And because the vector integrate randomly, we can actually sample all the cell that we have modified for the site of integration, which is a clonal genetic marker. So we can actually monitor clonality in the population. If you look at some of these early cases, not from our study here, but from another study in which a leukemia developed after a gene therapy, this was using an early generation type of vector based on gamma retrovirus. This is essentially the composition in terms of clonal composition of the peripheral blood. The gray bar indicate a multi-clonal, so you don't really differentiate the individual clone. But at a certain point, the clone emerges and starts to expand with time, becoming progressively more abundant. Such a clone typically bears a vector integration, which was semi-random, into an oncogene, and they become eventually a leukemic outgrowth. So essentially, while you start from a polyclonal composition, you may see in vivo cells which have received a gain of function by a random insertion near like a cancer-associated gene may expand at the expense of the other, eventually develop into a tumor. Now, using the new platform based on lentiviral vector that we have been using on all our studies, as we have learned some of the mechanisms, I have no time to go into the genetics of this, but essentially the pattern is very different. You can see essentially by the color coding here that you have several thousands of clones which are concurrently active. There is no imbalance between them, and this remains very stable in the long term. So essentially polyclonal engraftment remain stably maintained throughout throughout. And we don't see any sign of decreased composition or outcome, indicating that not only the patients are doing well from a clinical standpoint, but they also are, from a molecular analysis, showing a safe reconstitution. So if you look back by a meta-analysis to all patients treated with stem cell gene therapy, the incidence of genotoxicity, as you can see in this graph, is about 20% if you look in the long-term follow-up, but this only applies to patients treated with early generation gamma retroviral vector. There is virtually none, except for maybe a couple of cases reported among the hundreds of patients treated with the lentiviral vector new second-generation platform. So overall, you can say that potentially autologous stem cell may provide a preferred approach to allogeneic transplant in genetic disease because it's available to every patient. You don't run the risk of graft versus host reaction. Mixed chimeras may be sufficient for benefit, and in particular, you may build enhanced benefit by increasing gene dosage. There are, of course, outstanding challenges, but what I'd like now to move on to the second story, which is built on this one. So what is happening to this into this treatment is that the microglia cell become not only functional, but also the source of a conditioning and a cross-correcting activity for the other cell. So can we deliver into the brain not only a lysosomal enzyme, but also other biotherapeutics, maybe by similar approach, and potentially treating through a replaced microglia or macrophage population, providing growth factor, for instance, or, and that's what we have been working on, for instance, anti-tumor biologicals. And again, here the concept is any tumor, as you know, is made of by cancer cell, but also by an important stromal component, which is essentially recruiting blood cell, especially monocyte and macrophages, which are also becoming co-culprit of tumor growth. Typically, they are pro-tumoral, pro-angiogenic, and they also are immunosuppressive, preventing anti-tumor, essentially, immunity. So the idea is that we could potentially rescue this immunosuppressive microenvironment of the tumor by providing, for instance, some biotherapeutics like interferon or cytokine, which are very powerful immunostimulating molecules. But if you deliver, as you well know, like interferon systemically, you may have some benefit against a viral infection, cancer, or other diseases, but you also have severe adverse event, which is probably very well known to this community. Part of which is due also to this oscillatory pattern in which by recombinant administration you have peak and trough, and during the peak you may enter into the toxic concentration, while into the trough region you may lose activity. Ideally, stable activity of such a cytokine at the disease site, sparing the periphery, would be a much more powerful strategy and potentially better harmful. So this is what we have been working on. Essentially, can we use the monocytes which are recruited into the tumor, and as you will see in particular in the brain tumor, as a delivery vehicle for interferon? We will still put the gene for the interferon into the stem cell because we need to have a continuous supply of the modified cell into the patient, but we want to have selective expression in the monocyte lineages, and in particular, preferentially at the tumor site, to prevent, of course, systemic exposure. This requires a sophisticated engineering of the vector for its expression control, which is briefly shown into this schematic. We are using essentially a promoter to drive expression of our interferon, which is typically expressed in a subset of macrophages, typically associated with angiogenesis and tumor growth. So this promoter essentially would this promoter, however, also has some low-level activity into the stem cell compartment. So what we have done here is to build a cassette which uses the promoter and answer sequences of this type 2 gene, which is the one I was telling you about, which would have some activity in the stem cell and higher activity into the macrophages infiltrating the tissue, and in particular at the tumor site. And then we modify the cassette, the transcript, to include target sequence for microRNA. MicroRNA, as you know, as a micromanager of expression, they can suppress translation of a transcript. And so we identify microRNA which are present in the stem cell compartment, but not in the mature myeloid cell. We made the vector, the interferon transcript, comprising target sequence for this microRNA. So any sequence now, which is any transcript which is made in the stem cell to low level, can be cleared while in the periphery, and in particular the tumor site, where this promoter is upregulated by the hypoxic condition, we now have a full expression of the transcript and interferon delivery. So we show that this approach essentially allowed to condition specifically a tumor by delivering local interferon in a number of tumors, as I said, in particular in a glioblastoma model, for which I reported recently. You can see here that these cells in red are the one which we are expressing this type 2 essentially gene naturally. They would express our interferon. These cells are only present in the tumor infiltrate, and they are not present in the normal brain. So there is a selective component of delivery here. And then if you challenge the mice in which we have engineered in this way hematopoiesis, you can see a significant inhibition of the glioblastoma model grow in an immunocompetent mouse. And this also allowed not only to delay grow, but also to allow a fraction of mice to completely clear and reject the tumor by building an anti-tumor specific immunity. And by single cell analysis, we could show that we actually can modulate the microenvironment so that the macrophages population, I'm not going into many detail here, but we can see that we have changes in some of the tumor infiltrating macrophages, some of which, for instance, this population here identified by single cell analysis, which completely disappears upon interferon gene therapy. This population is typically made of protumoral macrophages, which are associated with their signature to poorer survival essentially in human patient. And on the other hand, we see appearance or enhancement of this population of inflammatory macrophages, which can typically stimulate immune response. This is where the interferon activation can be seen. So essentially, we can deviate monocyte differentiation in the tumor from a protumoral pathway to an anti, or if you're an immunostimulatory pathway. And if you look at the T cells which are present in the tumor, they have a lower sign of exhaustion. And intriguingly, if you compare, and I just skip to the graph here, interferon administration by conventional route versus interferon gene therapy, you see there is a much better benefit of the interferon gene therapy. The interferon administration works initially, but then essentially there is loss of the benefit. And of course, there is exposure of the periphery, including of the normal brain to interferon stimulation by systemic route versus lack of interferon activation by our targeted gene therapy strategy. So it is possible to achieve a very significant impact on disease, in this case tumor growth, by selective delivery of a cytokine which acts biologically to reprogram the immune infiltrate locally, and building on both a cellular vehicle, which is selectively recruited, and a specific vector design, you can achieve a superior activity than conventional administration. Now, through a spin-out of the San Raffaele Hospital, this strategy has now been entered clinical testing in glioblastoma patient in a typical dose-escalating trial. I can only briefly report that we have not seen any toxicity limiting, essentially scaling up of the doses into the patient. This treatment has been well tolerated. It's based essentially on an autologous stem cell treatment, which is following the diagnosis and the first surgery on the patient. And the cells which are modified and grafted to the input doses are stably present, and we have some evidence of interferon activation on the tumor side, which can recapitulate some of those data that we saw in the mice. Of course, so this is early data. This is a phase 1/2 trial, so we cannot comment on benefit at this time point. We have to go. But I think at least the feasibility of these studies in humans is highly rewarding. I will not discuss in vivo gene therapy, which of course, as I said, may well be suited for CNS disease, in which local administration of vector, more typically an AAV vector, can allow, of course, reconstitution or expression of a therapeutic transgene into a specific site. This is, of course, a very highly active area of investigation. There's also particular design of vector which can allow to cross the blood-brain barrier, so reaching the brain from a systemic infusion. There's drug as well, at least for ocular disease, which have been approved already based on AAV. And there is a lot of work ongoing, although there are hurdles here as well in terms of not every patient can tolerate an AAV gene administration because there is pre-existing immunity. There are the issue of toxicity by increasing doses. at the hepatic level and maybe a neural level. And however, I think this is an area which, of course, is very active. So to finalize, to close it, we are seeing an evolving pharmacology that moving from small drug and biologicals, and you can think what really biological are made to revolutionize current treatment of diseases. Now we have genes and even cell which become drugs. And of course, the power of capturing biological processes like this one for treatment disease is really a new area of medicine to start with. Challenges remain. These are complex drugs, of course, which requires better understanding of the biological process, of the innate response, and the immune barriers that we can find as we administer them, the safety of this drug, which can have long-term benefit, but also long-term adverse effect, has to be monitored. Bedside delivery become challenging, and societally, there is also challenges of really allowing this treatment to become available because of the complexity and the cost of manufacturing. So we are all concerned about making sure that eventually when we develop this drug, they are available to the patient who are in need of them. And finally, ethical issues may also have to be considered. This is what I'm discussing. We all been working on is somatic cell gene therapy. The treatment changed the DNA, but in a cell which will die with the patient. And of course, as long as we provide this treatment in the context of existing ethical norm and the rule of a clinical trial, which are performed based on risk and benefit, this is well essentially managed. Things may be very different when we actually enter the area of germline gene modification, which with the current editing strategy is possible. And here become much more challenging, of course, how to move along. We are not treating disease here. We are preventing potentially transmission of a disease-causing gene. Think about some of the predisposing gene that you have been discussing yesterday. Eventually, you could potentially consider edit that in a germline of an individual, but this is still technically very challenging, and hopefully this is not going to be in the clinic until we are ready to accept this in terms of safety and also, of course, ethically. Unfortunately, this has been done already in China in terms of testing editing of human germline. This, of course, has triggered outrage, and now there is a stronger, I think, emphasis on preventing further clinical testing until there is a consensus of how to move forward. But of course, this is something we have to be aware of. With that, I thank you for your time. And this is, of course, to acknowledge the many people in my lab, but also in the clinical staff which have been contributed to the trial I briefly discussed. Thank you.
Thank you, Luigi. Thank you. Now I call, so we shall discuss at the end. Now I call Stefano Pulchino to speak about advanced cellular and acellular therapeutics. Stefano Pulchino is professor of regenerative neuroimmunology at the University of Cambridge and Department of Clinical Neurosciences. He's an expert in the field. So yes, welcome, and you have 20 minutes.
Thanks very much. Let me join the previous speakers and thank Professor Comi and Fondazione Prada to give me the opportunity to speak to you today. It's always very tough to give a talk after Luigi Naldini. You have to be able to keep the audience alive and interested on the level of technology and the quality of science. But nonetheless, my job today would be to convince you in the next 20 minutes that there is lots of interest and lots of promise and expectations around advanced therapeutics with a specific focus towards cellular therapeutics and a little bit of evidence in favor of acellular therapeutics. So the outline I'm gonna go through will include a rationale and a promise at the beginning, then the technologies themselves available, the potential mechanism and mode of actions. I'll give you a couple of case studies as examples, and the final agenda. So the rationale for developing cellular or stem cell therapeutics in brain diseases stands on the fact that there is long-standing evidence that the injured adult mammalian brain is able or hospitable for regeneration. And that evidence dates back more than 100 years ago, and it applies to this very elegant yet very, very pioneer study from Ramon y Cajal and Francisco Tello, showing, as you can see here, that in the brain, if you put in very close contact a peripheral nerve stump with a sciatic nerve graft, they start to talk to each other, and there is initial evidence of regeneration. So that's a proof of concept evidence which has been expanded in the years following, and therefore the promise for cell therapies and the one for stem cell therapy is a little bit different. Cell therapies basically hold the promise to succeed where nature is not fully able to succeed, or where eventually nature fades. On the other hand, stem cell therapies hold the promise to succeed beyond the high number of proof of concept studies that we have read, starting from an initial very conservative cell therapy approach. So cell and technologies available is something important to keep in mind, and Luigi already gave a little bit of introduction on the capabilities that we as community have. In terms of cells, we have lots of evidence that cell therapies can develop out of somatic cells, somatic progenitor cells, either primary or differentiated, somatic stem cells, embryonic stem cells and progenies, and even induced pluripotent stem cells and their progenies. So there is lots of cells available to develop experimental therapeutic approaches. And in terms of technologies, again, technologies available to reprogram cells, to engineer cells, to develop chemically defined in vitro tissue culture protocols for large-scale expansion of cells, and bioreactors for large-scale production of cells for clinical applications. So there is two levels of expectation that I think we have to agree on. One is on the cells. And eventually, when you develop a cell therapy, you have expectation that your cells will be surviving, they will be differentiating in vivo, they will be integrating into the host, and rescue acute disease-associated functional deficits. They might be also able to deliver therapeutic moieties and molecules. In perspective, and this is a very hot topic nowadays, especially around chronic neurodegenerative conditions, they will be able to slow chronic disease progression and eventually preserve the brain or restore lost functions. So basically, this is what those expectations might be put together in a figure, in a graph, where cells are injected into the organ that is affected by an injury or a disease, and there is continuous dynamic interaction between the cellular graft and the host, with very, very much expectation that that interaction will be leading to the creation of new circuits and new interactions between the graft, the host, and eventually between the host and the endogenous pools of progenitors and stem cells, which is still not fully accepted in the field. There is also another level of expectation from the technologies, because with such sophisticated methods and tools in hands, we are in theory able to use cells which are able to recapitulate tissue-specific behaviors. They are available in embryonic and adult tissues, but they also are editable. So we can apply very sophisticated next-generation cell editing technology to make a good use of the intrinsic but also engineered properties of stem cells. They're also reprogrammable to pluripotency, they are expandable, and they are also multi or variably potent in vitro and in vivo. The challenges for developing consistent protocols of cell therapies in neuroscience are here listed. So ideally, we need to have cells which can be collected from sources which are easy to access. They need to show consistent stability in vitro upon expansion. They need to be safe and feasible. They need to have a druggable mode of action. They have to be easy to manufacture and deliver. And ideally, they also need to be traceable in vivo after injection. So these are big challenges, none of which has been yet fully addressed, which are a major driver for significant progress in the field. So in principle, putting all this together, when applying cellular therapeutics to regenerative neurology, we have three major strategies to keep in mind. Strategy one would be that of rescuing as many endogenous progenitors as possible around those areas of the brain which needs to be fixed. Strategy number two might be that of transplanting exogenous cells able to survive, to migrate, and to instruct specific behaviors in vivo. Strategy number three, which is very early stage in science, but very attractive, is that of applying the most sophisticated principles of cellular reprogramming in vivo to reprogram post-mitotic cells into the given cell of interest directly in vivo. And which are the outcomes of this? So I'm going to give you two case studies, one of which is on one extreme of the medal, and the other one is on the other extreme of the medal, which I think are very informative. The first case study I want to share with you today is the case study for Parkinson's disease. And as you can see from this timeline, everything started very early in the days, in beginning of 80s, when by combining different technologies and approaches, the fantastic observation was that of having successful preclinical evidence that functional delivery of catecholamines or survival of fetal ventral mesencephalon graft was achievable in animal disease models. And then there is lots of other findings, some of which are green because they are exciting, they have allowed significant and productive progress of the field, but there are also some failures in this very crowded timeline that I'm sharing with you. And just to make your comprehension easier, 1987 is the first successful human fetal ventral mesencephalon transplantation in human with PD. There is also some post-mortem evidence of long-term survival of cellular grafts coming a few years after. And then there is also 2014, significant evidence of very long-term survival of cellular grafts in Parkinson. However, there were also a couple of studies which were a little bit disappointing based on the initial hypothesis. And if we concentrate on the major findings, it is important to keep in mind that if you apply cellular therapies in people with Parkinson, you can collect very significant evidence of long-lasting functional benefits, long-lasting survival of transplanted cells, which can also be imaged by PET imaging. More than 15 years of survival. However, and we go back to 28, in a few cases, what started appearing was a very interesting concept biologically, that the host is not behaving properly, is not reacting properly to the instruction delivered by the cellular graft, and instead is delivering horizontally some dysfunctional signals to the cellular graft, which leads to the very innovative concept of host to graft transfer of disease. And this is the case of these two patients which were analyzed back in 28 after having been transplanted with bilateral ventral mesencephalon cellular graft in the striatum between 11 and 16 years before, starting showing not only a very consistent presence of exogenous cells at the periphery of the graft, but also very worrying appearance of Lewy bodies in alpha-synuclein neurons in the graft, which implies that the host was miscommunicating with the graft and transferring signals of dysfunction. And in the space, in Parkinson, there is lots of discussion, very active discussion nowadays, not only on the type of cell to use, but also on the agreement internationally around the protocols to develop, to characterize and standardize cellular graft, with a very big expectation that induced pluripotent stem cells or human embryonic stem cells will be able to help filling the gap of consensus around the nature, the homogeneity, and the developmental stage of the cellular graft to deliver in vivo. The other case study that I wanted to share with you is MS, which is by far more complex biologically, but also technologically, much more challenging as compared to Parkinson. And in this case, the timeline is a little bit shorter, but still very crowded. I would say that everything started in terms of cell therapies by late 70s, when people at Cambridge, with Bill Blackmore and Robin Franklin, started observing significant survival and functional integration of autologous shunt cells in the acute demyelinated nervous system of rodents. So that was the initial inspiration for the approach to be moved forward. And then a number of other preclinical, exciting evidence as proof of concept studies came over. However, MS is not a very simple disease. It's not a disease which is only characterized by acute demyelination. The MS brain is by far more complex than a focally demyelinated animal model. And in fact, the initial idea of transplanting lineage committed potentially remyelinating cells led to a very early small trial at Yale at the beginning of 2000. Unfortunately, that paper was never published, but nonetheless, 3 patients with MS were biopsied in their peripheral nerve, and they were subjected to autologous transplantation into the lesion of autologous stem cells. However, no evidence of cell survival was ever found. As I said, MS is a very complex disease, so I think there is lots of excitement from more complex advanced therapeutic approaches, preclinically and clinically, and I think we made a significant contribution in my early days in science in Milano by providing compelling evidence that by injecting neural stem cells, which eventually had been previously identified in the adult mammalian brain by Fiona Desch and Arturo Alvarez-Ruilla in mice with different models of disease, including experimental encephalomyelitis, providing evidence of neuroprotection, modulation of immune responses, but also later in the days to identify the first druggable or potential druggable mode of action for stem cell therapeutics. And these studies have significantly moved forward so that we have a couple of clinical trials, which have been started not only in Italy, 2 of them in Italy, and also completed, one at San Raffaele and the other one at San Giovanni Rotondo. So again, very exciting progress with very, very elegant proof of concept studies which have been produced from the beginning to now. This is a very elegant study that Steve Goldman conducted at the University of Rochester, asking whether it was possible to create human myelin in the rodent brain. And using a very elegant iPSC technology and very long protocols to make homogeneous population of OPCs, he was able to demonstrate significant, I would say massive presence of human myelin in the rodent brain affected by myelin dysfunction, proposing the iPSC approach for as a novel cell therapy approach for myelin diseases. On the other hand, and this is again another concept which is coming again, MS is very complex, MS is chronic, it is autodisimmune, it is also highly heterogeneous clinically, but also highly heterogeneous pathologically. And it is a big challenge to develop new protocols which will be able to interfere either with transition to progression or to slow down significantly accumulation of secondary damage in the progressive phase of the disease. Importantly, progressive MS is also characterized by a specific set of cellular behaviors in vivo which might be a good target for cellular therapeutics. Some of these are identifiable in the context of a specific subset of lesions, those which are called chronically active lesions, which are expected, which are proposed to be major drivers of progressive disease and disability. The cellular responses in the MS brain are highly heterogeneous at different levels, and cell senescence and physiological brain aging are acting as a major confounding factors in the understanding of how damage accumulates over time in MS. We are also very lucky to be living unprecedented days in science where very sophisticated, highly informative single-cell analysis are telling us what really is happening in vivo. And the combination of the single-cell analysis is providing the opportunity to investigate cellular and molecular aspects of disease pathobiology, and in the context of MS specifically, interaction between the brain and the immune system at an unprecedented level of details. And specifically in the context of disease, which might be a good target for cell therapies, but which pathobiology is still very, very little understood, the single-cell analysis have allowed us understanding that cellular responses in the brain are highly heterogeneous. Astrocytes are heterogeneous, they are found as disease-associated but not disease-specific. They also show features of aging and neurotoxicity. The same concept applies to OPCs and oligodendrocytes, with a big caveat that there are specific clusters of OPCs and oligodendrocytes that seems to be disappearing over time in the context of progressive MS, both at the level of lesions, but also in the context of the normal appearing white matter. Neurons, again, single-cell analysis are revealing a specific transcriptional signature reminiscent of higher vulnerability of neurons to neurotoxic damage in the context of MS. And microglia, again, we have heard several talks in these 2 days, are highly heterogeneous, disease-associated, and again, predominantly neurotoxic. So the environment where to apply cellular therapeutics is very complex yet challenging. And I think we provided a significant contribution, which I'm summarizing now with a very, very quick movie showing that by delivering both allogenic but also xenogenic neural stem cell grafts systemically or in general by using minimally invasive routes of injection, which can be the peripheral circulation or even the intracerebroventricular route of injection, it is possible to deliver substantial numbers of neural stem cells in the MS or MS-like brain, where stem cells are able to engage into very sophisticated programs for horizontal communication with brain cells, all of which overall lead to reduction or repair of collateral damage by establishing novel pathways of cell-to-cell competition with the brain cells. And the major outcomes of this very sophisticated interplay are a promotion of remyelination, are protection and preservation of axonal function, and also modulation of immune responses. Cells survive in vivo, and it is a beautiful neuron surviving over 30 days post-transplantation in the brain of a mouse with MS-like disease. They also can differentiate if the environment is hospitable for differentiation. In this specific case, you see two green cells which are found in the corpus callosum, very close to areas of demyelination expressing the transcription factor OLIG2, which is reminiscent of an early progression towards the oligodendroglial phase. And if you develop a very provocative experimental model where your host shows a significantly delayed remyelination following damage, which is the OLIG1 knockout mouse that you inject with lysolecithin as a focal model of MS-like disease, thus avoiding competition between the cellular graft in the endogenous pool of malinforming cells, indeed, you show much robust evidence of oligodendroglial potency of the cellular graft. There is also other means of communication with the host. This is a very nice electron microscopy image, immunogold actually, that we took several years ago showing that in vivo transplanted stem cells survive, but also they survive in close vicinity with blood vessels, but also they take physical contacts with endogenous myeloid cells, in this case macrophages or early myeloid cells. And if you go higher magnification, you realize the intimacy of this interaction between the exogenous stem cells and the endogenous macrophages, which implies also creation of cellular contacts and eventually also cellular gap junctions, facilitating this very intimate communication between the two cells. And indeed, this level of interaction with the host immune system leads to a much more exciting discovery that we made a few years ago when we observed that the TCA cycle byproduct succinate, which accumulates into M1-like activated macrophages and is also released extracellularly, and therefore is a major driver of persistent smoldering activation of myeloid cells, is also one of the substrates for which neural stem cells show great avidity. In vitro, in fact, neural stem cells are able to significantly uptake succinate out of the tissue culture media and are able to reprogram M1-like activated macrophages towards an OXPHOS metabolic profiling, which is reminiscent of an anti-inflammatory M2-like phenotype. And in vivo, in fact, if you apply the very same principles to the classical EAE protocol by which we try to reduce clinical deficits by stem cell transplantation, you see that the stem cells which are loss of function for the receptor of the succinate show reduced efficacy in ameliorating clinical deficits and also much lower capacity of reprogramming innate immune responses in the brain and much lower capacity to scavenge succinate out of CSF but not plasma. There is a final concept I wanted to share with you, that in the context of these very sophisticated matters of communication between cells and the environment, we also are aware that cells, any cell in nature is able to release extracellular vesicles and exosomes in the extracellular space. And this is the case also for neural stem cells, which are able to release and produce extracellular vesicles, which are able to traffic functional receptors to perpetuate intercellular signaling. They're also able to behave as a fully independent metabolic units. And in the context of this interplay of communication, Luca Peruzzotti Iametti in the lab started observing very consistently that there is a significant proportion, usually 1 out of 3 particles, belonging to the total EV pool, which is characterized by extracellular mitochondria, which are not only anatomically and structurally intact, they are also surrounded by double membranes, but which are also fully functional. As you can see here, they are fully able to breathe by applying high-resolution respirometry, and they're also able to target myeloid cells, which are very well established to have dysfunctional mitochondria, especially when they acquire M1-like pro-inflammatory phenotypes, and in vivo, by applying EV therapeutics in mice with MS-like disease, we are able to achieve therapeutic effects which are superimposable to those that we have historically observed with parental cell lines. So going back to the end, what is the agenda or the revision of the agenda that we might propose by putting together all these principles of cellular and in a small part of cellular therapeutics, that stem cells, I think we have to agree that they are very flexible, they are plastic, they are not programmable from remote, but they indeed in vivo work as a naturally occurring full combination therapy, able to fluctuate between neuroprotective functions, immunomodulatory functions, cell replacement. And in the verge of clinical trials, so we are very excited that phase 1 feasibility, first-in-man safety studies are getting to the end, but we need efficacy data to set the stage for more ambitious programs applied to cellular therapeutics. Mitochondrial metabolism and regulation of immune cell behavior is a very hot topic. It's an important mechanism or mode of action which might expand across different diseases. Route of delivery needs to address, in some cases, the whole brain behavior, much more than aiming at providing proof of concept evidence of mechanism of action. So eventually considering non-invasive or minimally invasive delivery route like the CSF or the bloodstream might be an option. Delayed chronic disease needs to be discussed very actively to identify new or revised window of opportunities for next generation therapies. Exogenous regeneration, which is what the Parkinson disease showed, is fantastic, but might not last or might not survive to host-to-graft miscommunication, which is difficult to control unless we propose combination therapies. Stem cells from patients, especially when proposing autologous therapeutics, might be dysfunctional, so we need to pay significant attention also to the epigenomic signature and to the functional traits of autologous stem cells. And in perspective, I think some of you or many of you might agree with this, the understanding of the molecular and cellular interaction and basis between the brain and the immune system in general is also a great opportunity and challenge to expand our ability to harness and manipulate the immune system to improve repair and health degeneration. So the final one is to acknowledge the significant contribution of funding bodies and members of my lab, and then I will be very happy to take your question in the end, and thank you very much for your attention.
Thank you, thank you. We shall discuss at the end of the session. So thank you again for this presentation. So it's my pleasure to invite Letizia Leocani to speak about rehabilitation and neuromodulation. Letizia is Associate Professor at the University Vita e Salute San Raffaele, and she has dedicated a lot of research to the integration of visual, cognitive, and motor functions. You have 20 minutes.
Thank you so much. I'm very grateful for being here to share with you some thoughts about what I think, how to use neuromodulation to improve rehabilitation. I have to say immediately I have a bias because I like a lot devices. And I will profit of this time to say something about how I think we can measure neuroplasticity and induce it and improve it with neuromodulation and non-invasive. We know that at the neuronal level, active neurons have more synapses, but at the network level, the things are more complex in the humans. For example, if you compare the cortical activation of a violin player with the amateur violin player, the professional ones have a more concentrated cortical activation, seems to activate less cortex. That's because they are more efficient, so they waste less energies. But there are also ways to measure plasticity not only during activity, but at rest, exploring the organization of motor networks, even in the primary motor cortex. And that's what we did, for example, with magnetic stimulation, where you can map the motor cortex and find examples of what we call plasticity after playing the piano for many years compared with no non-piano players that have less organized cortex with imbalance towards the dominant hand. But is this real plasticity, or is it that the piano players are gifted and they have already a well-organized hand motor cortex, and so that's why they choose to play the piano? So this is not the way to go and study plasticity. We have to study the induction of plasticity and changes that we see in front of our eyes when they happen. And so we organized the experiments with poor students studying the piano for a couple of weeks, with the thesis student going to their houses to teach them the piano, and actually using another method, which is EEG and not fMRI or magnetic stimulation. We showed a similar thing, that you can induce plasticity and better organization of the hand motor activation, which is more concentrated, if you did learn to play a task and if you did improve, and so your motor cortex becomes more efficient. But these were, physiotherapy students, very young. Is it possible to induce plasticity in older people? Because we're talking about neurodegenerative diseases, and most of them, at least the ones I deal with, are in older age. These are examples that could be misleading because they are very high performers, over 90 years old in their different fields. And I'm sure in this room there will be future brain athletes when they will be 90, but there is a bias here, of course, a selection bias, and these people have started very early to learn what they are doing, and they kept doing it. But can we learn new things when we are already old? We have the premises. At autopsies, there are new neurons forming even after we are 90, if we get there. And super agers have larger neurons in the hippocampus and less neurofibrillary tangles. Super agers are defined because they can remember after 10 minutes like more than 9-10 words out of 15, which normal elderly reach to about 6 words, and their neurons are better. So, but is this that their neurons are better because they use them better, or just that they have a good memory because they were born with a better destiny of their neurons? So again, autopsies are not the answer. But it was possible to induce plasticity, even measured with MRI, which is not, of course, with the sensitivity of histology. But people over 65 could learn very difficult motor skills, of course, with less percentage of success compared with under 25. So that's a very good news. But the bad news is that if we don't keep practicing, we lose it. So these changes were reversible after 3 months of stopping. So plasticity is nice, but it's also not eternal. So we need to keep doing what we want to keep efficient. And so this has an impact, for example, if we stop moving, immobilization is a bad thing, and we will see some examples at the end. But how can we improve plasticity besides training and training? Can we train less and make more efforts to learn something and to retain it? There are many devices that are being developed and under study with this purpose, although they were not born for that purpose. And among these, I will briefly touch the transcranial direct current simulation, which has the advantage of being portable, although the energies involved are quite lower compared with transcranial magnetic stimulation, which is a heavy device, needs medical supervision, but has a stronger energy, so it could be more promising in some circumstances. And about TDCS, so the electric currents, we have the possibility to increase the excitability of the motor cortex. This has been demonstrated in humans, showing the increased amplitude of motor evoked potentials after a single session. Of course, this could be replicated and lead to increased mechanisms of plasticity besides the short-term increased excitability. So the top part of the graph is the one I will be focusing on, because that's the part with the increased excitability that is more related to improved learning while the bottom part is the use of the cathodal currents instead of the anodal, which has inhibitory function, and it's more relevant, for example, if we want to reduce the neuronal activity. But I will not touch this because this is relevant, for example, in ALS, and Professor Di Lazzaro will touch this issue. But going to the application of this method to neurodegenerative diseases, if we pick Alzheimer's disease, we have a few studies with variable results, and we have to say that the sample sizes are small and the techniques are small, and also the duration of the studies are small. So no wonder why the effect size is small and doesn't last the follow-up, which is about 2 or 3 months. So maybe it's more promising to use a more powerful device like a repetitive stimulation, which has the same possibility to increase excitability, but has already been proven, and in the US it is reimbursed, to treat drug-resistant major depression. So it had FDA approval several years ago, so maybe that works better. Actually, again, the applications in Alzheimer's disease are quite variable. And again, we have small sample sizes, but it is also a direction that we can take as long as we can standardize and find the funding to have the sizes needed for approval and reimbursement. But a small size effect at meta-analysis is there. But we have a long way to work towards really making it available and meaningful to people. But how can we potentiate the effects of RTMS, for example, which is the tool that I will focus from here on? For example, if we want to target a degenerative disease like Alzheimer's or Parkinson's, we cannot focus on the dorsolateral prefrontal cortex that we did for major depression. Plus, we have neural networks that are degenerating, so we must be able to reach the widespread network that is involved in that particular disease. So I came across the possibility to use the H-coil, which is a derivation of the classical focal coil we use for depression, that has the possibility, it's an NIH patented device that has the possibility to change the shape of the magnetic field that I found more promising for widespread diseases like Parkinson's disease or Alzheimer's disease. But actually, I was a bit skeptical to use a helmet to improve cognition or extrapyramidal symptoms in people. So before running a clinical trial, I wanted at least to try some biological effect, because of course a helmet will have a huge placebo effect. So I'm really grateful for the help that we received by Daniela Perani, who agreed to perform at least a PET scan on a patient with aphasia and hypometabolism on the Broca area, that after a month of treatment or using a helmet like this, more concentrated on the Broca area, because we tilted the device, we could show that there was improvement in a network, but that included the Broca region. So we were more confident in spending time and effort with no funds on performing a clinical trial on 60 people with Parkinson's disease, with a helmet designed to stimulate both bilateral motor cortexes and prefrontal cortex. And we found a significant improvement in motor function and in tremor, which is more difficult to improve with placebo, that had no effect at the apparent stimulation, making the same noise of the coil, but with no magnetic field on the motor areas. And then we performed a similar trial 3 times a week for a month in people with Alzheimer's disease. And so after a month of these stimulations, we found a significant improvement of the ADAS-Cog that we saw is used also in a more expensive and sample size relevant clinical trials. And that was significant, although it was only 30 people. And so that was a good news. We could publish the work, but I'm not happy with this work, because after 2 months, the people that received the real treatment actually were at the same level of the ones who had received the sham. So I think we should not work to publish papers and show a p-value, but to do something that is useful for people, and something that is lasting 1 month, I don't think is useful, so we need to work more. How can we work more and improve it? Maybe we can couple this with training. Of course, we have to speak about rehabilitation. Why don't we couple training, rehabilitation with this magnetic stimulation? And actually, we found evidence, a suggestion that we should do that, because we could publish a paper in stroke, not a degenerative disease. This is just an example. And we found, of course, a significant effect by doing stimulation of the lower limb affected by the stroke compared with apparent stimulation. But that was only significant, but it wasn't meaningful, because only in 3 out of 10 people we could reach a difference that was clinically relevant. But if we couple the stimulation with the simple cycling, which of course is not a natural rehabilitation, but as I said, we had not specific funding for that study. So this cycling was what we had. We could double the success with the percentage of patients that had a clinically relevant effect. So I think that is the way to go, to couple this type of intervention with rehabilitation. And so the following trial that we performed, and we had the replication funded by the Italian MS Society, was in people with multiple sclerosis undergoing rehabilitation as inpatients, and who did improve significantly better compared with people doing receiving the sham stimulation in walking speed, walking endurance, which is the 6 minutes walk, and also in measures of spasticity at the clinical examination. So again, there was an issue with the reduction of a number of patients at follow-up, and this is also an issue. We need methods to avoid having the patients coming back to the clinic every time to get visits after they are discharged from the hospital. So I will talk about this in a minute. But we can also deal with the issue of not only the safety that we heard about this morning, but also about the dose. So it's not enough to find methods to couple activity and stimulation. We need to know how long the treatment has to be. And so this is just an example in the motor domain with the real piano player, professional one, with double lesion in the spinal cord affecting both hands, and the left more than the right. And she agreed to undergo a training with piano playing and magnetic stimulation. If we had stopped here after 5 sessions, which is the classical that you find in most TDCS studies, we would have found a significant effect, for example, for the right hand, but nothing much for the left. Maybe it was going to be significant, but who cares about publishing something significant if we miss the best part, which is that the patient is going to improve and reach a plateau. I think we should reach the level of no further improvement before we stop with the treatment, and not the p-value, because patients are not interested in our p-values. And so I think we should focus on that. And but maybe we are dedicating our efforts maybe to people who are not susceptible of improving. And we heard earlier that even in children, there is a point of no return after which it doesn't make much sense to apply gene therapy. But do we know all the points of no return of our patients in other conditions, like in neurodegenerative disease, in dementia? We can define it clinically, for example, in people with just mild cognitive impairment or mild Alzheimer's disease. The treatment with repetitive stimulation did last longer than in my study. So maybe in our study, we just included people beyond the point of no return that pilots know very well, and we should learn that. Also in the trajectory of our patients, we need tools to define it. And maybe in the motor domain, as in the example from our patients with our people with MS, we found that if we apply the real treatment together with rehabilitation in people with MS, only the ones who have still motor evoked potentials to the lower limbs are the ones who are going to improve their motor speed, while the ones with the exhausted corticospinal reserve did not improve. They behaved like people who undergo a sham. So I think we need to find markers also, not only of monitoring the worsening, but also to find the point of no return. And also, when we deal with rehabilitation in neuromodulation, we need also markers of plasticity and see whether our patient has a sufficient plasticity reserve. And in the case of Alzheimer's disease, there is the plasticity is compromised. For example, in the excitatory motor evoked potentials TMS, the amplitude of evoked potentials is not showing the similar increase that is showing in control subjects. And also, in Italy, it has been demonstrated that the level of plasticity that you can explore with magnetic stimulation in Alzheimer's disease is also proportional to the level of tau that you find in the CSF. So there is a biological limitation to plasticity in some conditions. But how can we identify people that can really benefit most, but also that are at risk of worsening, so they need earlier interventions? We can consider using also remote interventions to overcome logistic barriers. Think about COVID-19. And there is already the availability of a supervised TDCS, and there are many applications to perform remote rehabilitation. But I would like to focus on the possibility of performing remote monitoring. That's also because we are actively working on the landscape analysis together with the PROMS initiative. With Lisbeth Peters and Robert Hyde, we're performing a survey, a landscape analysis on what is available in multiple sclerosis to be able to discuss with people with MS what is really relevant for them and what we can improve. And I will show you one example of a study, IMI European-funded study, using a digital app and the Fitbit to monitor people with MS. But this is just to show you the impact of environmental factors on mobility of people. And when we were running the study, the COVID-19 came, and this is the Milan cohort of people with MS with the Fitbit on. And this is February 21, when patient 1 was found in Codogno. And we can find immediately the number of steps going down, even before we had the lockdown, because people were scared. And this comes from Google Mobility Maps. I was in lockdown myself, although I had to go to the hospital, and I studied Google Maps that were free for everyone to check. And we can see a sign of panic, people running to the store on February 21 in Italy, but also in Denmark. We have the Denmark cohort with the Fitbit, people with MS with the Fitbit, but it did not reduce the number of steps. Only people in Milan did. Why? I guess that if we combine the data, our data, with the Google Maps, we can see that we were forbidden to go to parks. So we were stuck in the house. People working in the hospital had to go and go back to the house. We could only walk the dog, but we could not go to the park. And people in Denmark could go to the park. So this is not disease-related, and this is not scientifically related or device-related. This is politically related. And so I think we need to discuss on the availability of mobility possibilities. So not only gene therapy is not available to everyone, but also the possibility to move is not available to everyone. And in this survey by Gianpaolo Brichetto from the Italian MS Society, together with the Rehab in MS network, we found that most centers for rehab were closed due to COVID. So really we need accessibility, and we need the possibility also to prevent according to our single needs. As people going into space, they know they have to work in advance on their muscles and bones. We should know in advance what to work on, because I think we want to live a better life and not to provide a better brain for autopsies. So with this, I thank very much all the people I work with and the Italian MS Society, my major source of funds. Thank you.
Thank you. Thank you very much, Letizia. So we are in the discussion session. So I wish to invite Professor Vincenzo Di Lazzaro to speak about brain stimulation in ALS. Vincenzo Di Lazzaro is Professor of Neurology at the Campus Biomedico University in Rome. So Vincenzo, I suppose you have 10 minutes, but you will see that on the screen, I think.
Okay. First of all, I want
you have 7. Okay, sorry.
To thank Professor Comi and La Fondazione Prada for the invitation, for this very kind invitation. I will briefly present the results of preliminary studies using brain stimulation in ALS. The brain stimulation has already been introduced by Letizia, but I just want to very briefly show the basis of the most popular and promising technique of stimulation, that is transcranial magnetic stimulation. When you deliver this magnetic stimulus to the brain, oh, sorry, how can I go back? Okay. When you deliver the stimulus to the brain, no, I'm not back. Okay, a single magnetic stimulus to the brain, you can record the muscle response from the contralateral side of the body, but what you can obtain in the brain is much more complex, because a single stimulus can evoke this high-frequency repetitive discharge of corticospinal axons. This has been demonstrated in patients with epidural electrode, in whom it was possible to record directly the corticospinal output. Thus, with this technique, we are activating cortical projections. We are activating both the excitatory and inhibitory interneurons, and depending on the intensity of stimulation, you can selectively activate, for instance, the inhibitory neurons. Thus, we are activating the synapses within the motor cortex. And thus, by delivering repetitive stimulation, you can modulate the excitability of the human motor cortex, inducing long-term potentiation, long-term depression, depending on the frequency of stimulation, the intensity of stimulation, and pattern of stimulation. And here we have 2 examples. This is a protocol of simulation that is termed continuous theta bar simulation, that is similar to the protocol used in brain slices that induces long-term depression. In green, you have the high-frequency discharge evoked in baseline condition in gray, and in green is the output recorded after delivering this protocol simulation. It is evident that we have a suppression in neural transmission in the motor cortex. The opposite is observed using high-frequency stimulation. Here in red, and in black you have the baseline excitability. Now, in ALS, we have an abnormal cortical excitability to transcranial magnetic stimulation because we have an enhanced excitability and hyperexcitability that can contribute to neurodegeneration. It may be the expression of this glutamatergic excitotoxicity. This is due to the impairment of both the inhibitory interneurons and an enhanced excitability of the corticospinal cells. Thus, it has been suggested that we can use repetitive stimulation to rebalance the excitability of the motor cortex, hoping to reduce in some way the disease progression. And we have several changes that can be induced by repetitive stimulation. We not only have the induction of long-term depression or long-term potentiation that is due to changes in calcium dynamics, we can also have some long-lasting changes due to the induction of genes. For instance, we have the induction of brain-derived neurotrophic factor, and if we can enhance the expression of this gene, we might have a protective effect in ALS patients. This has been shown for the first time in this study. Rats were stimulated with high-frequency stimulation, and after several days of stimulation at high frequency, we have an enhancement of BDNF expression. Thus, we have 2 possibilities to use protocols of simulation that suppress cortical excitability to reduce the hyperexcitability. And on the other hand, we have the possibility to use high-frequency stimulation to enhance BDNF expression, to have a neuroprotective effect. Starting from 2004, several studies have been performed, small studies, proof of principle studies, mainly from my group, evaluating these two approaches. This is the first study, a proof of principle study. We have only 4 patients. 2 were treated with low-frequency stimulation, here, and 2 with high-frequency stimulation. Because we only had 4 patients, we compared disease progression before the beginning of simulation, represented by this red dashed line, with that observed after the beginning of simulation, assuming a linear progression of the disease. Well, in the patients treated with low-frequency stimulation, there is a very slight tendency to reduce the progression. But what is very clear is that in patients treated with high frequency, we have an enhanced progression of disease. And for those patients, the stimulation was interrupted after a couple of months, whereas these patients were treated for several months. But what could be the reason for this enhanced progression observed in these patients treated with high-frequency stimulation? We made a parallel study evaluating the effects of high-frequency stimulation in ALS patients. We evaluated BDNF plasma levels. We are not sure that this will reflect the level of BDNF in the brain, but indeed we observed that there is a decrease, not an increase of BDNF in ALS patients during repetitive stimulation. But the reason for this is provided by studies performed in experimental studies performed several years later. And those studies evaluating the effects of repetitive stimulation on brain slices of the mouse showed that high-frequency repetitive stimulation can increase the glutamatergic A1 subunits of the AMPA receptors. So we have more AMPA receptors, and also it can interfere with GABAergic transmission, reducing the cluster size of gefferin, that is the protein that is associated, the structural protein that scaffolding protein for the GABA A receptors. Thus, it seems that with this approach, we have an enhancement of glutamatergic transmission, a reduction of GABAergic transmission, that is the pathological finding in ALS. Thus, this might not be useful in ALS patients. Thus, we decided only to evaluate the effect of protocols of stimulation suppressing cortical excitability. This is one of the first studies. We used continuous theta bar stimulation and evaluated the effects in 20 patients with ALS, 10 patients treated with sham stimulation, 10 patients with active stimulation. Again, we have a slight reduction in disease progression. Those patients were treated for 5 days a week, for 1 week a month. Then we decided to double the dose of repetitive stimulation, and in another group of patients, we performed 2 weeks of stimulation per month for 6 months. And we observed a more pronounced effect. 1 week seems to be slightly effective, and 2 weeks seems to be more effective than 1 week. It's just a very limited effect, but it's quite convincing. Now, the idea is to increase the dose, but the problem with the repetitive stimulation, magnetic stimulation, is that it is performed in hospital, and it's very difficult for the patients, for ALS patients, to come to the hospital every day. So we wondered whether a more chronic form of stimulation could be very effective. So we decided to perform a proof of principle study in one patient with ALS by implanting an epidural motor cortex electrode, and in this patient it was possible to perform daily stimulation at a low frequency. This is the disease progression here in gray before the beginning of treatment. Then we have 22 months of simulation here, this continuous black line. And we observed no effect at all after 22 months of simulation. The dashed line represents the disease progression, assuming again a linear progression of disease. But after more months of simulation, we observed a dramatic change in disease progression. After 9 years, the patient was still alive. So this suggests that a more chronic form of simulation might be effective. But this approach is invasive. Actually, this approach was evaluated in subsequent years in animals. This is a case of reverse translation. It was shown in this animal model of disease that bilateral motor cortex simulation can increase survival period. So this could be a promising approach, but it's invasive. And on the other side, we have the possibility to treat with non-invasive magnetic simulation. So we were in search of different methods of simulation capable of performing chronic motor cortex simulation non-invasively, ideally at home. And a few years ago, a new technique of simulation became available that is static magnetic simulation. This is a very simple technique. If you have a very powerful magnet and this magnet is positioned over the motor cortex, you will have a reduced excitability of the motor cortex after 20 minutes. It's an effect that is less pronounced than that produced by repetitive magnetic simulation, but it is there. Thus, we performed a proof of principle study in 2 patients with ALS. Again, we compared the disease progression before the beginning of TSMS with that observed after the beginning of simulation. Patients underwent at home static magnetic simulation for 3 times a day for 120 minutes every day. And again, here we have the possible progression of disease, assuming a linear progression of the disease. And also we have the progression of disease in a control population that is from the PROACT database that includes placebo patients of many trials. And in both patients, we observed a consistent reduction in disease progression after TSMS. This patient is still alive. He had a very aggressive form of ALS after several years of simulation. Thus, it seems extremely promising, and we decided, together with Vincenzo Silani, to start a study recruiting 40 patients with ALS, 20 patients with real, and 20 with sham stimulation. We have just concluded the study, and we are analyzing the data, and we hope that it could be successful as well.
Thank you. Thank you for this contribution. So the last presentation is by Agal Bergman, who is from Israel, and exactly you work at the Edmond Elili Safra Center at the Hebrew University, right? Yes. So welcome you. I think yours is the only presentation on DBS.
I'm afraid.
So yes. So it's important to have this contribution as well.
Okay, so thank you all, thank you, organizer. I'm aware that I'm one before the last one, so I'll try to make it shorter. This is the title, Deep Brain Stimulation in Parkinson's Disease. And this is an old story, but I would like to speak with you about the past, present, and future. This is my disclosure that can I have the pointer? Yes. So this is my disclosure. I'm working with Alpha Omega. It is not related. We are developing a navigation tool for deep brain stimulation procedure. It is not related to the subject of today. Parkinson's disease. Again, a very short introduction. Parkinson's disease is common among the elderly, about more than 1% of those that are older than 60 years old. It is characterized by motor symptoms, akinesia, rigidity, muscle stiffness, tremor, and postural and gait deficits, but as well by emotional, cognitive, and sleep deficit. Remember, the sleep deficit will come to it. The motor symptoms of Parkinson's disease follow the generation of the dopamine neuron. So you can see over here, this is in the healthy monkey, the tyrosine hydroxylase of the substantia nigra pars compacta. And after MPTP, you see that there is a big loss of dopaminergic neuron that lead to depletion of dopamine in the striatum. This is the normal striatum, and this is after MPTP. And we see very similar picture in the human theater, but in the monkey, we can record the electrical activity before MPTP, before dopamine depletion, and after. This is 3 electrodes in the globus pallidus. Each line is a spike action potential, normal monkey, and you see that after MPTP and dopamine depletion, there is increasing the activity in the subthalamic nucleus and globus pallidus, and moreover, this activity starts to display synchronous oscillation at the beta range between 13 to 30 Hertz. Therapy first line for Parkinson is, of course, we know now, it is dopamine replacement therapy. It was mentioned by Per, but after 5-10 years of treatment, most of our patients will develop the complication of the therapy, on-off fluctuation, and levodopa-induced dyskinesia. And therefore, when the MPTP monkey model was discovered in 1985, we started to work on it and to compare the physiology of the basal ganglia before and after dopamine depletion. To make a long story short, what we found is that there is hyperactivity and this synchronous beta oscillation in the subthalamic nucleus, and if we inactivate the subthalamic nucleus, either by muscimol or ibotenic acid, we can ameliorate all symptoms of Parkinson's disease. So now, actually 3 years later, this finding was moved to the DBS theater. We know today that high-frequency continuous stimulation of the subthalamic nucleus is very effective. Since 1993, we have almost 300,000 patients operated all over the world in many, many centers, mainly for Parkinson's disease, but also for dystonia and so on. The mechanism is still debated, but to make a long story short, I would claim that if we think about our central nervous system, the main goal of the central system is to be a state-to-action loop. You have your current state, you hear me, you see me, and your nervous system is taking this information and decide what is the next action. And then you do action on the world, and this loop is working all the time. But there are several networks. One of them is the basal ganglia, the automatic system, system 1 of Daniel Kahneman. And this system is abnormal in Parkinson's disease, and therefore our patient is losing his automatic repertoire. There is a second system, the cortical system, or Danny Kahneman system 2. This is the thoughtful, the one that you are aware of it, but because the basal ganglia are abnormal, we don't able, or our patients are unable to move the control to the cortical network. What we do with deep brain stimulation or with ablation, we inactivate the basal ganglia, and therefore we move the control to the cortical system, and our patients are able to have better behavior in the world. However, deep brain stimulation, unlike ablation, is reversible and adjustable. So this is very good, and this is what we do today. But where we are going, and already now, and actually more than 10 years ago, we've shown that, again, in the MPTP monkey, it is not that we have abnormal oscillation of all the time. Sometimes the activity is normal, and sometimes the activity is abnormal. And therefore, we tested if we can do closed-loop deep brain stimulation, namely to detect when the basal ganglia are abnormal, and to inactivate the basal ganglia only when they don't behave. And when they are doing their job well, to let them do their job. So this was shown over here, and actually the main bottom line is that the basal ganglia can be observed and can be controlled. We now know to recognize better when the basal ganglia don't behave by longer episode of beta and lower frequency, but these are detailed. The basal ganglia can be observed, can be controlled. Already Medtronic are coming out with their Percept device that enable closed loop DBS. What about the future? The future is even better, because right now we discovered that with deep brain stimulation, if we are looking at the specific phase of the oscillation, if we stimulate at the upgoing phase, we increase the oscillation. And if we stimulate at the downgoing phase, we suppress the oscillation. So with deep brain stimulation, we can not only mimic inactivation, we can suppress the bad guy and augment the good guy. And actually, what I do believe that the future will be circadian frequency and phase-specific closed-loop DBS. And the idea, again, is to say that if sleep is indeed the brain method to clean the waste product that we accumulate over the day, and sleep disorder initiate and aggravate brain disorders. So, for example, we have sleep disorder, there is decreased clearance of amyloid beta, there is increase in amyloid beta, we get Alzheimer, and we get more sleep disorder. But we can speak about the same about alpha-synuclein, exactly the same story. And we know that in Parkinson's, sleep disorders start 10 years before we have the motor symptom, REM sleep behavior disorder. So phase-specific closed-loop DBS that will stimulate at the upgoing phase will increase our slow oscillation, delta sleep, delta oscillation, restore slow wave sleep, and will suppress the beta, which actually persists even during night and related to our insomnia. Moreover, today we have machine learning hierarchical organization, so we can use the subthalamic nucleus neuronal activities that we mentioned. We can use smartphone and telemetry to assess the circadian rhythm and the Parkinson's symptoms. And of course, what is really important for us is the patient evaluation of his quality of life, because we don't like to repeat the situations that the neurologist is happy and the patient is not. And actually, I do believe that if we'll be able to treat the sleep disorder and to restore normal and good quality sleep, we may slow down, we actually will use the sleep mechanism, and therefore we slow down the progression of Parkinson's disease and maybe of other neurodegenerative disease. So this is one before last slide. DBS is highly effective therapy, and unlike pharma, is local, and this is the main advantage, and unlike ablation, it is reversible and adjustable. The past was, or actually what we are doing, the patient is coming every 6 months to the clinic and we adjust this parameter. Right now we are already switching to closed loop DBS that are working continuously and inactivate the basal ganglia only when the basal ganglia don't activate. And I believe that in the future we'll have machine learning, circadian, and frequency and phase specific that will enable us to augment the good oscillation and to suppress the bad oscillation, restore normal sleep, and use the mechanism of sleep to clean the brain waste up. And no risk, no gain. And I'm telling it not to our patient know it. I'm telling you to you neurologists, because I'm sure that also in Italy, like in my place, neurologists are not happy to send their patient to deep brain stimulation. Don't wait too much. We have good treatment. Give your patient good quality of life, because today deep brain stimulation is established therapy and we can help our patient. And this is the last one, okay, because again, we are very close to the cocktail. This is by Mahmoud Darwish, a Palestinian poet, that says that if you were told you are going to die here this evening, and I'm leaving it to to read, but I will go directly to the end. I will prepare my last lunch. I pour out wine into 2 glasses for me and for the one who will come unannounced. Then I will take a siesta between 2 dreams. So I wish all of us a good wine and a lot of Shana Tova, Happy New Year, Happy Jewish New Year, and a good beta, gamma, and delta oscillation in your awake and asleep basal ganglia.
Thank you. So come here. We don't have wine on the table, but please, I ask the speakers to come for a quick round of discussion. Please take a seat here. Let me just provoke you. I follow the first patients implanted with fetal grafts in Sweden. A long time has gone by and not much has happened, while in the DBS came later and in a very short while was developed. So probably because the DBS is working on the existing wiring, whereas grafts wanted to make a new wiring, and this made things complicated. So I don't know if that is true or wrong, but please, we make a tour of the table on the differences between physiology and grafting or cellular interventions. It seems that physiology is quicker, more efficient, at least, easier to... Okay, you go.
Yeah, can I? Because for me, the major problem with stem cell therapy at that time was not the host versus graft. It was the fact that the graft was working very, very good, and eventually most of the patients develop levodopa or dopa-induced dyskinesia because the graft was working so good and actually create too much dopamine. And I think that DBS is much more modest, okay? We are not trying to correct in current DBS. We are just actually saying we inactivate the abnormal basal ganglia and let the cortex, as we tell our patient, raise your hand, look forward and move, okay? We're moving to the cortical network, and therefore DBS was over.
Let's hear the cellular people. How do they comment to you?
I actually was trying to be very simplistic. There is evidence, and there is strong evidence of. I was trying to be simplistic. What you said is right. There is also evidence of excessive amount of neurotransmitters provided by the graft. But I think that we are very close to each other because cell therapy is actually trying to optimize the biology and the physiology in a completely different context. What we are missing is a complete understanding and complete capacity to manipulate those type of interactions remotely without intervening on a patient. But you're right. But there was also evidence of progressive loss of control by the graft when compared to the host, which is also physiological because there is spread of disease and there is communication.
Just to say that some of my friends are trying to combine now deep breath stimulation and stem cell therapy.
That's something I was going to say because it's also preclinical evidence of combination therapy. Yep. But of course, the tight control of how DBS works on the biology of the cells is not easy to control, but that's possibly the next level.
Other comments on this point? Yes, please.
No, to me, the major limitation of these functional interventions, which are fine, is that we are dealing now with global generalized CNS diseases. So we are able to correct something that is very relevant for the patient, no doubt about that, but is not targeting the disease outside where we produce the stimulation or the injection of cellular therapy. So I see this like a classical combination, as I said before. It could be a good way to combine, but not to target. And having said that, my question to Nadine, in fact, is you mentioned that there is the possibility to modulate quite extensively the microglia, which has been in these 2 days the protagonist in some way of the scene. Are you or is anyone of the groups in the area of gene therapy trying to follow this way, so to try to go and to modulate with the gene therapy microglia?
No, I think that's a very good point. And in fact, the potential, of course, of targeting microglia through a stem cell-based approach is to have really a widespread delivery, and so potentially impacting disseminated disease rather than locally. And targeting a very relevant factor in the disease mechanism, which I think, as you mentioned, has been happening to be more and more involved in whatever mechanism this is. So I know that there are several groups who are looking at this, whether to replace microglia with modified progenitors which either deliver, again, biotherapeutic, which are immunomodulatory, or preventing, essentially regressing a sort of an inflammatory state by this approach. So I think the challenge, of course, these are treatments which are quite invasive, because it's an autologous transplantation which requires conditioning, which is per se harsh, and that's what has been also at the moment constraining the application of stem cell therapy to very severe diseases. If we could eventually go into, and there's a lot of effort to improve conditioning approach to make much less harsh and dangerous, this application would be, I think, much more doable and feasible. However, the question will be, are we still replacing the microglia now? Because if we lessen conditioning, some of those biological approaches which are undertaken are not probably reaching the brain. So these are dual challenges. But I think on the other end, the platform of modulating microglia by gene or by replacement is very relevant and is being investigated.
If I may add a couple of comments. I think what we have agreed on is that microglia is a cellular target, and then technology is available to, first of all, understand what is the dynamics of the behavior of microglia under homeostatic or steady state conditions and in the context of disease, and then with a high level of sophistication technology we have, which goes from monoclonals to small molecules to gene therapy. I think there is good opportunity to be able to fine-tune specific cellular behavior in vivo in a way that would be protective for the course of the disease.
I have a question for Bergman. I'm fascinated by the idea of restoring sleep in early phases of PD, other neurodegenerative disorders, but the question is that you have to choose a different target, cannot be the subthalamic nucleus, and thus I wonder whether it will be more useful to have non-invasive stimulation, to try to develop more effective ways of producing the stimulation, activating the deep structures so you can change your target more easily.
So of course I agree. If you can avoid surgery, avoid surgery. Okay, no question about it. Okay, it is. We go to surgery. Actually, it was written only when pharma reaches limit, when optimal therapy, optimal dopamine therapy by a good neurologist that you trust, tell you that, and still the patient does not get the quality of life that he's looking for. Whether the subthalamic nucleus is the optimal target, from my point of view, and I don't have the time to elaborate about it, the subthalamic nucleus is the driving force of the basal ganglia, and the basal ganglia is the driving force of the brain. but of course I'm overkilling, okay? And there is a very, the nice feature of the subthalamic nucleus is that it is small in compact nucleus, and therefore we can really affect it and get a huge effect on the brain without too much side effect if you are on target. I completely agree that in the future, okay, when we'll develop some kind of this deep TMS that will go up to the subthalamic nucleus, it will enable us to do it in non-invasive way, and moreover, to test other possible targets. We have to do it.
Can I go? The panel is ready. Sorry, it was, yes?
Can I ask a question on the same topic, yes. Hagai? I think there is something missing in this sleep story. Sleep is part of a plasticity cycle. The beautiful work of Giulio Tononi and his group, and also myself, about the slow wave activity being local and being the result of a healthy, hefty LTP during the wake period. So this LTP drives slow wave activity. So in Parkinson's disease, we know that there is a big problem in terms of plasticity, both from a neurophysiological evidence and also from behavioral evidence. And also, there are studies that we have done showing that RTMS, after learning, that was deficient in Parkinson's disease, if you test it 5 minutes later, you don't have any effective learning. So you do RTMS local, specific for the type of learning, and the day after, they are retaining the skill, and the sleep is normalized. So the whole story of the slow wave activity in Parkinson's disease should start from the point of view that there is an impaired LTP formation. And there are a lot of studies showing that, both neurophysiological and behavioral. So also there are studies showing that in the early phase of Parkinson's disease, slow wave activity is reduced, and levodopa therapy doesn't do anything.
Oh, thank you for this comment. I will leave to Letizia, maybe to, the table is ready for a couple of reflections, yes?
Thank you. Molecular therapies, whether gene, cell, immune therapies, target the cause of the disease, and of course they are very relevant, and our therapies target networks and are not very specific and cannot be proposed to prevent a specific disease. However, we can consider that treating symptoms is not irrelevant, because when I said there is a point of no return, I was talking about inclusion in a clinical trial, but I was not considering the individual patient considered in a holistic manner, as Paola Zaratin likes to consider a person with a disease. And so in a single individual, there is always a symptom that we could target with the non-invasive stimulation, and so give a hope to different stages of the disease while we wait for the molecular therapies to save us from a disease. Second, we could build a better reserve by considering these devices also for healthy learning.
Oh, yes. Not, yes, thank you. So I think we now have to close, so I leave the podium to Giancarlo, who is here for the final session. Thank you very much.
Okay, so to detect PA, which I think this is a beautiful place. Qualcosa stava parlando.
Giancarlo, a te. Very good. Okay, we are almost at the end of an heavy day. Unfortunately, by definition, conclusions come at the end of the story. So we have this session just with the aim to try to review a bit what happened in these 2 days, and also to listen to the voice of the other stakeholders, patients associations, industry, and I think it is very important to have also such a contribution. Spent 2 days listening to the academic people. Unfortunately, my co-chairman, that I have to thank so much, because Pierluigi Nicotra has been very instrumental in organizing these 2 days. Unfortunately, he tried to do what we recommended to do in this today, to do physical exercise, but unfortunately he exceeded, and during a tennis game he broke the tendon, Achilles tendon. So, and I don't know if it's available. I think he should be with us. In
Yes,
any
I'm here.
case, Pierluigi, I hope today is better. How are you?
Yes, I think as you say, I mean, physical exercise should not be overdone, and of course, contrary to the general wisdom, to me it caused a problem, but fortunately not as severe as I thought. But yes, with a lesion that stopped me from flying. So my apologies to all the others for having missed the conference that I was very much looking forward to attend. And from what Giancarlo says, it seems like has been a lively conference, and I've been listening to the latest debate. So I basically will, if you wish, Giancarlo, make some few considerations in general, given the theme of the roundtable. Is that okay?
Please do.
So, a few things which probably, and I apologize in advance if these things have been already discussed during the conference. The first thing, I mean, why don't we have effective treatments? I think that's a key question. And of course, the answers to these questions are several, are multiple. In the first place, I think we have underestimated the diversity of disease. Do we have one type of Alzheimer's disease? Do we have one type of any of the other neurodegenerative diseases? Probably not. We have subsets of diseases, and therefore patient stratification is an essential thing to be successful in clinical trials. Unfortunately, the regulatory agencies still demand, especially for dementias, the inclusion of a large variety of patients who are not necessarily stratified. I think we can learn from the recent announcement from ASI and Biogen about the lecanemab, where they approach this problem by stratifying patients in phase 2 and by having therefore a very, very good subset of patients, and then finally showing efficacy in cognition and in slowing down the progression of cognition in Alzheimer patients in phase 3 studies, which is a very positive message to the entire community. So the first thing it is we should try to work together with the regulatory agencies to be able to improve the design and the outcome requested for clinical trials. I think that's one reason why we haven't yet found effective treatments in many conditions. The second one is that these are systemic diseases. We cannot think that we will find one drug to cure them all. We're not in the Lord of the Ring kind of business, so there is not going to be one drug to cure them all. There's going to be a set of drugs. There's going to be polytherapy. And for polytherapy, we have to try to understand the mechanisms much better than we do now. We are working actively in my institute on the immunological side, on trying to find agents which can work on the immune system and be used in combination with agents which lower pathological proteins, for example. So that's another approach. Of course, combinatorial treatments will be successful in the moment in which there is one treatment which at least shows efficacy. And I think the recent results with lecanemab for Alzheimer's disease may be very, very encouraging to follow that direction. The third thing is the way that this type of research is funded. I can speak for Germany, but I think this is a general problem. If we look at the European level, there is very little funding for studying neurodegenerative diseases. Whereas the United States has increased dramatically the funding for research on neurodegenerative diseases from the year 2013, 2014 until now. The funding for neurodegenerative diseases has increased steadily and significantly. In other countries in Europe, this has not happened. In Germany, the German Center for Neurodegenerative Diseases, which I direct, is the main organization that deals with the research on neurodegenerative diseases. Being an extra university organization, we have a number of advantages. The first one is that we can perform directly clinical trials. We can sponsor clinical trials. We can work collectively in collaboration with a number of universities. And we have the same kind of platform everywhere in our sites. So we can do multi-center studies with stratified patient cohorts. We can develop our own drugs in collaboration with industry coming from our own research. And therefore, we have a continuum from the fundamental research, to the study of potential interventions, and also to population studies and the understanding of risk. So in my view, I think a concerted efforts of research organizations in Europe would be essential to develop new treatments for neurodegenerative diseases. And I think that's a goal, but of course, this demands an increased amount of funding. Of course, in these times, asking for more money seems to be outrageous. But look what has happened with other conditions. So look the amount of money which has been invested in cancer, in HIV research, in other conditions. I keep saying that, of course, we had the disaster of the corona. We don't know yet whether actually this will lead, as many are suspecting, to an increased number of cases of dementia or other neurodegenerative diseases. It may be the case. Nevertheless, with corona, we have a clear example of how a substantial amount of money has been invested. Of course, this is an infectious disease. It's easy to find the therapy because we know the target. But at the same time, I think this has created a generalized effort into the scientific community, which has been extremely important. So the same type of effort can be done for neurodegenerative diseases. And the private-public partnership is one of the ways by which this can be achieved, by investment in both sectors. And finally, sharing data. That's another thing which is extremely important. We have been at the forefront of swarm learning, of trying to be able to share data without moving the data from where they are. There is an interesting article from a consortium which we have led with Joachim Schulze, which was published in Nature last year, and shows one way by which we can share data without necessarily moving the data, without necessarily having access from everybody to the data, but just using artificial intelligence and software, which are basically present on the cloud, that can actually elaborate this data very fast and sharing them without breaking the European GDPR, or without basically having any problem in sharing data with all the other partners of the consortium. And with this, I would like to close. I think these are the main points that I would say probably could guide a better way to be able to generate new medicine, new treatments for neurodegenerative diseases. Thank you.
Thank you so much, Pierluigi, for this very, very clear overview of some of the key aspects that you have to take into consideration. What I would like to do with this roundtable is to ask now each of the members of the roundtable just to come here when I call them, and just to particularly try to approach two aspects. One is which is the critical point they see in terms of future evolution, and particularly concerning the guests, particularly from the point of view of the association they represent. And second, to give a direct advice to the Prada Foundation Human Brains about what to do in the future. So, and I would like to start with a fundamental component that is the patient voice. And here the patient voice is particularly coming from the Patients Association. And I start asking Tim Cozzi to come. Tim has, I have to say, has a very unique point of view, because he has been the director of the Fast Forward organization of the National MS Society, in charge to try to bring ahead new potential treatments in the early phase, and to try to bring a bit such an important achievement. And then he also has been responsible and coordinator of the founding activities of the National MS Society, and more recently the soul and the key person, let me say that, of the Progressive MS Alliance, which has been already a tentative to combine together different stakeholders. So, Tim.
Thank you, Giancarlo. I'll try to live up to the introduction. Thank you, everyone, for your attention. I know it's late in the day. Since I have 4 minutes, I'm going to keep my remarks quite brief to 3 what I'd say key points in my mind. First, this has been an amazing conference, and when I saw that I had 4 minutes, I wondered what can I say in 4 minutes that has not already been said, and so I'll focus instead on some of the points you made, Giancarlo. So from the frame of MS, which is what I will bring it, but I would offer that the points I want to make, I think, will span across all of the diseases. The first is, it begins, and I think we must, as for a global campaign, come back to the patients. These are some pictures of individuals living with multiple sclerosis, and I think you will see within this collage the range of the disease, from young people to individuals who've lived with MS for many decades, who now live with consequences of the disease that was unmanaged because there were no disease-modifying treatments at the time. And I would start with any campaign begins and ends with the perspectives of people living with whatever the disease is, and we must keep it focused on that, and also understand, as this picture illustrates, the range of the disease and how it impacts the disease. Those who are diagnosed today will have far different experiences than those who have lived with it for 15 years, but we need to be faithful to both and not treat it as an either-or, although our health systems will treat it as either-or. I was reflecting on this conference, and then it occurred to me that if we had had this conversation in 1992, 40 years ago, the number 0 would have been the number of disease-modifying agents we would have been talking about. Now, fortunately, a year later, we had beta-seron, but today we have 20-plus agents in multiple sclerosis. And what that says to me is that we are able to see significant change. The work with aducanumab opens the door for Alzheimer's disease, and I would submit that the story of multiple sclerosis gives me hope that across brain diseases we can have transformational change. It requires, in my view, a commitment to 3 things, and I would offer to the Prada Foundation that the ability to have a global campaign rests on 3 elements that are interconnected, and they are, if I can get it to go, I think fundamentally, the green, we must continue to understand disease mechanisms. The progress we are seeing is because of the fundamental investment in disease science. And while we need to drive towards more therapies, the fundamental understanding of disease underlies all of this. So we must sustain that. We must also, as part of a campaign, I believe, foster scientific and technology innovation. And I separate those because we do need the intellectual drivers, but the technology innovation that has given us the rapid ability to test NFL from blood-based biomarkers require technology innovation in addition to scientific innovation. Without both, we will not get the progress we need. Finally, I think the final point I'll come back to, we must engage the community. I took Dr. Bateman's comment today around the indication of, could somebody balance a checkbook as an important part of their life. I don't think the FDA will ever put in a label that this is approved for the effective balancing of a checkbook in its label. But for people with the disease, those things, those moments matter greatly. And while they're not clinically measured, they are fundamental to living life. And I believe that we must continue to engage the community, both to have clinical impact, but also what I would describe as life impact. And that even modest, a 15% improvement in quality of life is 15% I wouldn't have. One has to balance the benefit and risk of all of these things, but I would submit that we must continue to engage the community and treat all of these as a virtuous cycle that work together to give us acceleration for disease. So with that, I thank you for your attention, and thank you, Giancarlo, for this opportunity.
Thank you so much, team, for your suggestions that I will take care of, contribute to our final action. I would like to ask now Mario Battaglia. Mario Battaglia is the president of Fondazione Italiana Sclerosi Multipla, that is the scientific arm of the Patients Association. And Mario has been for a long time president of the association, and he is also involved in many international activities of patients advocacy, and very recently he has a clear role on patient-reported outcomes initiative. So many aspects. Also for you, many different point of view. Which are your recommendations?
Thank you, Giancarlo. I try to complement what Tim Kotz has already said, because I'm speaking of MS again, with a few comments. First of all, some suggestion mixed the two comments. What we do as MS Foundation is, of course, not only fund research, but I think that our foundations, our societies should also promote and coordinate as we do. Of course, it means that the patients, along with the scientific side, will work together. We have to foster a research that aims to promote a social impact. And this has already been said today and yesterday. And we have to engage people affected, and also citizens. And we have to advocate. This is the main issue. I want to go to a point that is the brain health that was touched yesterday, because I think that, of course, we have been speaking of brain health for all neurodegenerative diseases. We moved in a way in the years from tertiary prevention to secondary prevention, and yesterday we were speaking about primary prevention. But this means to speak of prodromal phase is difficult in the sense that it has a profound impact on personal, social, and economic side. And this should be considered because moving from the research that is testing, demonstrating, going to early diagnosis, and then coming to advocate for early treatment is not an easy issue. Today it was used in a comment in a discussion the word feasible, affordable, and this means a lot. We have been speaking about patients, and I think that engagement of patients is a main issue. Listen to patient. The comments that Cummings and Montalban gave us today were really important. But what was underlined is that we need that the patient can contribute with a scientific methodology. Otherwise, this is not. So the PROs that were quoted is a topic that is important. And this is important in an era where we have the data coming in, or that can come in 24 hours, 365 days. And this means big data, so the management of big data, the linking to the registries that we have already, the problems that were just quoted by Nicotera regarding the protection of the data. And so we have to consider all this issue. Of course, the main issue in our opinion, is a shared agenda. A shared agenda among us, I mean neurodegenerative diseases, associations and professional societies. We need to have a common vision, common strategies, and as we did in MS, as was already quoted by Giancarlo and Tim, we can share also resources, and this is quite important in a way where we can approach everything in a multi-stakeholder approach. And we need a global approach, and finally, we need to involve institutions, public institutions. We need to do it at international level, as was said, speaking about a global campaign, at national level. We need international plans, national plans, and we need to focus also on the public funding, as it was said today. And as Nicotera just said, using the words that I had planned, we need a synergy between private and public, between us all together. If I can stop with a comment, I have been listening all this time of COVID, after COVID, we can say. We need a new normal. I think we need a new future, and the societies, the patient societies and scientific societies can construct the new future.
Thank you very much, Mario. Thank you. Thank you. We move now to another association I call John Hare, who is the President and Chief Executive Officer of the U.S. Foundation. And even, I think, with a very long-lasting experience also in other areas of health. So you had learned a lot, and then you transfer in this new era your knowledge and expertise.
Yeah, thank you very much. Well, it's a pleasure to be here, and I just want to thank everyone for welcoming us. I put together some slides, because I'm just going to kind of cut through them, because I think most of you know clearly what Parkinson's is and its different symptoms. And then I'll just pause here briefly just to note that in the United States, or in the world, we think there's somewhere around 10 million individuals who are living with Parkinson's. That's probably an undercount. It's mostly a disease of the elderly, but 5% of people living with the disease have young onset, which may actually be somewhat of a different disease. More men than women get the disease, and we are about to publish a new study in the United States that shows that we had been saying for a really long time that about 60,000 individuals are newly diagnosed each year. Our new study is showing that it's somewhere between 90 and 120,000, so it's considerably greater than we thought before. Parkinson's Foundation is a US-based foundation, but we truly are global. I'll talk a little bit about that. We have a number of mission areas: research, care, patient education, and patient training. In our research mission, I just want to focus on a couple of things. And then the underlying theme of my talk really is just about collaboration and how important it is. We fund a lot of basic research with academic medical center researchers all over the world. That's just part of what we do. $6 or $7 million a year to that. One of the things that we have launched within the last couple of years is a genetic study. So we are now seeking to enroll in a trial called PD Generation, 15,000 individuals with Parkinson's, testing them for genetic mutations and providing counseling and then returning the results. We've already tested 6,000 individuals over the last couple of years, so we are starting to accumulate a lot of people who have GBA, LRRK2. Those are the two main ones, obviously, and then Parkin and a couple others. But it's an amazing study that's really just starting to accelerate. And there's lots of opportunities for us to partner and collaborate with industry and many others. We have just within the last 2 months launched a partnership with Parkinson's UK, an equivalent organization size and mission in Great Britain. They have built a platform to do drug discovery and development. They've been at it for a number of years now. They funded 11 targets. They've evaluated 23 targets, funded 11, looking both at symptom control and disease modifying. So we are now going to join that effort, bringing a lot of the resources that we have available to us, charitable resources in the United States, to that effort. We've been working on an outcomes project for many years. It's a very large study. There's a huge Italian cohort in it because of the Fresco Foundation, so we're pleased to continue that work. But I just want to really focus on some of these big scale initiatives that we're trying to put in place at the Parkinson's Foundation. We have a very significant care mission, obviously. We have 51 centers of excellence all around the world. 14 of those 51 centers are not in the United States. We have just launched a whole new subset of care centers called comprehensive care centers, because we're trying to make sure that we understand what's going on in the community. We've been really very focused on academic medical centers. This is a way to reach out and see what's happening in the community through large neurology practices and primary care, where the vast majority of people in the United States are cared for. We've got a number of other, the other one to note is that we have a network of excellence, which is the Fresco network here in Italy. So there are 6 centers in Italy that participate that, and a lot of really great collaboration. You'll hear from Lice Ghilardi about that later. And then we do a number of other things in patient outcomes. I'm just going to move very quickly. We have a whole series of programs on patient and care partner, education, professional training, and community education. But the things I want to talk about really are our primary goal has to be to change the course of Parkinson's disease. And that's going to really require an investment of resources for scientists, basic research, drug discovery and development, and the type of work we're doing with on PD Generation, which is really building an infrastructure so that the more patients we know with genetic mutations, the better we're going to be able to enroll them in clinical trials. And I do believe very sincerely that this really will take a global initiative, global partnerships. We at the Parkinson's Foundation, we're a $50 million foundation. We've doubled our size in the last 6 years, and we're very proud of that. But $50 million for a disease that affects 10 million people worldwide is not enough. So the final point I'll make is, once in a previous life, I worked in cystic fibrosis, which in the United States is about 30,000 individuals. And on an annual basis, we were raising, $200 million a year for that very small disease. If you look at all the money that we raise privately for Parkinson's, not industry, not government, it's less than about $400 million. I think the market is underperforming in terms of resources that we can bring to Parkinson's, Alzheimer's, MS. There's so much ability to raise money. I just think we're one of those where the neurological diseases sort of fly under the radar screen. It's generally speaking a disease of older people. So we need to do a lot more in terms of going out and raising funds and getting our message out there. Thank you very much.
Thank you so much for all these quite good suggestions. We move now to another society, and it is an Alzheimer society, and I call George Vandenberg, who serves different roles. He's co-chair of the Davos Alzheimer's Collaboration, is the chairman of and co-founder of the US Against Alzheimer organization, and many other activities. So we are really listening to you. What is your suggestion and which are your opinions about?
Well, thank you very much, Professor Comi. Thank you very much for having me and all of us in this extraordinary setting and this extraordinary session. So I am I'm not a doctor, I'm not a researcher, I am a businessman. But driven to this field by 3 generations lost to Alzheimer's over 40 years. And if we don't pick up the pace, I know my children and my grandchildren are going to die, and a lot of people are going to find their children and grandchildren passing away. So I am as passionate as any researcher that you ought to succeed. Now, in the United States, it was said that we've been successful in raising money. And indeed, I just want to give you a quick, because I'm involved deeply into politics in the United States. One of the goals was to raise money for Alzheimer's research in the United States. It's gone from $400 million a year to $3.7 billion a year. Over the last 8 years, close to $17 billion invested in this disease. Now, that needs to be sustained, but it needs to be matched by Europe. It needs to be matched by Asia. The resources in this field need to be sustained over, like cancer, 50 years. And so we need to continue to use our political will and our political efforts to drive it. This is Nicotera's, Pierluigi's point. I think I chair the Davos Alzheimer's Collaborative. Just a brief word on what it is. It is a partnership between industry and the World Economic Forum, designed to address at the Gavi and CEPI global level what we have to do to take on, and this is Alzheimer's, but we can extend the platform, an infrastructure around the world capable of responding to this disease in the same fashion that we've responded to many other diseases. This is a depiction, you've seen this before, of the distribution on the left of where we've done genetic research. And the column on the right, the distribution of people of the world. So 80% of the genetic research has been done in white Europeans or European descent, that is Americans mostly, and the global population of this disease is distributed quite dramatically outside of that field. So I don't think the global response to this effort is anywhere near the right distribution. So let me, I'm going to offer 3 ideas. The first, we have to take into account, as we think about this disease, the continuum of global collaboration. It is research. Disease understanding is essential to drive all of this, and it is each one of these chains. It is important to get regulators into the conversation, as Pierluigi suggested. I suggested it's vitally important to get payers in the conversation. Our government insurer in the United States has already said it's not going to cover lecanemab. So we can be very excited about that drug, and we patients are extraordinarily excited, and we'll be very noisy if our Medicare system does not cover that drug. But its current posture is it's not covering that drug. Did not cover the Biogen drug, the aducanumab drug, not covering this drug, not going to cover gantenerumab, and it's not going to cover donanemab. It has already made that decision. So how do you reverse that decision? It's a political action effort. So I'm suggesting that we need to do this entire chain and think about this entire chain of value creation, because what your research in actually getting to us patients has to take account of all those steps. I'd also say brain health is part of that proposition too. Number two, we as a field must include and effectively collaborate across governments, across countries, in order to achieve this. No one government is going to solve this problem. No one sector is going to solve this problem. And so we need, as a high resource nation, to assure that the infrastructure we build to solve this problem solves it for all the patients, not just the patients in Europe and not just the patients in the United States, but patients across the world. And so, third point, we've got to collaborate across all sectors. Governments can't solve it, researchers can't solve it, patients can't solve it, industry can't solve it. We can't do it alone. This is going to be a team sport. And so we've got to get on the team. We've got to the point just made earlier, figure out what our plan is and what our plan of action is. So what are we doing at the Davos Alzheimer's Collaborative? We have funded pilots now in 19 healthcare systems around the world, from Armenia to Brunei, to Brazil, to Jamaica, to the United States, to Japan. Why? Basically to drive them to change their business models to be able to detect and diagnose this disease at the primary care level. Guess what? There are not a lot of PET scans in Armenia. There are not a lot of PET scans, quite frankly, in many countries of the world. We're not going to be able to do this with existing technology. So faster technologies, digital cognitive assessments, and blood, which is obviously genetics and biomarkers. We are recruiting over 20 existing longitudinal cohorts of size around the world that now represent 130,000 participants. We'll be doing this up to 1 million participants over the coming years to add research measures relevant to Alzheimer's disease into those cohorts so that we can take what they already know about diabetes, what they already know about heart, and then add Alzheimer's measures and cognitive measures to what their work, aggregate that data or cause it to be aggregated through the cloud and through a new AD workbench tool for data sharing with the Gates Ventures Alzheimer's Disease Data Initiative, and then redistribute that, allow that access to that 1 million person cohort with all its diversity and all its biomarker and genetic data and all its digital cognitive data, and make that available to all the researchers around the world. Intellectual capacity is not distributed 90% to Europe and the United States. 90% of that intellectual capacity around the world could be applied to this disease as long as we build the capacity to allow the rest of the world to have access to that data, a data sharing point that Pierluigi made again. And finally, the clinical trial support mechanism. We have 90 trial sites in the United States. We have 30 now in Europe. We're now moving into Kenya and to low and middle income resource countries, hopefully with different drugs. My guess is that in 10 years, there will not be one monoclonal antibody actually infused in low and middle income countries. You need the equipment, you need the cost. It's just imperative that we get lower cost drugs, lower cost clinical trial systems around the world. So we're trying to build this infrastructure that includes the rest of the world and makes a real difference. Now, we're focused on Alzheimer's, but as I mentioned, because these existing cohorts already have measures in a lot of other diseases, we think it can become a platform that's much broader than Alzheimer's and it's going to include brain health. We're announcing in the next couple of days a partnership with Worldwide Fingers to include clinical trials in risk reduction coupled with metformin and other existing diseases. So they're low cost, available to low and middle income countries. So we need to work together. We need to work globally. We need to think about how to include low and middle income countries, not as an afterthought as we did with COVID. So thank you very much, Pierre.
Thank you very much. I think that your attention to extend to all the countries this intervention is really a fundamental aspect. They are not so represented here, but the aims was to have, for sure, and we have, contribution from Asia, but we have to extend to Africa and other countries. That's very important. Thank you so much. Now we would like to ask Alice Ghilardi to come. Alice Ghilardi has a bit a double role. He is a person who connects this group with the next group because at the same time she has some responsibility in the Fresco Foundation, which has been mentioned sometime by John just a few minutes ago. But at the same time, he is a professor of neurology, deeply involved in research in Parkinson and not only in Parkinson diseases. So, Richard, what is your suggestion?
The cure is not yet here. We heard it. Overall in Parkinson, I'm talking about Parkinson disease. However, the increasing research and knowledge that we heard today and yesterday about Parkinson disease has not been transformed into effective therapy, but it can inform something else beside the cure. The care of a patient. And so that's what the Fresco Foundation is really involved in, improving the care, the quality of life of patient with an integrated, multifaceted approach. Here we have three means to do that, promoting physical recovery. We heard Letizia Leocani speaking about it with different things. We are focusing on visual motor recovery because we think vision and motor are important, and also cognitive recovery, emotional well-being, because Parkinson's disease is everything. But this can apply also to Alzheimer's disease. Alzheimer's disease is not just a cognitive disease. It has also a motor and emotional component that are very important. And overall, all these diseases, from multiple sclerosis to Parkinson to Alzheimer, are related to deficit in plasticity that are altered. And that is an important evidence. And all this type of approach can improve plasticity. Physical recovery, the rehabilitation since the 40s has been introduced as the only therapy besides surgery in Parkinson's disease because levodopa was not yet introduced. However, the results were really, really scarce and bad. So only recently, rehabilitation has been reintroduced in Parkinson's disease in the last 10-15 years with introduction of aerobic component that promotes plasticity, and also, as Letizia said last night, with fun component. Music therapy, yoga, tai chi, whatever the patient likes to do. And also cognitive techniques are important. And the other thing is that aerobic component not just increase the physical recovery, but also the cognitive recovery. There are a lot of papers on that. And also they enhance sleep. Sleep is really improved by aerobic exercise. But there is a need for specialized personnel that can perform this rehabilitation strategy. And we are working with the Parkinson Foundation to do that. Visual motor recovery, we are using all visual art, from art making, art observation, and overall art therapy. This is artistic creation with psychotherapy, and we are working with NYU and other groups. And we found, we have pilot data already published, that there is an enhancement of the visual spatial exploration, visual motor integration. And again, cognitive recovery and emotional well-being are used to exercise to improve executive function, to to remove the social stigma, psychological support, and also social engagement are very important. Besides that, we have needs. We have needs for patient care centered. And so, as I said before, there is a need for specialized, dedicated professional. So we are engaged in training and education, not just a professional, but also patient. Patient has to be empowered. They have to know what we are doing, and they have to inform us, and we have to listen to them. So active engagement of a patient, and overall we have to focus, and our foundation is together with the Parkinson Foundation is focused on caregivers and family education. There are studies showing that the family, the caregivers of patients with neurodegenerative diseases are at higher risk to develop neurodegenerative diseases themselves because of the chronic stress that they are due to. So these are the list of the different activity in which the Fresco Foundation is engaged. We are partnering with Parkinson Foundation, facilitating the activity in the Italian Network of Excellence for Integrated Care of Parkinson's Disease. We are participating now for 7 years of the POP, the Parkinson Outcome Project, and also to the PD generation study that we have been already started. So we are education program professional, patient, caregivers. We have RFAs for fellowship, clinical research fellowship for young people. Young people are not interested in anymore in Parkinson's disease. We want to attract them there. And we have clinical research grant. And also from a scientific point of view, we are doing every 2 years a symposium on plasticity, because we think that plasticity is one of the key that has not been yet explored for the care, not just for the care, but also for the cure of neurodegenerative disease.
Thank you so much. Thank you so much, Lice, because I think you touch a point. We of course, we need to look at the future, the new strategies, new therapy, but not to forget the present and the quality of the medical center from one side, and particularly the area of I don't like the definition of neurorehabilitation. I prefer just to how to prevent and how to try to deal with the problem of dysfunction. But this is a very key point. Okay, so now I would like to ask Katrin Lubeski. We have now some person who has a direct responsibility in driving big research institutions, and they have at the same time, of course, the scientific background, but from the other side, the concrete necessity to have things moving and to try to facilitate that. So Katrin is the medical director of the Brain Institute in Paris, but also involved in many organizations, ARCEP, that is the French Society of MS, and more recently a very important role in the Progressive MS Alliance. Katrin?
Thank you so much, Giancarlo. So difficult to speak, it's a difficult exercise. Many things have been said, and I am rather at the end. But to accelerate therapy, I think that I will just give a few simple ideas. I will be short. First of all, cross-fertilization between different diseases and different pathophysiology is crucial. It's fruitful. We have seen it during these 2 days. It's really inspiring, and I think this is key to try to advance and find new ideas and new pathways. So we have been discussing about different diseases: Alzheimer's disease, multiple sclerosis, Parkinson, Huntington disease, ALS. They all have in common to be neurodegenerative disease. MS have also an inflammatory component, but if you get removed of the relapse, it's a neurodegenerative disease. And what is common to these different diseases? There are many things that are common, but one is aging. Aging is playing a key role in the pathomechanism. And the other key mechanism is the role of microglia. And I think that microglia, and we heard so many talks about microglia today and the day before, and I think to accelerate therapy, it would be good to have better treatment to target microglia. BTK inhibitors are coming, but we don't know yet the results. And I think this might be an interesting perspective. Still an unmet need. So then a few ideas I will quote. And this has been said already, but development of blood biomarkers, of course, is crucial. And for that, we need to collaborate, to have big network and all the program that have been said and what you are doing also, Giancarlo, at the Progressive MS Alliance and the European Charcot Foundation is crucial because we have to work together and to share the data, first thing. The second thing, this has been said probably even not now but before, the importance of the cohorts. The cohorts are crucial to understand the development of the disease, and we have to focus on highly characterized cohorts with multimodal evaluation, genetic, biology, imaging, clinic. And I think that we have to focus, and we heard of it today and yesterday, on pre-symptomatic cohort. I think this will be so important to better understand and to better treat and to better prevent. And for that, we have the pre-prodromal phase of MS, which is radiologically isolated syndrome. We have prodromal Parkinson disease, we have prodromal Alzheimer's disease, but also we have the pre-symptomatic phase of the genetic disease, which are very, very important to understand. Third point, we need, this was alluded also, we need novel trial design. For example, if we want to work on brain repair, the trial design that we have at the moment don't have the good outcomes and not the good trial design. We have to find new outcomes and maybe to try to go to the multi-arm trials with the adaptative design, and I think this would be important to go fast. And in this context, and I think this hasn't been said, I think it would be, or maybe, yes, one of you said this, it would be important to go to the repurposed drugs, because those drugs have been used in other indications. We know that they are safe. We know already something on their mechanism, so it's very important. And there have been a paper some years ago with Marco Salvetti on the importance of repurposed drugs. And the last point, before my very short final conclusion, is that, and you alluded to this, it's important to have ways to support the very early phase. You know, when there is a target that is identified in a lab, it's very difficult to go to the proof of concept study because we cannot go to a pharma because it's not yet a drug. We don't know if it's going to go bad or well. So we need some platform and financial support for these proof of concept studies. Finally, and then I will have finished, so it's crucial to, and at the Paris Brain Institute we are doing that, to keep the very tight links between the clinic and the research, clinical research, but also basic research. And I think this is absolutely crucial for innovation, disruptive innovation, as we said. And this bench-to-bedside approach is very, very important. And finally, and this is my last point, I think it is important to attract the next generation, the new generation of clinician scientists. And for that, we have to find ways. At the Paris Brain Institute, we have specific call to try to attract this next generation, but they are also from other institutions. So I think this is important. We have to attract them, but also to train them and to have the next generation of the clinician scientists. And with that, I will only finish by thanking you so much, Giancarlo. These 2 days have been absolutely inspiring. Thank you very much.
Thank you. Thank you so much for all the advices. And I would like now to ask if we can be connected with Professor Alan Thompson. Alan, here you are. Hello, we miss you here in these 2 days. Professor Alan Thompson is really in a perfect condition to provide us some suggestion because he is at the same time Dean of the UCL Faculty of Brain Science and Chair of the Neuroscience Program and quite deep involvement as Chair of the Progressive MS Alliance until a few months ago. So, and many other aspects. So really, Alan, which are your suggestions about the things to do?
Giancarlo, thank you very much. And apologies for not being present, but I have really enjoyed some excellent lectures over the last 2 days. I had presumed that at this point there'd be recurring themes, and that is certainly the case. So I'm just going to make a couple of general points drawn from the Progressive Alliance and also from the Dementia Research Institute in the UK. And these have all been made before. I just want to make 4 points about how we might go forward to the next level, if you like. The first, there are 4 points. The first is collaboration. That's been mentioned over and over again. And this is collaboration across specialities, clinical research, etc., but also across sectors, as we've heard, universities, hospitals, regulators, all of those affected by the neurological conditions. Across countries, this has to be a global initiative. And although we always aspire to that, actually achieving it is really quite difficult. And across conditions. And if nothing, these last 2 days have demonstrated such commonality across these neurodegenerative conditions, and particularly the point that Catherine just made, that they all have a prodromal phase where there's pathological change but no clinical manifestation, which is a great opportunity, but also a huge challenge. So collaboration is the first point. The second for me is focus, to be really clear about what we are trying to achieve, a clarity of vision, and a vision which stimulates, encourages, and engages everybody. And third, and I don't know if we've mentioned this very much, is leadership. So I think if you're going to create something worldwide and really drive this area forward, leadership is critical, effective, collegiate, ensuring delivery. So that's key, and that will resonate with you very well, Giancarlo, and with Pierluigi. And the last one for me is enabling. And that, of course, is really code for funding. We've heard about public and private partnerships. We've seen a huge discrepancy between funding of this area compared to many others, including cancer. And so getting secure funding is really critical. And again, going back to the Alliance, I think the 2 areas where such funding has been particularly effective is in stimulating blue skies research and drawing people outside the field into the area, and secondly, these international networks, which really bring the world together around a very specific point and then deliver it. So in simple terms, and it was Steve Hauser who talked about a visionary global initiative with effective leadership, very secure funding, and a clear focus. That's what we need to move forward. Thank you very much.
Thank you so much, Alan. As always, very inspired. And I would like to ask now to Alessandro Di Rocco. Alessandro Di Rocco is professor of neurology at the Zucker School of Medicine, but at the same time he's having a key role on the movement disorder, and particularly on Parkinson's disease. And I would like Alessandro to ask your opinion and suggestions.
Well, first of all, thank you, Giancarlo. And I start re-emphasizing what has been said by many people here. I really didn't think that I would learn so much by listening to colleagues talking about multiple sclerosis, dementia, ALS, and other diseases that are not Parkinson. And we all tend to be confined to our own little world. And I think this idea of cross-fertilization, across specialties, and across also expertise is really important. What I hope to add on is just a little bit of a perspective on kind of I'm foremost a clinician. So what I sort of bring here is a little bit the other element, what Dr. Nicotera was emphasizing, what are we doing really? Why have we not been able to develop effective treatments? And while, Elena Morrow yesterday talked about being galvanized, and a lot of the speeches were galvanized, we're very excited about, some really exciting things happening in research. And while it's, today also was very exciting, talking about 90 trials that are active going on for Parkinson's disease alone. There are hundreds of failed trials. And the tension between this excitement, this galvanizing, and going back to the clinic, where there is really nothing new. that has happened in the last few decades, except perhaps, you know, a different awareness, so kind of an ecological awareness. So we're much more aware how important exercise, sleep, well-being, caregiving, and so on is, and probably this has made a big difference. But in reality, not much is different. Not only that, but if you look at the United States, the data are really discomforting. So as you may know, and this is may not have reached a lot of our years in Europe, life expectancy in the United States is decreasing, not increasing. That's before COVID. And this is I think one of the most sobering studies that has been. I'm sorry to be, you know, the downer through this excitement, but the life expectancy and the age-adjusted mortality in Parkinson's disease point to a very dim scenario. So age-adjusted mortality in Parkinson's disease has increased from 1999 to 2019. And this is regarding of age, sex, race, ethnicity, conditional living, geographic location, and so on. This is database on Medicare data, so the largest available public database that we have. And not talking about COVID, I know there's a little time, and this has been touched upon by many colleagues about how, you know, we all expect there's going to be some additional effect. And I think in clinic, maybe you're already sort of dripping down some of this effect. I think we're already seeing that. And this is a study from Richard Main and Pete Schmidt, which was largely discussed about how, you know, this is experimental data on mice, how COVID can be a predisposing factor in developing Parkinson's. So one sort of note that sort of connects me also to some of the other speakers here is that on one hand, I think one important data is that the 73 World Health, WHO World Health Organization, in its assembly in 2019, defined Parkinson as one of the healthcare priorities for the first time ever, and created a committee. I was privileged and humbled by being in that committee. And that committee sort of identified two key elements. One is that this is in line with some of the epidemiological studies which have been really conducted in the United States and all over the world. For whatever reasons, the number of cases in Parkinson's disease are increasing. And the number of deaths measured in 195 countries through a variety of ways shows that there is this pretty steep increase. And at the same time, the daily, which is a standard measure of disabilities, the disability-adjusted life years, has been increasing worldwide. And this is sort of a call of action which resembles what Mr. Brandenburg was mentioning. So I won't go for lack of time, but you know, this idea that we need to target the world in different ways by increasing, by looking, by understanding more through epidemiological data, awareness, looking at whatever preventable risk reduction. Pesticides in Parkinson's is one example, and work on access to diagnosis, treatment, caregiver support, and so on. And I'll conclude saying that, you know, was this one of the awareness of this meeting is that while we're working with this group in Geneva, I had no idea that a few miles away in Davos, there were others doing some similar work. And Davos and Geneva are not that far. And I guarantee you, nobody in Geneva has any idea at the WHO of what you're doing in Davos. So I thank you again, Giancarlo, because it's through this cross-fertilization that I think we can do important things. Thank you.
Thank you very, very much. We need now to connect with Frederick Destrebeck. I think he should be connected, I can expect.
Yes, I'm here.
Are you there?
Yes, I'm here, Professor
Oh,
Comi.
hello, hi.
Hello.
Good evening. Good evening. We see you very well, and here we are discussing about future steps and critical points and how to fill in some gaps. And we are here just to listen to your point of view. Frederick Destrebeck is the executive director of the European Brain Council. And I think you are there for 6 or 7 years now, or even more.
Eight. Today is my anniversary.
Yes. So a long time in order then to become really confident with all these type of problems. Friedrich, please.
Thank you. Thank you very much, Professor Comi, and thank you to the Prada Foundation for inviting me to present on behalf of EBC. And a word of apology for not being able to be with you, but as you can hear, I'm struggling with a bad flu that prevented me from traveling to Milan as much as I would have loved to. I'm actually skipping a few slides of my introduction to avoid any duplication with what was already said, and maybe go immediately to the core focus of EBC's action. Just a word about the organization. We are a membership-based organization active in Brussels to promote brain research, brain innovation, and brain health. And in doing so, we bring together the voice of patient organizations, clinical and scientific societies, as well as the private sector and industry, in order to speak with one voice in developing our agenda. On the next slide, you have a vision of the latest policy document that we adopted that was entitled Fostering Innovation and Improving Outcomes, and was really trying to make sense of all the various initiatives and policy issues which we would need to prioritize and target in order to create the change that we are advocating for. And a few of these were already mentioned, and it goes, of course, by the increase of support and increase of funding into brain research, but also strengthening partnership with the industry, but also establishing compelling narrative and economic studies to demonstrate the case for brain disorders. Next slide, please. And in doing so, EBC conducted a study that demonstrated the cost of inaction, the cost of non-treatment into brain diseases. So we conducted an economic study that demonstrated the cost efficiency of improving outcomes for patients living with a brain condition. And I would like to refer you to our website for more information. Next slide, please. Next again. On the basis of this study, we established another narrative which was built out of the testimonies of key opinion leaders and experts by experience, but also clinicians and researchers. So we did this story of this storytelling in rethinking the management of brain disorders. Next slide. Into the field of multiple sclerosis. And all thanks to Professor Battaglia and Professor Zaratin for having contributed to this study. On the next slide, you see that we have expanded this in the field of migraine, and this is still being released and published in the context of the migraine awareness month and weeks that we have just gone through in the recent days. And next slide. And we have recently launched a similar exercise on rethinking the treatment pathway in Alzheimer's disease. And you are all warmly invited to follow us on social media. We will have a webinar in January 2023 to release the outcome of this exercise that we are running now with FPR. Next slide, please. But the topic of my talk today is really this project that is coming to an end now in October, but where we have tried to integrate all the major initiatives that are active in supporting brain research at the European level. So you are all familiar with the JPND, the Human Brain Project, and the ERANET Neuron. And actually, we were tasked as EBC to coordinate this project where we joined forces in order to avoid fragmentation, but also build a common agenda. And this common agenda was really meant to feed into the future brain health partnership that is going to be launched by 2025. And this partnership is really meant to integrate those large-scale initiatives And EBRA was really paving the way on the basis of a very careful analysis on the key priorities that need to be pursued for the future, and also actively engaging at a global level. So I know that my time is up, so I'll finish here, and I'm happy to contribute to the discussion afterwards. Thank you very much for your attention.
Thank you very much. Very important points, very intense activity. I would like to move now to Professor Chiara Carrozza. I don't know, because I know that there was some problem. Are you there? Okay, perfect. Yes. Chiara, hello, hi, good evening to you. Chiara Carrozza is again in a very unique condition to provide us her views, because she is the past Minister of Education, University and Research of Italy, and she is now the director of the Italian Research Institute, and she's a bioengineer by formation. So again, he was responsible of at the highest level in our country of the health and of research. So both aspects. So I think your view about what is in your view for neurodegenerative diseases, what can be done, how the government and the institutions may contribute from this point of view.
Thank you very much, Professor Comi, and I have listened to different presentations on different parts of research and outstanding results. What can I add? I can add some answers to the questions that you asked before the beginning of this session. So, what is the critical point that I see in this research? I think we need a sort of global vision, a strategy for neurodegenerative disease. So how to make a sort of roadmap for the research in different for different pathologies, but engaging also public actors and the government and the society to explain what they can expect from research results in the near future and what can be done for that. And explaining that we need fundamental research, so research on the different processes that are involved to the molecular level, to the studies on human patients, on biomaterial, on protocols, but we need also a vision for translational research and a vision for rehabilitation, because I think we need more practical guide for clinical facilities facing every day with patients and needing some response and some protocols to be applied in a short time. So we need a long-term vision, but also a short-term vision, and also for the support for patients and the new technologies that can be involved in rehabilitation and in the treatment in the long run with an interdisciplinary view. So I strongly support your initiative and also the idea of having a sort of roadmap to be published in order to understand the achievements so far and what is needed. And the second, an advice for Fondazione Prada and for you all for what to do in future. So I think we need infrastructures. I already listened to different speeches speaking about infrastructures. We need research infrastructures. That means systems and methodologies to share and put data on databases that can be used and also analyzed by different groups. And infrastructures are fundamental. I come from high energy physics, so I was trained at CERN, and CERN is a place where all physicists all around the world can make experiments and can make research. They research, but there are common projects going across different countries and engaging different research community in supporting the infrastructure. So I think that the infrastructure is the key word today. And second, I think we need more interdisciplinary view, so more exchange with different community. For example, bioinformatics, bioengineering, and artificial intelligence, and so in a systematic way. So not with individual approach, but with a collective approach, because infrastructure are based on the idea of engaging a sort of roadmap of an area and a common strategy with international agreements. And then I also suggest you to go to in silico medicine. That means to accelerate research results and results from clinical trials and from basic research. We need more computational power and more initiative in order to support the idea of developing models in silico. In silico means in simulation, in order to accelerate research and exploration of different therapies and different protocols. So I was advised that my time is off, so I can thank you very much for your attention.
No, thank you so much to you for being with us and to provide us so relevant suggestions. Thank you so much, Chiara. I would like to ask now, we move now to the industry and to biotech company, but before I would like to ask someone who has the responsibility to be really the communication point with the industry, Marco Salvetti. Marco Salvetti has the role for MS in the Progressive MS Alliance. He is the chairman, co-chairman, we see the other chairman in a minute, of the Industry Alliance, which is an entity that has been created with the aim really to facilitate the interaction of industry. That worked quite well. We will listen then your voice also about this point.
Thank you very much, Giancarlo.
And of course, sorry, just to say, Marco Salvetti is professor in neurology, chairman in Rome at the Sant'Andrea Institute.
Thanks, Giancarlo. I will focus on just one problem and offer two. I'd like to say solutions, but I will just offer two proposals. The problem is this one, which is well known, and in fact many leading journals publish also complex models to try to solve the problem of translating preclinical models to human, to clinical research. But besides these complex translational procedures, there is some methodological issue that it could be quite simple to solve. And the methodological issue, in my opinion, is this one, is that when we test therapy in the animal models, we look at biology and efficacy, but then when we go to clinical research, we somehow forget the biology, in spite of the fact that the two biologies are different. So this is a paradoxical situation. And we look for efficacy. Also, these days we have concentrated about biomarkers of efficacy, but we should also concentrate about biomarkers of biological significance and biological meaning. So, as International Progressive MS Alliance, this organization, among his future initiatives, is considering whether and how to try to foster research in what we call experimental medicine trials. In other words, early phase trials that focus from tests of efficacy to studies of disease mechanisms. Something that is informative even in the event of a negative outcome. Yesterday, Professor Lassman said that we should publish more negative results, but this is very important, but it is important as well to have to be sure that these results are really negative, because they may be clinically negative but biologically informative. And so if we don't have the biological information, we are losing key points. And the paradox is that very often in phase 4 clinical trials, we re-investigate the biology of the treatment but it would make much more sense to investigate the biology of the treatment in earlier phases of research. And the second point, the second proposal is that, yes, there is a lot of research on cross-species translation, but there should be more research in cross-disease translation, because there are the biological basis to do this, especially as far as the commonalities on the genetic basis of common multifactorial disease and rare Mendelian diseases. There are recent and very elegant demonstration that these commonalities exist, and in our disease, Sergio Baranzini, who we heard yesterday, also demonstrated that this is true, for example, also for progressive multiple sclerosis and hereditary spastic paraplegias. Thanks a lot.
Thank you. Thank you very much. The point that you have raised about the necessity to support clinical evidences with what is behind the effect, I think is a very relevant. It emerged a bit today, but it was very important to emphasize this point. Now, I would like to ask to come Chiara Liberati. Chiara Liberati is a biologist as a formation. She's at present Director of Discovery Research at AXAMS, and she has been involved in the initial phase of connecting scientific discovery with the process of advancing then the development of potential treatments successfully, in some cases, I have to say, and particularly in the area of neurodegeneration. So it's up to you to tell us something about your type of point of view.
Okay, thank you. Can I still call you Professor Comi, or should I call you Giancarlo?
No, Giancarlo is fine.
Okay, Giancarlo, so thanks for the nice invitation. We didn't talk so much in these 2 days of drugs, how drugs are identified and then developed. So I'm here to give you my contribution on this very important topic, which is how to identify meaningful molecules that you know better than me, with a very low probability of success, maybe from here in 10 years after painful drug discovery and expensive drug discovery could have the chance to reach the market. But before that, could have the chance to reach your hands for clinical trials. And AXAM is really positioned in the very, very early steps of the drug discovery. Towards about the company, for those who are not familiar with, first of all, we like to call ourselves innovative partner research organization and not just a simple CRO, contract research organization, because we really believe in innovation and in innovative ways to make early drug discovery. And our key expertise is really identification and generation of bioactive molecules, applying high-throughput screening. So you know that drug discovery, modern drug discovery starts by testing libraries of molecules in a different kind of assays, typically in vitro assays. And these assays also during years have evolved, try to be more meaningful and looking for meaningful, more meaningful endpoints than to be relevant in vivo and in the clinics. We are based here in Milan. We are more than 180 employees now. And I would say that we are really focused on having scientists, so people coming from academics with a PhD. We have 60% of employees with a PhD. And our roots are in big pharma. We are a spin-off of Bayer, so we know how to make drug discovery thanks to the experience we made as a Bayer group in Italy. So our ambition is to provide good molecules to biotechs and pharma, as a starting point then for optimization with the drug discovery programs. Personally, I'm responsible for proprietary discovery programs because we take advantage of this technological platform, which is based on high throughput screening, also to run internal proprietary programs where we invest, we decide the target to work on, and we develop the molecules in collaboration, for example, with academics. And these programs are 100% focused on neurodegenerative diseases. That's the reason why Professor...
May I ask Chiara, trying to take some advantage for your presence, where you see the difficulties in this very initial phase? We have a lot of compounds. How do you suggest, or which is your recommendation, if we have to try to facilitate such an acceleration? Is there a bottleneck that has to be in some way?
Absolutely. Some of the bottlenecks are on this slide, on the next one. The first one is funding. So we touched that. I don't know if Professor Carrozza is still online, but you know better than me that this research is very expensive and funding is not well adequate. So we cannot rely on public money. We have to put a lot of creativity in fundraising. And this is what we made successfully. You can see here, we founded 5 companies, new companies, so biotechs in the last years. The first one in 2016, Acusia, a German one. And I can tell you they are running phase 1 clinical trials with a molecule we identified in our labs in AXAM 5 years ago. So 5 years from HTS to clinics phase 1. Hearing loss.
So the time is the second problem.
Yeah, but in this case, we were very fast because of different know-how that we put there. Second, Rewind Therapeutics, and I'm very happy that Catherine is here because Catherine is in the Scientific Advisory Board or the Rewind Remyelination, and also you, Giancarlo, are there in the Scientific Advisory Board. Then Libra, Amyotrophic Lateral Sclerosis, 2019, MUNA, 2020, and the CSO of MUNA is here, Nils Plath, and he can give you suggestions from a biotech point of view. And lastly, Lario. And also Lario is a very interesting case because they are dealing with Parkinson and epilepsy. We neglected a little bit epilepsies in these 2 days. But I can tell you that Lario is born thanks to some confidential information we got from a PI who identified a relevant target for a disease, and she shared this information with us, to us, before publication because she trusts us, and we developed the molecules together. And this is a provocation to Catherine. If you have good targets, good idea, don't be afraid to come to biotechs, and we can find a way to develop them together. Because I think this is very important. Basic scientists, they are sometimes afraid or scared about biotechs and big pharma, and they don't talk to them for several reasons. So this is one of my...
Very positive message. Thanks so much. Thank you so much, Chiara. And I would like to ask now to Lucia Faccio. Lucia has also a double experience because she's been for a long time the director of research and development at Fondazione Teleton, Italian Teleton, which is the major research source of financial support for research in genetic diseases, particularly hematology. And now she has a different role because now she's a partner of the Safinova Partners, which is a European venture capital, one of the most important. So we are talking about money today. Money matters a lot, of course. So, and again, for you is how do you advise how to try to speed up the process and particularly how to succeed to find money?
Thank you, Professor Comi. Really glad to be here. I have to say that these 2 days have been amazing in terms of, suggestions, potential opportunities for collaboration, for exploring different ways to basically try therapeutic, try to find therapies for neurodegenerative diseases. And as a venture capitalist, now I'm wearing this hat. It's really very, very important to accelerate those innovations that come out from academic research to make sure that then they can become visible also to pharma companies. Basically, venture capitalists that nowadays are helping and taking that risk that pharma companies are not able to do at a very early stage, and create small startups in which they basically work a lot on the academic knowledge. They put together a management and a competence in terms of clinical and regulatory development that then can make the opportunities appealing to companies. I have to say that obviously neurodegenerative diseases have had a lot of failures at clinical stage, and it was very interesting to see today how the community of scientists and clinicians were really thinking of what do we need to change in terms of how to structure in a proper way the clinical trial, how to identify and to characterize the patients that will be enrolled. All these aspects are the ones that venture capitalists want to see when there is a proposal. So you come with a product and maybe not a product, maybe with an approach to a disease, and for example a target, as Chiara was saying, and then you can try to find the people that know how to develop then small molecules that are able to target that specific target or monoclonal antibodies, or you approach with innovative therapies like gene therapy and gene editing.
To be very a bit provocative, is this a good time or is it a bad time to try to find such a process of financial support?
Okay, let's put it in this way. There were better years than this one. Let's answer in a diplomatic manner. But it's also true that from an investment point of view, at this stage, companies that are at an early stage are somehow more appealing than the ones that are listed, obviously. So, and also the ones that are listed are very much looking for other opportunities to renovate their pipelines. So I would say that the opportunity is there, and that venture capitalists that in Europe are present but not as aggressive, let's put it in this way, as US, are anyhow, I think, a good tool to arrive to a proper evaluation of the companies that then can survive through all the pathway of development. Really, the large evaluation that are in the US countries sometimes make it almost impossible to be able to refinance and to move forward companies. So definitely a good time, and I'd love to be still involved in this discussion. Thank you very much.
Thank you so much. I would like to ask now, we are almost there, so I would like to ask now Neil Platt to speak on behalf of the biotech companies, because here is really the important step in order to start the journey of the magic pill.
Thank you very much, Professor Comi, and thank you very much for the invitation. Putting my glasses
on.
Sorry,
just to introduce you, Neil Platt is the Chief Officer of Muna Therapeutics.
Thank you, yes. And putting on my glasses as a drug hunter, one of the topics I would like to draw the attention to here is really the necessity to develop human-centric disease animal models human-centric models, research models, and I just realized that Professor Salvetti also has already introduced you to this necessity. But I think we've all been in a situation where we have seen beautiful data from drug candidates produced in mostly rodent models that then went on to fail in the clinic, leaving us with a question whether it was the right compound that we tested and the wrong concept, or it was actually the right concept, we just didn't produce the right compound. Nevertheless, however you look at it, we had an issue with translating from one species to the other, mostly from rodent to humans. Now, 10 years ago, we already agreed that it doesn't make a lot of sense to look at the behavior of rodents, fortunately, so we moved into what is referred to usually as endophenotypes or intermediate phenotypes. And this field has really gained a lot of momentum about a decade ago with the advent of the genome-wide association studies, then the exome sequencing, the whole genome sequencing, moving on to expression analyses, really telling us a lot about human risk factors from a genetic perspective. We use this information now to build the right models and to identify a lot of novel drug target candidates that we study, as Chiara also alluded to, in molecular and cellular assays. Those are then taken forward into an understanding of the impact on the brain circuitry. And as was mentioned ample amount of times during the conference, it is then extremely important to build the right biomarkers that address the same biological mechanisms in order to investigate these also in humans, because again, symptoms or even diagnostics we cannot address in animals. So I'd just like to give you an example of how you can actually take that information into a research drug discovery context here on the left-hand side, just taking again the information derived from humans, specifically the multi-omics now, the genomics, but also the transcriptomics, the proteomics, putting that into a context where we can identify drug target candidates, and then study those in, first of all, human-derived cells, which is really an advent with the stem cell biology that we have also now as a tool in our box, and then taking those human cells and actually implant them into the brains of animals, which we can do now, the so-called xenograft implantation, which we also do at Muna, in order to be able to study these cells in an innate context, in the context of brain functionality. The other topic that I'd like to draw your attention to, and that we briefly touched upon during this conference, and where we also use human-centric approaches, is the phenomenon of resilience. We still see, for example, in Alzheimer's disease, about 30% of postmortem analyses where we see very prominent amyloid pathology. However, these subjects have been reported to be cognitively normal. Usually referred to as resilience, maybe it's just a delay in the onset of cognitive decline. Nevertheless, we likely are looking here at phenomena that could be interpreted as either neuroprotective phenomena or resilient phenomena, or actually as phenomena that are prone to lead to neurodegeneration. And what we are doing in the study that you see on the slide here is that we are actually examining the brain, the postmortem brain of subjects both with pathology and the absence of cognitive decline, but also pathology and the presence of cognitive decline. And we apply spatial transcriptomics to actually understand transcriptional, differential transcription processes related to healthy tissue as well as related to pathological tissue to understand which factors might contribute to neuroprotection and to resilience. So all of that to close in saying that it is critical that in drug discovery we are having as human-centric as possible or as practicable models introduced already very early onwards, and that we then can carry throughout the drug discovery stage into drug development as well. Thank you very much.
Thank you very much. We close now with 4 contributions coming from pharma companies. Of course, without pharma companies, no way to come up with a drug available for our patients. And I would like to start with Shapira. Are you connected? Hello, hi, good evening. Now we are expanding to the night, but I think it is very important just to listen what is your opinion about this topic. Shapira is head of science in Biogen Digital Health. Before he was deeply involved in Hoffmann La Roche in development of digital system, which has been, by the way, presented in a couple of presentations today. So, Shibi, what is your view about the future of how to accelerate neurodegenerative diseases?
Thank you, Giancarlo. This is a pleasure to be with you even remotely tonight. And in 3 to 4 minutes, I'll try to share in summary how we're looking at that question by trying recently to embed within a biotech company like Biogen, a digital health research organization, to help accelerate transforming patients' life by making personalized and digital medicine become a reality in neuroscience. Why are we doing that now? Because we believe we're at a point in time where the progress of technologies are becoming exponential and converge. You think about the cost of genome sequencing that has gone from 100 million to less than $400 in 20 years. The power of machine learning and model training has exploded by more than 10 billion times since 2010. It's doubling every 6 months. And we know everything about the adoption of wearable technologies and outpacing internet. So we think that data and computational methods are coming together and may enable to create exponential opportunities to transform the way we think about medicine development. In this respect, and for that purpose, Biogen has created within a biotech company, a digital health research organization, mostly to enable research and development via the development of differentiating personalized medicine capabilities. And I'll give you a few examples of what it means, especially for research and precision phenotyping of disease and enrichment of trials with the right patients for certain solution. One of the things we've done recently is to use a real-world data set of research-grade information, which is called MSPass, which has gathered more than 20,000 patients now from 5 leading MS centers, which we're now starting to use as a discovery data set to understand potential traits of MS disease that could be associated with genetic cues. We're creating Radiomics Innovation Lab. This is something we did recently in collaboration with a medtech company called Terra Panacea to be able to use machine learning on our medical imaging input, again, to develop algorithmic solution that could enable the creation of precision phenotype to enrich predictively clinical trial. And we're equipping our clinical trials with attempt to use digital health technology to develop digital biomarkers. It seems like a buzzword, but it is a long journey to actually establish the understanding of how you can transform the resolution of how we go after functions such as cognition, motor control, or walk in a broad sense. We're trying to be systematic in this respect. We're integrating those technologies in MS, in SMA, in ALS trials, recently in Parkinson's disease trials, and hopefully in Alzheimer's disease one day. In Alzheimer's disease, what we're doing now, and that's in part related to why I'm not with you tonight, is trying to accelerate, and you heard about the good news about lecanemab. In parallel to that, we started a massive effort in collaboration with Apple to test a relatively simple but a bit of a moonshot question, which is, can you actually detect early signs of MCI or quantify the risk of SCI through consumer wearable technologies, so using information collected by an iPhone or by an Apple Watch. This is a long endeavor. We've enrolled more than 20,000 participants in this so-called intuition brain health study with the hope to potentially develop a capacity to early detect mild cognitive impairment at the population level using passive monitoring. Another example of where we play is the field of using digital technology to directly induce a therapeutic intervention in a collaboration with MedRhythms. We're trying to develop the first prescription digital therapeutic that would improve walking impairment in MS using a combination of sensor software and testing the impact of a music-based rhythmic auditory entrainment of walking capacity. So in a nutshell, that's how we think about acceleration, believing that therapy development for neurodegenerative disease will actually augment probability of success somewhere in a place where the exponential progresses of biology and technology may intersect. I thank you for your attention, and again, I regret not being with you tonight in person.
Thank you, Shivi. Thank you very much for underlining that also technologies very important, particularly if we want to detect very early the presence of a disease. And now I would like to ask Jan Shadrack from Roche, where he is involved as a head of the therapeutic area, neuroscience and rare disease for global medical affairs. Thank you so much to be with us and listen to you.
Thank you, Giancarlo. Hello, everybody. It's a pleasure to be here. Sincerely, this has been one of the finest neuroscience today's meetings that I have ever attended. So deep thanks to you, but also deep thanks to all the lovely accompaniment and help from the great Prada organization and organizers. So I keep it very light, no slides, just a few points of perspectives. And I think one main point I thought we should remind how we started ourselves, how we started yesterday in the morning about brain health, holistic brain health. And I think that's something very important to keep in mind, that this is now really the area to embrace it, to make it a new standard, because it's a very fragmented discipline. And I mention this because understanding from brain health before it's diseased and brain health in medicine are two super important parts to enable better societal health. And the societal health, I think, we're in very, very challenging times, I would say, in what is going on the planet. And I think we can contribute as neuroscientists to better understand a healthy brain functioning that also will help, hopefully, to better societal functioning in the future. But just looking at the medicine side of brain health, the burden due to neurological and psychiatric diseases is tremendous. But what we don't know is exact good data of societal burden. And so we are engaging now with the help of the IHME, the Institute of Health Metrics and Evaluation in University of Washington in Seattle, to really enable a brain health atlas regarding the latest data on societal burden due to neurological diseases, and not only the direct healthcare costs, but especially the indirect costs that are emerging due to this disease state. So for instance, in Alzheimer's disease, it's very common that a spouse or child stops working, so that impacts productivity, GDP, and so on. So we really need to understand this data more precisely to also understand how our innovation efforts bring an impact, a positive impact to society. And I think that's also a plea to you as neuroscience crème de la crème experts to keep that dimension in mind beyond the innovation. The innovation in neuroscience, it's exploding. We're very excited at Roche about what's emerging. I agree with the comment that was made that we are at the dawn of a new age. If you think about how innovation has accelerated a few years, it's truly exponential. I think the curve will continue. I'm super excited about the possibilities that lie ahead of us. And a very important point that has been made several times is the power of collaboration. I think that is so key: collaboration, academic centers, and innovation in the industry, but also Shibi and ourselves, or Novartis, Florian and our group. We work very closely together on the disease area level. We're very competitive on our products, on our solutions, but we need to raise the tide in a similar standard, because if everyone develops different approaches, then it's like Apple and Samsung, and you get crazy with all the different cables and stuff. So one prime example is neuroimaging. The algorithms are all over the place. You know, if we start to harmonize the algorithms in neuroimaging just a bit better, then we have better cross-communication, better big data, and can accelerate clinical research and innovation much better. So we heard a lot about basic science. We heard a lot about clinical science today. What's close to my heart is to translate all of this into the reality of medical practice. And that's another plea I have, maybe a suggestion, that while we have sophisticated designs and endpoints and complicated Z-scores and sum of boxes and so on, for us, our challenge is how do we make this scalable? How do we introduce this in the already very challenged healthcare system where there's a limited capacity of neurologists, where there is really challenges in access. And I think looking at now disease modification in Parkinson's, in Alzheimer's, we don't even know how to measure it in real life. How do we know it works? How do we know somebody responds? We probably won't do all the scales and tests that we did in phase 3 studies. So we need to find ways to really bring this into the routine medical practice. So that's something that I think a lot about, and I think we always appreciate input. And maybe two final comments to you ask also recommendations for Prada. Very pragmatically, keep that risotto yesterday. This was one of the very best I ever had. So that was superb. And I hope we can continue this journey. But I think one important aspect we haven't addressed is the issue of stigma. And if we can find ways to, continue to accelerate the science, but also leverage the power of the community and engaging policymakers, government players, patient organization, big names in the fashion industry, in the art industry, to address this huge elephant of stigma in neuroscience conditions, I think we would gain a lot way. And maybe then in our meeting in 2030 at the Prada Foundation, we look back and say, look how much have we achieved. So thank you very much.
Thank you very much. I would like to ask now, we are almost there, Maximilian Schuyer from Janssen. He is the director of MS European Medical Affairs. Nice to see you here with us and listen to your point of views.
Yeah, good evening everyone from my side, and first of all also a big thank you from my side to you, Professor Comi, but also the Fondazione Prada for inviting me here. It's really not only a pleasure but an honor to be here. And well, after 2 days of really awesome presentations, let's see what I can add here in the next 4 minutes. I think a lot has been already shared by researchers, also by industry colleagues. Let me try to summarize it from my side. We talked a lot about it. So the area and body of research here is really difficult to access. Yeah, we also partially discussed about it, but I also worked in infectious diseases and in dermatology. And in dermatology, you can just make a biopsy of a psoriatic plaque, and you can have a close look at it. It's way easier, therefore, to develop drugs. Time of diagnosis, often relatively late in the course of the disease, so there's clear need. I mean, we had, I think, 2 or 3 hours about that. And in today's sessions, there's a clear need for early, accessible, but also affordable diagnostics, and then therapeutic biomarkers. Here, it's nice to develop very, very safe and very precise biomarkers, but they also have to be accessible and affordable. Well, then in neuroscience, again, I worked also in other indications, internal medicine, infectious diseases, dermatology. We have sometimes more indirect and soft endpoints versus, for example, oncology, where you can have a biopsy and very clearly make precision medicine. A lot has been happened there in the last 10 to 20 years. And also, those soft endpoints sometimes make approvals and pricing harder. So especially in psychiatry, I worked partially in neurology, also now soon to transitioning into psychiatry, pricing is super hard. Yeah? And if we don't get adequate prices for our drugs, we can't spend it for future drug development. So it's also mandatory to get an adequate price for it as well. Because of that, because it's so complicated, less companies and are still active and less investment versus other TAs in the CNS area here, as it is lengthy, risky and expensive compared to other TAs with a, in psychiatry at least, currently a relatively limited return on invest. And the predecessor here from the industry, from Roche, the colleague mentioned it, there's still lots of burdening stigma around some CNS and neurodegenerative diseases, and even with primarily neurodegenerative diseases, you have psychiatric symptoms which are sometimes handled with a stigma. So let's now come to the potential solutions. That's my last slide here already. Okay, the format has been a bit this-shaped, but okay. We have to find more accessible biomarkers. We are on a good way in some therapeutic areas here. In some diseases, Abeta 4240 was mentioned, neurofilament light filament chain. And then we have to, and this is super important here, and this is why I also like this meeting here so much, let's talk to each other and not with each other. So quite often, pharmaceutical industry, for example, in the press being depicted very negatively. One is talking about the other. Pharma says, hey, HTA bodies, why don't they reimburse our drugs? And then payers are also involved. And we have a common goal, and this is to improve the outcomes for patients. And if we do that well, everyone benefits from it. So we have to talk to each other. Then public-private partnerships. Diane TU has also been mentioned. We have to set up risk and investment sharing models, such as public-private partnerships here. We could also even involve not only the usual suspects like pharma, biotech, scientific societies, but maybe even politics. That was also briefly mentioned. And last but not least, yeah, bring the burden of neurodegenerative disease more into public and further reduce stigma. And I think this is what we did here actually. And my advice to the Fondazione Prada would also be, it was a great event, do it again, and maybe next time even include HTA bodies, payers, and maybe even politicians. And thank you.
Thank you. So I would like to ask now to close these interventions to Florian von Ryson. I think is really, we left you last, Florian, because you have the two roles, the two heads, because at the same time you have the responsibility of clinical development program in neuroscience in Novartis, but also you are leading the industry forum. So again, two heads, and then a very special possibility for us to reach advice and to listen to you.
Thank you so much. So thank you to you, Professor Comi, and the foundation. A beautiful event, really. I repeat it like everybody else. It's an unusual thinking, and you have even thought inviting pharma. That is very nice. Thank you very much with that. And I start with a little poll. I will see if you're still alive. So please, who believes in neuroscience the glass is half full or half empty, meaning for the future? Race, we start with half empty. Who believes the future is not so bright? Finding solutions, getting it done. Nobody? Come on. Okay, so glass half full? Okay, 10 people are sleeping. Okay, that's good. Okay. So I try to summarize what I can see from my side, and you're right. I have the double head with this the industry forum at the Progressive Alliance. But essentially, what I do every day, and can I have my next slide? There's a disclaimer. I represent obviously my own opinion here, and not. And do I have to click? Okay. So within Novartis, I'm doing drug development, so I'm a hands-on drug developer. And it's funny, I was reviewing the whole day, the 2 days, and this is my daily work. I have teams working in Huntington, multiple sclerosis, Alzheimer, ALS, Parkinson's disease, all. So this was a great refresher. Thank you for that. This was really perfect. It means as well, we need, what we have to do, we need it, and I find the word after my disclosures, we need the powerhouse. We need the R&D powerhouse to make it happen. Meaning we need combine the forces, and as an industry partner, I need to be able to go to my colleagues, or either in a biotech company, I just go to next door and ask my colleagues in oncology, do they have anything I could use in CNS? I can ask my colleagues in autoimmune diseases, do you have an NLRP3 inhibitor? They have. Things like that. I ask my colleagues in neurodegeneration, do we have a TREM2 I can use in another disease? We have. I think this powerhouse concept is super important, that we use the information we have in a big company like Roche or others or Novartis. We can do it within the same buildings almost. And if you're in a more connected world, you have to reach out to smaller companies. But we have to do this, otherwise we will not make it. But I'm really confident we'll do it. I tried to put around the green dots, deciphering it. We need to understand the biology, as was repeated. We need to discover appropriate targets we have. We mentioned the microglia. This is in diseases like MS, Parkinson's, Huntington, ALS. It's always the same question. How can I influence the microglia? Do I understand this? And there's really a hope that the next 5 years will bring really progress into that. And our development pipelines would be different than it is even today. On the top, you see the biomarker questions. We have to do back translation, yes. And we do too much, not too much, I'm not saying. We should do back translation, what we do, but we shouldn't stick there. We should then do this to transform it into really the forward-thinking biomarker development of fluids we heard about, imaging and digital. And indeed, it must be affordable, must be usable, must be simply to use, all of this, but not stick only backwards, because we might go in the wrong direction. That is simply something to think about. Existing data needs to be pooled. And one example, in multiple sclerosis, we were offering to combine all data from, I don't know how many sponsors, we are now 5, 10 sponsors. We want to bring all clinical data from all clinical studies, from relapsing MS, from progressive MS, into one big data pot, which could be and would be accessible to researchers, to other companies, something we work on, and we hope we can get great news in the close future, that we can do that, to build on existing elements already. Collaboration, private partnerships, yes, we need those more and more. We need to talk to each other. Crosstalk is definitely what is the right word here. And we need to have this permanently and repetitively. So to the foundation, I would say, please come every year. I come here every year if you want, and we can have this conversation, the progress we made since last year. It would be great to have. Early engagement was mentioned with patients, with caregivers, regulators and payers. Yes, we heard it very clearly. If we have a beautiful developed compound, it took us 5 years to recruit the studies and $200 million, and it's not reimbursed. This is not really fun. So meaning you have to do this holistically from the beginning. You have all the stakeholders at the table early enough, not too early for some, but early enough they can give inputs, and they agree what you do. And at the end, you need to calculate the overall societal cost you might generate, you might help. A drug is only one part of management of a disease. It's only one part. But we need to calculate what is really the value for societies, what's the value for a system. Does it really help to reduce the use of money? I think that is very important. And last but not least, not necessarily in the order of importance, the blood-brain barrier, or my biggest personal enemy in drug development, because killing so many things. So we need to overcome it. We need to circumvent it. We need to find strategies. We need to find platforms to overcome it, vectorizing antibodies, AAV9 we heard around. We need to develop maybe small molecules which simply cross it, the blood-brain barrier. So this is definitely foreseen as a thing to accelerate drug development, because this is simply our daily hurdle and our daily thing to do. With that, I think my powerhouse is pretty full, so I'm happy to see you back in next year, if it's only possible. And then, yeah, I stop here. I have one slide, but I will stop my talk because it's already 2 minutes over. Thank you so much.
Thank you so much, Florian. So we are really at the end of this very long day. I will not provide any summary of the discussion. Take your own memory of what has been said. And what I can guarantee is that the Prada Foundation will continue to, first of all, to have these interactions at all the levels, and we have already planned to have again this forum in 2 years' time, exactly to be back and to see what happened in these 2 years, how we have improved. Second, we will be in continuous contact with all of you because we will promote some specific actions based on the recommendation that we have received during these 2 days, and particularly during this roundtable. I take the clear message that is arrived. We have to expand to regulators. We tried, but didn't succeed, but of course it's something that we will repeat as an exercise. We are a bit reinforced now by what happened in these 2 days, so I hope that things will be more positive. A couple of notes. For those who didn't have time until now and still have the desire to visit the exhibition, I obtained that the exhibition will be open until 9 o'clock, so people can go and after a glass of wine and something snacks, you may enjoy to visit. Let me just end expressing my gratitude first to all those who have contributed here. First of all the speakers, moderators, because you are the players, and to win a good soccer game, what matters is to have good players in the field. And second, all those who have assisted, and really thank you so much. You have been perfect. Really perfect. And finally, let me really thank Prada family, and particularly Miuccia Prada, for these beautiful days. Thank you so much, and see you in a couple of years. It went well.